Hello everyone, I'm sorry for this. Hello everyone, I'm Ezgi Olgun, IEEE GTÜ EMBS chairwoman. We gather here today for present you the first day of SoScience event. We will be listen our academician for 2 days in this event. We prepared this event with the team, which is 30 people. You will get to know them each in 2 days. They will moderate our guests. The aim of this event was gather scientist developments for the world around the one proof. We want to be united for this event. We were preparing while we got some bad news. As you know, Ukraine is war right now, and we pray for them. We waiting good news as soon as possible. Without further ado, I will leave the word to our moderator, Sena. How are you, Sena?
I am fine, thank you. How are you?
Fine, so thank you.
Hello everyone, from the first session of SoScience, I am Senagül Yılmazel. I am a first-year bioengineering student at Gebze Technical University.
Hi, I'm Sude Usta. I'm a molecular biology and genetics prep student at Gebze Technical University.
Today, computational structural biology at Utrecht University, Professor Dr. Alexander Bonvin is with us. Thank you so much for accepting our invitation. Welcome to SoScience. The subject of the session is solving 3D puzzles of biomolecular interaction by integrative modeling. If you want, you can start, Professor.
Thank you very much. It's always nice to be invited by students to present. You know, that's the greatest honor. So I'm glad I could make it this morning to join you. So I will start sharing my screen. If everything is right, you should now see my presentation for this event. So I'm Alexandre Bonvin from the Biofood Center in Utrecht University, part of the Faculty of Science in the Netherlands. Utrecht is in the center of the country. My name is not very Dutch, if you are wondering. I'm originally from Switzerland and from the French part of Switzerland. So bon vin in French means good wine. So a little bit about Utrecht, where I'm based. So Utrecht is quite an old university. It was founded in 1636. And what you are seeing here is our academy building, the old center of Utrecht. And this is where all official events of the university are taking place, like getting your master degree, defending your PhD thesis. All faculties take place, all defense take place in this old building. So it's a very nice location. And Utrecht is a beautiful city that you should for sure visit if you get a chance. Where I'm located is a bit outside the center of the city, the science park where the university medical center is located. And this building here is the NMR Center. NMR stands for Nuclear Magnetic Resonance. So we're using NMR in this center to study biomolecule. So this building was specially built to host a lot of very high field magnets that you see here in this picture, where we can study biomolecule at the atomic level. And if you watch carefully, you can see me at work in this window here. This is my office. So we have a very strong infrastructure, experimental infrastructure to study biomolecule, a lot of magnets. But I'm not doing experiments, so I'm doing computations. My research area is in the computation area. And so we also have a computational infrastructure in the group. So we have about 2,000 CPU cores that we are using. But we also have access to distributed computing resources around the world. And I'm going to explain you a bit more of that in the second part of my talk. And all those things that we have been doing have been done in the context of several EU projects over the years, which are providing access to this computational resource that are key to serving a large community of users, as you will see. So this is what I want to discuss with you today. Check time, yes. So I will start with a general introduction about biomolecular interactions, and so what is the topic that we are working on and why is that relevant. Then I will explain you how we are tackling the problem of modeling those interactions, so basically solving 3D puzzles in the computer. And I'm going to antibody-antigen complexes, and another one which is basically the result of going into lockdown 2 years ago, which is doing a drug repurposing screen against COVID-19. So this will be more the science part of my presentation. And in the second part, I want to reflect a bit on the path that took me where I am now as a full professor in Utrecht, but also discuss a bit what it takes to do the kind of research that we are doing, say structural bioinformatics, and also, as you will see, providing a lot of services to users worldwide, developing software, but also operating web portals where people can come and do their computation. So this is kind of, what it takes to follow an academic career and to do this kind of work. So 2 parts. So let's start with the science first. So as you're probably all aware, everything in life starts with the genome. So we have here a chromosome, DNA, and this is basically encoding for all the information of life. But at this level, things are maybe a bit boring. So maybe I'm being provocative here, but you can see the genome, the DNA, as the hard drives of your computers. So it stores information, but it doesn't do much on its own. So from the genetic information, protein are expressed, which brings us to the level of the proteome, which is the ensemble of all the proteins that might exist in a cell. And this is where the action is taking place. So you can see the proteins as the CPUs of your computers and the DNA as the hard drive. And you need both for a computer to work, so that's clear. So proteins are very important because they are involved in all kinds of biological processes. So they are going to make molecule, break molecule, they are going to recognize signals, they are going to give signals, they are small factories. So this is really the workhorse of life. And if you want to understand how things work, you have to look at the protein level, how these work. And proteins do not work on their own, but they work by interacting with other proteins. And that's a key feature of most biomolecules. So we're speaking of proteins, but even nucleic acids, RNA, everything, they don't work in isolation. And those interactions are extremely critical. So we started with the genome. You go to the proteome, which is the ensemble of proteins that are expressed in a cell. And the next level of organization will be the interactome. And the interactome is basically the map of interactions between all the biomolecules of the protein in a cell. And so that's a complex network where the connections are interactions, the points are the molecules, the molecular components. And if you disturb this network, miscommunication in network basically is often the cause of disease. So point mutations that might, of course, unfold the protein, but that might also affect the way that they are communicating can lead to disease. So understanding how molecule communicates, how they interact with each other is extremely important. And this has many applications. So looking at structure of proteins, of biomolecules, experimentally or in a computer, it's important for understanding the impact of mutations on disease. You might want to learn how a molecule works and how to engineer them if you want to make better molecules for materials. You can also have those for, of course, health application, things of antibodies, so biological. So often we think of small molecule as the drugs, the medicine that we're taking, but now we see more and more that proteins are actually entering the market as drugs. Antibodies are a good example of that. And there's also a lot of work going on for food applications. So in the food industry, think of crop here. You want to make your corn grow better in salt condition. You want to protect your tomatoes from pathogens. So there's all kind of engineering applications that can take place there. And knowing the structure of proteins and knowing how they interact also give you a starting point for drug design and to combat disease. So structural information here is key. So I explain you, so we started at the genome, we went to the proteome, and went to the interactome. And the interactome can be seen as the social network of proteins. And what you are seeing in this picture is the center of Utrecht, where there is a canal going through the city. That's the old Rhine, actually. And what distinguishes Utrecht from other cities in the Netherlands and Amsterdam, maybe a lot of you know Amsterdam, have been in Amsterdam, is that we have these terraces at the water level. And this is where a lot of social interactions are happening at the macromolecular level. And in the cell, you also have social interactions, but then at a microscopic level between proteins. Okay. So again, the relevance of studying biomolecular interactions, protein-protein interactions should be clear by now. So how can we study those molecules to get atomistic insight? So we want to understand at the atomic level how they are built and how they work. And there is, which brings me to the structural biology of interactions. So we're not going to see all the methods here, so I'm just using a few names. So if we look on the right side, this is the experimental side. So there are experimental methods that allow you to basically get three-dimensional structures of protein at atomistic level. The oldest one that was delivering structure, that is delivering structure, is X-ray crystallography, where you basically crystallize your molecule, and using synchrotron radiation, you can get the position of basically the electron, the atom in a system. And then comes nuclear magnetic resonance. So this is the center where I'm located. So we can study molecule in solution by looking at what we can measure by NMR are distances between atoms. And then you can transform those distances into 3D object of protein. So these are the classical, oldest structural methods that allow you to solve structure of proteins. In the last 5 years, probably, or 10 years by now, cryo-electron microscopy has been booming in terms of solving structure because of technical revolution in the field. And this is now a method of choice to look at very large assemblies. Looking at structures, isolated structure, is in general easier than looking at complexes, so assemblies between structures. So these are the three classical structural methods in structural biology that allow you to get atomistic information. Next to those, there is a lot of other methods, some of them you might have heard of or not, that are giving you small pieces of the puzzle. So some methods might tell you that one amino acid is very important for the interaction, but they don't tell you what it does. But you just know that for some reason it must be involved in a binding. Some methods might allow you to measure a few distances. Not enough to build from scratch the structure of your molecule, but valuable information maybe to model the complexes. Some methods are giving you information about the shape of those molecules. So you don't know the atomistic details of the molecule, but you get an idea of what is the shape, the globular shape of the molecule. So all these are basically small pieces of puzzles that you can use in combination with computational methods to build models of those large macromolecular machines. So we are moving from the right to the left here, from the experiments into the computations, so the in silico world. And here there is a number of methods that allow you actually to generate models of proteins when you cannot study them experimentally. Some models are using homology to known proteins. So we have the protein database, which has existed for 50 years. So we celebrated last year the 50th anniversary of the protein database. And there's more than 150,000 structures of proteins in that database. So if you find proteins that are related to your protein of interest, that are resembling your protein of interest, you can try to model it by homology to what is already known. When it comes to complexes and interaction, this is much more difficult because there are much less structures of complexes than there are structures of components of those complexes. And there are other computational methods. But today, what I want to focus on is docking as a computational method to basically solve 3D puzzles of biomolecules in a computer. What I should also mention, and maybe some of you are aware of that, is that we are in the era of artificial intelligence, and Google DeepMind, actually DeepMind is a company, a sub-company of Google, made a major breakthrough in the last 2 years in terms of predicting the structure of proteins. So they started with a method, it's called AlphaFold, and then this AlphaFold 2 that came, which is extremely powerful to predict structure from sequence. There is even a database now available where they are publishing 100,000 of proteins for which there were no structures, where they have models. And now AlphaFold is also being applied actually to the prediction of multimers, so assemblies. So artificial intelligence is there and it's going to remain here. And there's a lot of exciting work being done. It's probably not going to solve all the problems, but so we'll probably need still the classical computational method, then also, of course, the experimental method, but AI is there to stay. I'm not going to go into AI in my presentation. So what is docking? So how do we solve 3D puzzles by molecular docking? So molecular here refers to molecules. So we want to understand how two proteins bind to each other. You should also be aware that many complexes that are formed in our cells consist of more than two molecules. So the complexity of the problem is larger. So here we are speaking only of two molecules for now, but many complexes have many components. And this makes the modeling, the solving the 3D puzzles much more challenging. So what is the problem? What do we have to do? So if we know the structure of the proteins, of the components of the complex, and we can model them, what we have to do is to figure out how they will bind to each other. How do they assemble into a complex? And what you can do for that is you could basically fix a protein in space, in your Cartesian space. So assume that the blue protein is fixed in space. And then what we have to do in a computer is basically sample all the possible rotation of the orange protein here. So you're going to rotate around the X, Y, and Z axis. And for each rotation that you sample, you have to translate this molecule in 3 dimensions around the blue one. So you're going to translate along X, Y, and Z for small steps. And every time you're going to measure with some function, how good do the molecules fit to each other. So what is being used in this function to basically measure the quality of the fit? There are different factors that enter this. So one of it will be the complementarity of the surface. So do they fit nicely into each other? And we can measure that in some way in a computer. So that's like a geometrical fit. It should fit, like a hand should fit in a glove. But next to that, you can add the chemistry and the physics of the system, and you start. I don't have many equations in my presentation. But what you see here is the Coulomb electrostatic potential. So that's simply a law of physics. Those molecules, those proteins that we are looking at, the atoms in them have small charges. So some of them are positive, slightly positive. Some of them are slightly negative. And if you have charge particle interacting, then the Coulomb law basically is describing you how these interact. So if you have a positive charge will be attracted to a negative charge. And if you have charge of the same sign, that's a repulsion. So this gives you an energy. So you can basically measure the energy of the system. And the other part, which is often used in this kind of function to decide if the fit is good or not, is the van der Waals interaction. So there are forces between molecules that are also uncharged that are causing the atom to be attracted to each other. Think of helium as a gas, or argon, which is a noble gas. But argon, it's not charged, but it can exist in a liquid state. So if a molecule, or argon is a single atomic, is a single atom, but if a molecule or an atom can exist in a liquid phase, there must be attraction between the atoms to cause this liquid phase. Without these attractions, everything would be in the gas phase. And for uncharged particle, the van der Waals interactions are basically describing this kind of attractions. So this is a classical representation of the system. So you're going to generate a huge amount of models, and for each model, you're going to calculate some score to decide if it's good or not. So in terms of methodology, what we have to do, so we have some kind of an energy landscape, or I call it conformational landscape here, but you could also describe it as the interaction landscape between the molecules. And you have to sample this landscape. So you have you don't know how the landscape looks like a priori, so you have to sample it blindly. And you can do that systematically, as I was explaining, rotate systematically one molecule, translate it, measure the energy. And on the y-axis, what you see here is this kind of function that measures how good the model is that I just generated in a computer. So the sampling phase is basically populating this landscape. And what we call scoring in this field is basically measuring how good a given model is. And the assumption, if your function that you are using makes sense, are accurate enough, the assumption is that the lowest energy state of the system will correspond to the real complex. So that's, in a nutshell, how docking works. Now, if you have experimental data, you can also try to use those data to bias the sampling. In that case, you might not have to search the entire space, but you might concentrate the search in some regions. And you might also use the data to actually change the energy function that you are using to assessing the quality of those models. And this is the scoring part. So to come to integrative modeling, we see these days more and more that we are studying very complex machineries in the cell. So a large molecule that consists of multiple components that might not always be very well defined in terms of 3D structure. You have to realize also that proteins are not rocks. Proteins have an internal flexibility, and there are also regions that do not have a well-defined 3D structure. So all of this complexity makes it very hard to study them by experimental methods. But there are many methods, and I mentioned, X-ray, NMR, cryo-EM, but there are also other methods which could be as simple as doing mutagenesis, running a gel to look at your protein. So many methods are providing you pieces of the puzzle. Could be a few distances, could be identification of one amino acid being important for the interaction, but also bioinformatics and evolution, conservation of amino acid across evolution is also an important source of information. So if an amino acid or residue on the surface of a protein is conserved through evolution, there must be a good reason why nature does that. And that reason is often that this amino acid is critical for the interactions, for binding to something, or maybe it's part of an active site to do some kind of reaction. So you can also extract a lot of information from bioinformatics, from sequence analysis. And since 2000, the human genome has been sequenced. We have a lot of sequence information. This is also the reason why artificial intelligence has progressed that much over the last year, because of the access to this huge amount of sequences from many, many organisms. So what you do in integrative modeling is combining this information from different sources together with computational methods to generate your models. So you integrate information from different experimental sources or bioinformatic sources with computations to get to the final 3D model. This brings me now to Haddock as an integrative modeling platform. And Haddock is the name of the software that we have been developing for more than 20 years by now. So the original publication of the software, of the ID basically, was in 2003. And what distinguishes Haddock from other methods, and for sure at that time, was that we had found a way of incorporating information which is kind of fuzzy information. So again, I explain you, we might know that an amino acid is important, but we don't know what it does. So can you use this information to guide the modeling process? And to do modeling, what you need to do is to be able to define this function, these energy functions. You need to define those in a mathematical way. Mathematics is the language of science. And if you want to develop software, you need to transform your problem into a mathematical description. And with that, then you can program it. And we basically found a way to encode this fuzzy information into some energy functions that will basically be optimized in a computer so that the final model satisfies the data that you put in. And since the original publication, where we started from NMR data, being in a large NMR lab, we have been expanding the capabilities of Haddock to account for a lot of different sources of information, including this kind of shape information that might come from low resolution cryo-EM, but also small angle X-ray scattering. What also distinguishes our approach from other software out there is that we can handle more than 2 molecules. There's a lot of excellent docking software out there that have been there well before we were entering the field. But many of them are limited to 2 molecules. So you have to model 2 molecules, and maybe you can take the outcome of that modeling and then add a third one. but there are not so many methods that can model simultaneously several molecules. Of course, the more molecules you want to model at the same time, the more information, the more data you need to bias the modeling. Further, I told you that proteins are not rocks. They have flexibility. So in Haddock, we allow for flexibility to basically optimize the interactions. So how do we describe molecules in a computer, basically? So we need to describe so we are describing atoms, and we're going to work with atoms. But then we need to describe how those particles are interacting with each other. Okay, so we need to transform our chemical knowledge of proteins, of biomolecule, into something we can work with in a computer. And this is basically done by defining some kind of energy function that represents the system. So we're going to describe bonds between atoms. So you see here 2 atoms. We're going to describe the angles. We're going to describe rotations around those bonds. And we're going to describe those interactions that I just described to you before in the docking, when introducing docking, electrostatic interactions between charged particle and van der Waals interactions. So this will be the chemistry / physics of the system. And to this energy function, we're going to add additional terms that represent the experimental or bioinformatic information that we have. And then what we have to do is an optimization problem. We want to find the minimum of this function. And there are different techniques to do this kind of optimization problems. And here we use classical mechanics based on minimizing the energy, but also letting the system move in time using Newton's equation of motions. And everyone knows force equal mass times acceleration. And this is basically at the basis of molecular dynamics. So this is how we basically do the search. These kind of optimization problems are everywhere in your life. When you use navigation in your car to go from A to B, that's also an optimization problem, because you enter your starting point, you enter your end points, and there are many routes that will bring you there, but you want to find the shortest route or the fastest route. And there are algorithms, there are programs behind that, there is software behind this navigation software that basically optimize to find the optimal route. So these are also optimization problems. So we use techniques that are applied in many fields, but we apply them now to biomolecules. So I mentioned Newton's second law. So we're basically going to let the system evolve as a function of time. So we are simulating how proteins will move, or the atoms will move in a system. And for that, we use Newton's second law, which says that force equal mass times acceleration. So I don't want to go into too many details, but the force, so force divided by mass is acceleration here. Again, this is things that you have seen in high school. You might have forgotten about it, but this is basic classical mechanics. We know to calculate the force because once we have defined the energy function that describes the system, which I've just been showing you and explaining you, you can calculate the forces that are acting on the atoms by calculating the derivative of this function with respect to the position of atoms. So this is, again, a mathematical function. So once we have defined the energy, we know how to calculate the forces. And once you have the force are telling you in which direction the system wants to move to minimize its energy. And then we use Newton's equation of motion to basically integrate or propagate the system as a function of time. And what you are getting out is basically like a molecular movie of how a molecule or protein will move as a function of time. And you can use that as well to model interactions between proteins. So in Haddock, our protocol for generating models of complexes has 3 main stages. And we use optimization techniques, so minimizing the energy, but also molecular dynamics, where we have a time dimension in those protocols. The first stage, the molecules are considered rigid, and the only things that we allow to change are the translation and rotation of the molecule. In the second phase, we're going to introduce flexibility in the system. We're going to heat up the system so that you can optimize it better. And in the final stage, we do a final refinement, and sometimes we add the water to it. So why water? So you might, we are living in a water world. Okay, 70% of our bodies consist of water. And proteins look the way they look because of, not because only of the amino acid sequence, but also because of the solvent. Okay, so in proteins, you find hydrophobic amino acids, more oily type amino acid in the inside, and you will find more charged polar types, water types amino acid on the outside. And this folding of protein is because we have water mainly as a solvent in our cells. If we were living in a world that will consist of chloroform, proteins will look very different. You will probably find more charged amino acid inside than the hydrophobic one on the outside. Okay, so it's not only the sequence that defines the structure, but it's also the solvent in which we are working. Small side step here. So this is how this minimization works, and you see here what's happening in a computer. Actually, in a computer, we only work with zeros and ones, so we are working with coordinates of atom, and you can visualize things. But you see here two proteins. The initial step, they are separated in space, they are rotated, and then they start approaching each other. And they approach each other because we have defined experimental information. And in this case, these are the spheres that you see here at the interface. These were identified by NMR as being important for the binding. So we don't know how they bind to each other, but we know that those regions should come into contact when forming the complex. So this is this we can repeat 10,000, 100,000 times in a computer. And for each model that you get at the end, you have also an associated energy. And then we're going to select the best few hundred model out of this computing, and then we're going to optimize them, because at this stage the protein are purely rigid. They can move in space, but there's no change in the structure of the protein themselves. And of course, proteins are flexible, and flexibility is an important part of their function. When you give a hug to your friend, you open your arm and you close them. So flexibility is also important there. So at the molecular level, the same things might happen. So you need to adapt the structure a little bit when it binds to its partner. And this is what we are doing basically in the second phase. So now you see a model here, it's moving again, and now you see that the side chains start moving. So we are optimizing the interface. And in the second phase, you will see the backbone, which is this ribbon representation, start changing as well. So I'm doing dynamics, the side chain will start moving now. And you see that the backbone, if you look at this region, if you can see my pointer, this loop is going to flip over to adapt its structure to the binding to the partner. So these are small conformational changes that take place upon binding. So the hugging between proteins, basically. And we might take those models and then add some water in a computer around those to optimize a bit better the model. Here you only see the backbone, by the way, but of course there are atoms over the entire protein. So we work with all the atoms and we slightly optimize. So these are the three stages basically that we are using in Haddock to go from starting structures of isolated protein together with some experimental data in the best cases, or bioinformatic data, to go to the final product, which is the 3D model of a complex. And we have energy functions. So we use energy functions during the calculations, but we also use energy functions to measure the quality of the fit. And at different stages, there are different functions. These are typically linear models, although we're also working now on artificial intelligence to measure the quality of those, training models to do that. And basically, how we measure the quality of the model at the end depends on three major terms. So what you see here in green, so this is the electrostatic energy, and this is the van der Waals energy between the molecule. So this is the interaction energy between the molecule. Okay. The blue term that you see here is what we call a dissolvation energy. And that's the cost or the bonus that you get by removing water from the interface between the molecule. Okay, so proteins are solvated. You have to remove water when they bind in most cases. And this might cost you energy, or this might give you energy. So this is the solvent effect in some way. And the final term in our function describes the experimental information that we have put in the system. So in this example, we'll have a few distances, for example. So this is how we basically calculate the fitness of our model with respect to the chemical and physical rules that we have defined in our energy function, but also the experimental information. So that's a rather simple linear model. Now, all of this is implemented in a software, Haddock, which you can obtain and run locally, but there's quite a lot of computations involved in doing all that. It's not something where you press the button and then 10 seconds later, your answers are there. An average run might take, maybe depending on your computational resource, but if you can use, say, 100 CPUs, it might take half an hour to do a small protein complexes, but larger system might take a few days. So you know, it's more than, well, half an hour. You can go for a long coffee break and come back, and you will get your results if you are the only one using the computers. But often people are sharing computers. So because of that, we also build a web server, a web portal where external users can come and they can submit their data, set up the run, set up the parameters, and we are running the computations on our side for them. And we also guide them in the interpretation of the data. So this is the address where this is running. So we have to date more than 27,000 registered users from over 135 countries worldwide. And so we started the first version of the server in 2008. So the server has been operating for 14 years, and we have served over 400,000 docking runs since that time. And we could do that because we had access to not only our own computational resources, which have been evolving over the years, but also distributed resources around Europe and the world. So we have a lot of, we have access to quite a lot of sites in Europe. I mentioned that we have access to about 100,000 CPUs. So this is kind of grid computing, or you might call it cloud computing. Maybe some of you have heard of BOINC, this kind of system, you know, to search for SETI at home, folding at home. So these are typically distributed computing. So we send the workloads, we distribute them around Europe, but also sites in Asia are supporting us, and in the US. So 60% of all the computing that has been done since 2008 has been run on those resources, and the remaining have been run on our own servers. This is our user map, our current user map. So you see, so I updated that map yesterday. So we have more than 27,000 users. Majority of them are from India. So there's a lot of bioinformatics going on in India. And then come EU aggregated. So those two are about the same. Then we get the US and China. We still have to do some work more on Africa to color the map, but otherwise you can see that it's very well used. And you see some other server here. So we are not only developing Haddock, but we are developing a lot of other tools that all have to do in some way or other with modeling and understanding biomolecular interactions. And as of yesterday, there were almost 600 users coming from Turkey. Actually, I think one registered yesterday, if I'm correct. So you have to register to access to the resources, because we need to know our users also, because this access to these large computational resources. So, you know, how much computing, how well used are those resources? So you see here a timeline starting in 2019 up to December of last year. So we were processing per month, on average, about 3,000 modeling runs coming from external users. And then you see 2020, March, April, we start going into lockdown. That's the start of the COVID-19 pandemic. And we directly notice a large increase in the number of modeling requests. So almost a factor of 3 in the modeling. It looks like you see the COVID waves here. This was the first wave, then this was you know, October 2020, second wave, and then you see the third and fourth wave. So since that time, we started asking users, so the users could tag their submission as being COVID related. For us, it was important to know that, because we are depending on grants from the European Union and the Dutch Research Council to do this work. And it's important to be able to report, you know, the impact and relevance of the work. So we can show that, yes, it's being used for COVID-related research. And about 1/3 of all the submissions since March have been related to COVID. So we see, so we don't look at what users are doing, but sometimes people are asking us questions. So there is a lot of people looking at antibodies binding to the spike proteins, for example, or small molecule drug design against COVID targets. And this is on the right side. You see the number of unique users per month. So okay, how many unique users do we have? Some users will use the server a lot and submit many runs, but so again, it was around 300 up to the pandemic, and then this has more than doubled since then, and it's sustained as more than double the usage. So we've seen a large increase. This also means that this has put strain on our servers, on our computational resources. So we have to, when you operate these services, you have to also adapt them to meet the demand. And at that time, in 2020, there were more sites in Europe that basically gave us additional resources to support COVID-related research. So when we send out jobs into the world, we can tag them as being COVID-related, and in that way you get access to resource that you will not have otherwise. So that's just, and since my group is doing computational work, we were never really affected by the lockdown. As long as the network was up, we could, access our servers, do our work, do our research. So we have only seen an increased load in research work since the beginning of the lockdown, while the experimental groups, at least in the Netherlands, the experimental labs were shut down for several months. So there was no research allowed. People could not be in the lab. We were all at home. I was also at home. My group was at home, but we have been working hard during that period. And this is the number of, just to show you also, this is the number of CPU hours that we use per year on those grid resources. So this is again the COVID effect that you see here. And I checked the statistics for 2021, and we are even higher. We should be somewhere up there. So this is a huge increase, again, boosted by all the COVID research. So Haddock is really our main research project in my group, where we are developing methods to improve all this modeling that I explained to you, and we also have collaborations and applications. But next to that, we are also developing a lot of other software, and all those that you see here are also available as web portals. So we have to operate a complex infrastructure, and they all have to do with biomolecule interactions and basically this 3D puzzle solving. So these have to do with bioinformatics, prediction of interfaces. You might want to understand which amino acids are very important for the binding. Some are targeted to antibodies, some are targeted to cryo-EM. So this is a complex machinery. So this was about Haddock. And now I want to give you just a few examples of what you can do with this kind of modeling. And we're moving into antibodies. So antibodies consist of two chains. So they have this kind of Y-shaped molecule. They consist of two chains, a light chain that you see in a kind of magenta color and a heavy chain that you see in a more grayish color here. And antibody recognize their targets by this hypervariable region. So there are 6 loops in these regions that are strongly evolving. We have a large pool of antibody, and those loops are changing quite fast, and they are changed to try to optimize the binding. So when you get infected by a virus, it takes some time for our body to produce the right antibodies that are going to block the virus. So we know actually where the action is taking place when you look at antibodies. We know that this is in those 6 loops. So 3 loops coming from the heavy chain and 3 loops coming from the light chain. And for us, in terms of modeling, if you want to model how does an antibody recognize its target, this is information that we can use, that we can give to Haddock, because we know that the binding must occur in this region. And we wanted basically to test different ways of using this information and test how well we are doing in modeling antibody-antigen complexes. And if you are doing methodological development, software development, you always need data to benchmark to optimize basically your computing and optimize your methods. So for that, we use complexes for which there are experimental structures that have been solved. where we know the structure of the free form of the molecule, so the starting point for the binding, and we have the structure of the complex as well. So we know the start point, we know the end point. And then the game is to basically optimize your software, your method, so that you get the best result as possible. We tested, there are a lot of excellent software out there. So we tested 4 different software. So this is the work of Francesco, former PhD student in my group. So we tested our own software, Haddock, where we can basically input the information about the hypervariable loops in the antibodies. ClusPro, another excellent docking software, and web server, and ZDock. So these are 2 famous software that allow you to model protein-protein complexes, but they can only handle 2 proteins at the same time. And LightDock is another software developed partly in my lab, mainly by Brian Jimenez. And LightDock has also ways of incorporating information into the modeling process to bias the modeling. But ClusPro and ZDock also have ways of using this information from antibodies to bias the scoring, actually, the identification of the good from the bad model. So what did we do? We tested 3 different scenarios. So this is the antibody, and on the antibody, we know where things should be binding. There's always challenges because the conformation of those loops might be changing. And when things are changing, it becomes very hard to model those. So on the antigens, that's the protein which is targeted by the antibody, we have 3 different scenarios. So here we might say where we don't know anything about where the antibody is binding. So then we target the entire surface. The second scenario is where we have some knowledge where things are binding, and there are experimental data that allow you to map these kind of interactions. So they are not fully accurate, but we assume that we have some knowledge, say, residues within 9 angstrom. And the third scenario is the perfect scenario. You have the perfect interactions, you know exactly where things are binding, but you don't know how. So you don't have the contact, but you know which surfaces are being targeted. So that's the best case scenario, that's the worst case scenario. And then you can measure how well you are doing in these different cases for all the software. And this is the results already of a lot of computing and a lot of testing. And you see a lot of plots with numbers and colors. The different columns correspond to the different software. So the first column is how well ClusPro is doing, then you have Haddock, you have LightDock, and ZDock. What you see on the x-axis in all those plots is the number of models that you are considering. I told you we might generate 10 or 100,000 models, and then you have this energy function to score them, and you rank the model based on this energy function. So what if you only look at the first model? What if you only look at the top 10 model? How good are they? Is there a good solution in the top 10? So this is basically measuring how many models do you consider when you look at the results. And what you see in the y-axis for all those plots is the success rate. And we have a way of measuring, is this prediction good or bad? Because we know the answer. So in short, the more green you see in those plots, but also blue, the better it is. The higher the bars, the better it is. And the darker the green, the better the quality of the model is. Okay, so you can see that, the last part that I have to explain, so we have 3 rows. In the top row, we have no information on the antigen side, so we target the entire surface. Here we have loose information on the binding site on the antigen, and here we have the perfect information. So you can see the more information as we go from top to bottom, in all cases, the more information you can give to your software, to your modeling approach, the better the results become. That's true of all software. But you see that overall, in all scenarios, actually, Haddock is getting the best performance. And this is, to my opinion, because we are using the information that we have directly from the start of the modeling. So we use it to bias the modeling, while some of the other software are only using it to score, to distinguish what is good and bad, but not biasing the modeling. So this is this has been published in 2020 in Structure. So you can basically read everything about it if you're interested. Since then, we've seen a lot of interest also from pharma companies to use Haddock for modeling antibody-antigen complexes. So this was just a short application. So showing you, yes, we can use information, knowledge about where the binding site is to bias the modeling, and this pays off because you get better model. But you should also realize that, when we have very little information, you see the success rates are pretty poor. And this is true of all software. So in Haddock, we have a success rate of only 25% if you take the top one, and this is the best of all. If we look at the top 100 model, we're just above 50%. So the problem of modeling complexes, modeling interaction is not yet solved. And this is even more true when it comes to antibodies. So I mentioned AlphaFold, artificial intelligence. It's getting there as well for complexes, but it's using evolution and conservation as a strong component of the prediction. And antibodies, they are not conserved. The loops, these hypervariable loops in the antibodies, they are evolving fast. And you cannot use the same information to bias the modeling using artificial intelligence. So I think this will still remain a challenge to the field for the years to come. So now a second example. So we're going from macromolecule, protein, into small molecule. And this is the work that we initiated in 2020 when going into lockdown. And this is showing you basically the adventures of Tintin et le coronavirus. If you have thought that we refer to Haddock as being the fish, in English, Haddock is a fish, we like to think of Haddock as being Captain Haddock that you see here, which is the friend of Tintin, the famous cartoon character. So that's why we choose this name. And actually, Haddock stands for High Ambiguity Driven Docking. So it's an acronym. We can use it because it's an existing word in English. It's not protected by copyright. So that's how we think of Haddock. So over the last 2 years, we've heard a lot about COVID and SARS-CoV-2. So probably you know everything already about it. So this was a paper coming in Nature in 2020, explaining a lot of what are the important proteins and how to target them. Since then, there's tons of information that have been published. So this is really just a very short overview. So the spike protein is on the surface of the virus, and this is basically the protein that binds to our cells. And the receptor that the virus is using to enter our cells is the ACE2 receptor. So this interaction is basically the first step that takes place. You might also heard a lot that we look at proteins as this being only proteins, but in reality there's a lot of glycans, sugars attached to these proteins. So it's a much more complex system that we're looking at. So one possible route of blocking the virus entry will be to have small molecules that prevent the binding to ACE2 receptor. So that's one target for drug design. Another important protein in the viral cell cycle is the main protease, because once the RNA material of the virus enters your cell, it's being translated by our ribosome into a long strand of proteins. And you need to cut this into pieces to generate the proteins that the virus needs. And the actor here is the main protease. So if you can inhibit the main protease, you prevent this cutting of the polyproteins. So this is another way of blocking the virus. Okay, and there have been, I think we went in, this was a structure of the main protease of the virus that was released basically in May 6. And, the pandemic started, we ended lockdown in March, and there was a huge amount in structural biology to solve protein, and it went extremely fast. So you have very quickly, you have starting point to do drug design. And another target for drug design is this RNA-dependent RNA polymerase. Okay, so this basically, it produces the viral RNA, which can be used by the host to generate new viral proteins, but which is also needed for the virus to package it and release new variants. Okay, so those three proteins are three of the main targets for drug design. So before going into the small molecule design, I just want to illustrate also how powerful is the knowledge of the structure to understand disease to some extent. So this is work that was done by Joao, and Joao is a former PhD student of mine, and Joao moved to together with Panos, who is yet another PhD student. And he did this work with Michael Levitt, Nobel Prize in Chemistry, 2013. So what did they do? They look at actually transmission, the potential of transmission of the virus to different species. Now you can, so what you see here are in blue are the ones that at the time when the work was done, so this is December 2020, it was published, so they did the work quite some earlier, but what you see here are different species. The blue ones are the ones that are known that could be infected by the virus at the time, and the red ones were known that they were not infected. And then you can ask the question, why is that? So we can take the sequence of the ACE2 receptor. So this receptor is conserved through evolution, through all organisms basically have these. And you can measure how similar are the sequences of those proteins in different organisms. This is what is basically plotted here on the x-axis. So the similarity of the receptor with respect to the human sequence. And if you look at them, some of them that have high sequence similarity indeed can be infected, but you also find some that have rather low or lower sequence similarity that can be infected, and some with low sequence similarity or high sequence similarity like the rat that cannot be infected. So sequence only does not explain you what's happening here. If you instead model all these structures, so you generate 3D model of the spike protein binding to the ACE2 receptor, and you can do that by homology modeling, and then model all the mutations in those sequences. And then you calculate the Haddock score, so the interaction energy between those proteins. So this is the score that I explained you before. And you see, so now the sorting of those species is based on the Haddock score. So the lower the score, the better the binding should be. And you see now that while sequence was not giving the answer, it seems like the score that we are calculating has some predictive capability, because you see that the blue ones have lower scores than the red one in general. Interesting, there's goldfish here, so I'm not sure if the virus will be able to infect the goldfish, but anyway. So adding the structural dimension to this kind of predictions makes you, gives you the ability to understand much better how these systems are working and make better predictions. Now back to the drug screening. So I'm going to show you some results for one of the protein. We targeted all 3 receptors that I show you. And what we did, and what many groups did worldwide, was to basically use those structures to screen for small molecule. Now, we didn't want to screen blindly, so we developed a protocol for screening which makes use of homologous information. So if you have, there's a lot of those proteins in the PDB, the structure are known, and there are small drugs bound to those. So we use this knowledge of other drugs binding to those as information sources to do our screening. So it's another bias that we put into Haddock. So basically, we identify a template structure which resembles the structure that we have to model. This is the work of Manon and Panos. Panos, former PhD student, Manon, former postdoc. We transform the small molecule in this identified template into some kind of 3D shape. And then we define distance restraints between the ligands that we want to model, or the ligand that we want to screen, and this shape. So now it's kind of a free-body docking. The shape is inside the protein, but this helps very much in improving the docking results and improving the success rate as well. So I don't want to go into more details. This was published recently. So what did we do? This was March, April last year. We took, no, 2020 actually. We took about 2,000 drugs that were approved, and we screened all of those against COVID-19. And then you get for each drug, so for all the drugs that you see here, a small subset, you get the score given by Haddock, basically, and we rank those. So you see here the top 10 for against the main protease. Some of these drugs, like if you read EVIR at the end of a drug, it means that it's an antiviral drug. So it's already interesting to see that antiviral are scoring high. And then, of course, you cannot trust blindly on the docking results only. So we gave a set of about 30 of those drugs were selected for experimental testing by collaborators in Utrecht. And what we saw, so this is an in vitro test. You saw that our number 2 hit and our number 5 hits are both showing activity against the protease in the micromolar range. Okay, that's the concentration that you need to basically block the protease. The problem with those drugs is that if you do in-cell assay instead of in vitro assay, they are toxic to the cell. So out of this screen and out of the screen that many companies and many groups have been doing, basically there is nothing that really came out. So there were no approved drugs that were effective against the virus. Also not the one that Trump was advertising. This has all been done in the context of a large European project, together with many different groups across Europe, but also a lot of pharma companies. And we could do this screening quite efficiently by using these distributed resources around the world. So we could do all this computing in 3.5 days, which would have taken much more time if we had to run everything on our own resources. And this is just a snapshot of March. You see here March 11 until March 17 in 2020. That's when we did the screening. And these are all the sites where those things have been running. Now, where are we going in terms of, where is the research field going? So that's a small perspective. So what I show you is try to explain you how integrative modeling works. And this is what this picture tried to summarize. So you see here a mass spectrometer, a cryo-EM machine, you see an NMR machine, you see a biochemical assay, high throughput assay, and you see software. And the combination of all of these basically is what allows you to get to these 3D models of these large and complex assemblies. And the complex that you're looking at here is the 50 years anniversary complex of the PDB. So we created this fake complex just to celebrate this anniversary. But what this picture is also showing you is that these molecules, these complexes, they are not static, okay? They are dynamical assemblies. And what you see at the bottom is kind of an energy landscape that describes this complex. So integrative structural biology is really moving, I think, into in the future into integrative structural biology of dynamical landscape. So we don't want to characterize only one state of the system, but we want to characterize the complex landscape of the system as it changes, as it moves. So this was the science part. So now I still have some time, and I should leave time for questions, of course. That's the fun part of all of this. So now I'm going to reflect a bit on my own experience and show you the path that I've been taking to get where I'm today, and also then discuss, what are I'll show you we are operating, we're developing software, we're operating web portals, we need a lot of computing resource. So what does it take to do all of that? But let's start by having a look at my career track. So as I told you, I'm originally from Switzerland. I became Dutch. So I studied chemistry in Switzerland and got my master. Master was not existing at the time, but I got my master in '89 at the University of Lausanne. And then I went to Utrecht University to do my PhD and got my PhD in '93 there. Actually, the reason that I go to Utrecht was not where I thought, I should go to the Netherlands, and because chemistry in Utrecht is so fantastic. No, I already met in Switzerland my current wife. So no, it's not chemistry, it's not science that drew me to the Netherlands, but it's my wife. Anyway, so I got my PhD in '93. And during that time already, thanks to the supervisor, I had the chance to go to different meetings. And I went at the time to a fantastic meeting, the Keystone Meeting Series, which is in the Rockies in the US, in ski areas. Of course, when you tell to a Swiss guy that you can go skiing in the US, that's very nice. So that's where the networking game basically started. So even on the ski slope, so there were nice breaks in those meetings, but I will be talking to big professor in the field and start the networking. What I also did is during the last year of my PhD, basically in the third year, toward the end of the third year, I took the opportunity of those meetings to also visit a number of labs. So knowing that I was going to the States, I contacted a number of labs that were interesting at the time to say I will be around, could I come visit? And usually the answer was yes. An important point I want to make here for you, if you consider your future careers, what is important when you are looking for a lab, don't look at the name of the university. You don't need to go all to Harvard, Yale, and all those places. I think what is important to look at is the group and the PI with whom you're going to work. So that's the human factor. Okay, so you can do fantastic research in a small university, but with a very nice group, and you can do crap research in a top university. So look really at the people with whom you're going to work. You're going to spend quite some time interacting with colleagues, interacting with a professor. So pick the places where you should have fun in your work. I think that's a very important part. So be which is why it's important if you get the opportunity to visit lab. Okay, there are also other ways of getting visits. If you're, the Erasmus program where you can go for a short visit to lab. So try to connect to the places that you think are interesting. So that's my first message to you. So out of this visit here, I actually visited the lab of Axel Brugger at Yale University during this after the meeting, and he invited me to join the lab for a postdoc. So I spent 2 years from 94 to 96 at Yale University. And for that, I got a grant from the Swiss National Foundation. In 96, after the first postdoc, I decided that I wanted to come back to Europe. I was seeing my career more in Europe. My second daughter was born in the US, so she's an American citizen. And I went to the ETH. Actually, I could have gone to the ETH straight for the first postdoc. So I had 2 offers, and I decided to go for the US adventure at the time first, and knowing that I could come to Europe to a nice place as well. And here I did my second postdoc with Wilfred von Gunsten, working on more molecular dynamics and simulation type of things. And while I was there, that's the luck factor, I guess. At some point, I got a call from Utrecht telling me, well, there's an assistant professor position opening. Are you interested? So this position was at the time not advertised. So that's, you need to get lucky from time to time. And I think that was a lucky moment at that time. So yes, I moved back to Utrecht. And I explain you, Utrecht is this, we are this huge NMR facility there, and operating since 96 as European facility. So this is where the networking became more and more important. So I got involved in European project, got to travel a lot within Europe for all these project meetings. And you talk to people, you learn a lot of people, you understand how things are working. So networking, and you see networking is a team here. It's growing and growing over the years. So 98, I started as assistant professor at Utrecht University. Got a national grant in 2001, so it means I could get my first own PhD student at the time. And this is about the time where the idea for Haddock came. And the idea for Haddock was actually, I got the idea because of a presentation of a PhD student in the lab who was saying, well, I want to solve the structure of this complex, but I cannot manage to get the data that I need from NMR. But I know which amino acids are important for the binding. And this is why I basically, the idea came of, okay, let's try to translate this information to some kind of energy function. And this was the right time. So you also need, if you have a good idea of it has also to be the right time for things. So there are three key components. It should be the right time, because you might have a fantastic idea, but nobody's interested in because it's not the right time. At the time, there was a lot of work. The structural biology was moving toward complexes. So it was the right time to come with that idea. It has to be the right location. So, there should be the facilities, the colleagues, everything around you should be in place so that you can carry out the research that you want to do, and you should be the right person. I think these 3 points. So I'm the person to do the research, it's the time, and I'm at the right location. So these are things, if you write grants, these are things that you have to show and demonstrate. So I became associate professor in 2003, and got a big grant from the Dutch Research Council, which allowed me to hire 4 PhD students, one of which S.G. Carassa, who is now at Izmir, was one of. So this was a fantastic group of students at the time. And then things, start working. And you see here, so this is the number of citations since 2000. So you see, before I was doing some simulation work, you see, from 2003, this was the right idea, then things start going up quite significantly. And this is, again, 220, 221, we see again an increase. And I think that this is, again, a COVID effect, because many people are using our tools to study COVID-related problems. This leads to more citations of the tools, of course. So I became full professor in 2009 in Utrecht, director of chemical education. Research is the first part. Teaching is also an important part of what we are doing. Actually, when you are working at universities, teaching in principle is the first task. We don't have a very heavy teaching load, I would say, but I like teaching. You get in contact with students, you can also attract students. So I took the job of director of the chemical education for 3 years. So that's, of course, a lot of work, but that's part of the job. And then started working a lot in European networking. So I coordinated an EU project. So these are all the VNR project, and this is the project which is at the core of all the computational resource that we have access to and all the portals that we are doing. And during that period, as coordinator, you have a lot of networking. You have to go to Brussels a lot. So you play the networking game. So again, it's building the network, building your contact, knowing what's happening, getting involved into project, and it pays off. And since 3 years, or well, 2.5 years, I'm now a scientific director of the Biofood Center for Biomolecular Research, which combines NMR, cryo-EM, mixed-ray crystallography, biochemistry, computing, all of that. And in that role, I'm also vice head of the chemistry department. I used to travel a lot. So all this networking, you don't do it from home, of course. So I used to travel a lot to go to conferences, to give talks. I like also very much to do workshops, organize workshops. I organized an EMBO workshop in Izmir, but because of the COVID, we had to do it online. But we hope to organize it again probably next year in Izmir together with SG. So these were my travel statistics. So, you know, that I was almost 5 months traveling, 150 days of travel. That's a lot for 2019. A lot of kilometers, so maybe not very sustainable if you think of it now. But of course, this was my travel in 2020, which was pretty much zero. So those 2 trips were just before the lockdown. So, and last year has been pretty much the same. So you can do the networking, you know, online, but it's not the same. You know, going to conferences, going to workshop, if you get a chance, is important, because you can ask all these informal questions to the PIs that you encounter, having a cup of coffee or beer in your hand in the evening. So that's much more informal. It's not the same that doing things online. So if you get the opportunity, do it. So now for the last part, I want to discuss a bit, you know, what is important for the kind of work that we are doing. So what are the challenges? So we have been now operating these services, this web portal, for more than 10 years. So we started having the Haddock portal in 2008. So it's almost 14 years now. And so you need different key things to be able to do this kind of work. And this was very much made possible by several European projects and all the computing resource that we got through those projects. So this is, again, what I'm speaking about. We started in 2008, and now we are here 2020, 2021, where we have more than 50 million CPU hours that we are consuming per year, serving for free a community of users which is worldwide. So users don't pay for using the resources. So this has to come from somewhere. Okay. And this large community that we are serving also, you know, it costs efforts. You have to build it up and you have to support it. So all of that takes efforts. So what is important in here? So this is just the landscape of European project over the years that has been evolving. So we started with VNMR, and then the next project came up, and then a number of other projects comes up. So all of this, you know, you are delivering service in this project. You have to do work. You have to write reports. So by doing all this work, you get also invited to the next project, or you contribute to the next project. So you have to do good science, but you also have to do your network. You also have to do all the more the administrative parts that come with all of this. You know, if you are reliable partners, people want to have you in the next project as well. If you don't deliver, then you are in for one project, but the next one you are left out. So this is, again, the networking aspect. So over the years, we have more and more projects that are supporting our efforts. And all of this is built around this VNMR virtual research community. So VNMR is quite a well-branded name in EU landscape, but also worldwide. So there's a website, vnmr.eu, where we are basically explaining what VNMR is about and describing also the services and the portal that are operating. And now currently we are part of this European Open Science Cloud initiative in Europe. So we are well positioned there. So, what are the challenges and the solution to all of that? So if you are more in the software business methodology, I think if you develop software and nobody is using it, then it's a pure waste of resources. So open science is important, open software is important, share your data, attract your users. Without users, it's a waste of money. And to attract users, you have to offer them top-of-the-line solutions for their research, okay? Which means also top-of-the-line software. And you will have to provide training, tutorial, and support to your user. You cannot put something out there and say, well, figure it out how it works. They might try to figure out, but then if it doesn't work, they don't come back. So they use it once, they don't come back. So if you want really to be successful in attracting a large community of users, you have to provide training, you have to provide tutorials, and you have to provide support. So we have all of that. So we have a support center, we have online tutorials, we have a YouTube channel where we sometimes record tutorials, and we have been organizing many workshops over the years. Actually, the first Haddock workshop ever was organized in Istanbul at Boğaziçi University, together with SG. So this was the launch of a long series of Haddock workshop. On our website, we have many tutorials where we guide basically user on making the best use of the software. We also have a forum where people can ask questions, and so it means that I'm also myself answering quite a lot of emails and questions on a daily basis. That's part of what makes the success of Haddock, I think. You should also make the life of user easier, if possible. So it means that you have to hide some of the complexity of your software behind the portals. You have to help them to interpret the data, provide maybe single sign-on solutions. We keep monitoring satisfaction of users, also for reporting purposes. So we have this smiley monitoring on our portals, and you can measure. So 5 is the best, 1 is the worst. So you can see that we are doing a good job in general at keeping our users happy. So next to having users, you need also an infrastructure to be able to run all this computing. Okay, and that's important. So we've been in production for more than 10 years, and this infrastructure has been coming out of all this Europe project that we have been involved in. And we have agreement through EGI. We are collaborating a lot of EGI, which gives us access to 50 million plus CPU hours, and it's opportunity access. So it's not reserved resource, but we are making good use of that. And then, of course, you need to attract users, you need to have access to the computing part, but you also need to develop software and maintain a complex infrastructure. So having a piece of software is one thing, having a web portal, operating web portal is another thing. I don't want to go into details here, but this is what a user will be seeing. He inputs data and he gets results back. And behind that, there is a complex machinery. And you need to operate that machinery, you need to get the expertise, you need the people who have the expertise for all of that. So it's a lot of work, but also funding. So to finish with, what are the key success factors, in my opinion, to do all this work that I've been describing? I think the most important one and first one is what I will call the manware. Okay, we have hardware, software, all of that, but the manware. So it's again the people. I told you, if you want to look for your next PhD or postdoc position, look at the lab, look at the people, look at the PI. So it's very important to select people in your groups that are fitting in a team, that are team players. So we're going to spend a lot of time together, working together, and it should be fun working together. So that's yeah. So I like to always see people in person, if possible. When I'm hiring someone, I ask them to give a presentation. It doesn't matter that much what they talk about, but I just want to see them in action and see how the interaction is with the group. People will be talking to all my group members, and I'm getting feedback from the group members. So you want team players, basically, and that's important. You can do great research with a small group of very good and dedicated people. You don't need to have a huge lab. I never had a huge lab over the years. I think maybe I had at the maximum 5 PhD students and 5 or 6 postdocs, and then you have master students. But so it's fluctuating. It's hard to have a constant number of people over the years. But the manware is probably most critical. So that's the human factor. Of course, if you are doing this type of work, so software. So you want to have, so we have our own software that we develop, so that's key to our operation. So we need software, and we need to keep the software to the top of the line. Because as soon as your software starts to become outdated, you don't develop new research with it, new methodologies, then at some point the users are going to drop out. So if you want to keep things to the top, you have to constantly improve your tools. We need hardware, meaning computers. All this, I show you that we are doing a lot of computing. So without the computers, we cannot do that. So that's also something that you need to realize. And for that, you need, of course, money to pay people, money to pay the hardware. We like to have the hardware under our own control. So we don't want the university IT to tell us what to do and how to do things, because when you operate these very complex machineries, you need to be able to fine-tune the system. And finally, you have to be willing to provide support. Okay, I told you, without users, we are wasting resources. It means that we need to support the users. And this is an effort that it takes. And this is also part of the success. So manware will be for sure the number one, the people you are working with, and then all the other components are important. And networking. So to achieve all of that, the networking is important. So going out there, meeting people, talking to people, having informal contact with people, that's extremely important. And you can never start soon enough. So do that during your study already. Take all the opportunities that you have to do that. With that, I'm coming at the end of my talk, so I want to thank the people over the years that have been contributing. This was a view of so this one is probably 3 years old. Of course, before COVID, view of the group. We have many projects over the years that have been contributing to all the work that I've been presenting you. This is more the kind of group meetings that we have been having over the last 2 years. So we are slowly going back to normal now. So all the people in a group. Again, all the project with BioExcel, which is a Center of Excellence in Computational Research, European project, which is very important and key to all the software development that we are doing. And with that, I want to finish. Thank you very much for taking the time to listen to me. And I will be more than happy to answer any question you might have at this time.
Thanks so much for this productive presentation. Could we ask our questions? What some more advice would you give to students who want to be interested in biomolecular science?
Okay. So advice for students who will be interested in biomolecular sciences. Yes. That biomolecular science is it's understanding life. It has all to do with disease. So it's a very important field of research. We see in our university, health is one important thing. Food is another important thing, and then there's all the sustainability. Other things, being so sustainable energy, renewable sources. These are pillars in our university. But biomolecular sciences, if you're interested in understanding life and having a role there, that's important. Data are becoming more and more important in all of that. So also invest in developing skill in data analysis. A bit of programming, or maybe Python or R, these kind of things, because we are moving more and more into a big data era where also artificial intelligence is going to play an important role in all the fields. So I think that's important to develop skills in those fields as well, which you can apply to any type of research that you want. But you see, it's becoming more and more. A lot of research is becoming more and more big data.
It's really helpful for us. Thank you for answer.
And another question from a viewer. Thanks for that precious presentation. It takes a lot of effort to spend so many days traveling for conferences, educations, and knowledges. What was the most efficient conference for your studies so far? Well,
I've been to so many conferences, that it's hard to tell which one is the. I think the conferences that I prefer in terms of are the rather smaller ones. I don't like so you can go, you know, there's a Biophysical Society meeting running this week in San Francisco. There you have several thousand people, so it's huge. Okay, so that's, I like smaller meetings. I like the Gordon conference type of meetings, but also EMBO workshops. So we're organizing ourselves these EMBO courses where you might have between 20, 30 participants, and you spend a week together with all the PIs and all the students. So you have a lot of interactions. You have not only interactions during the lecture and the practicals, but also in the evening you are together. So small meetings would have my preference to the very large ones, because again, the amount of interaction. It is a lot of days of traveling. Yes, I realize that. You have to like traveling, okay? If you don't like traveling, you should not do that, but you need a little bit of that. The good thing when you are traveling is you don't have management meetings and all that things, okay? So this is the boring part. So when I'm flying, I will be, you know, reviewing papers or doing work or preparing presentation. Being away, it's not like you're only going to the meetings. So for the type of work that I'm doing, and I think the last 2 years have also shown that it doesn't matter where I'm always, as long as I'm online, I can work. So, but yeah, you need to like the travel. But you know, the travel, there's conference, and there's always also the fun part of those things. So that's it's tiring, because I would say that you do much longer days. You know, you go to a meeting, you attend all the lectures, you go out in the evening for dinner with some colleagues. So you make much longer days maybe than what you will be doing if you are just staying at home. So it's after a week of conference or a few days, then you get home tired. So it's demanding, and you have to remain sharp. But again, if you have to like it. I like the interaction with the people, so I love the meetings. So it's not. I see now 2 years online. You know, it's tough. If you are attending a meeting online, you can also do something else. So you can check your email in between, or you know, whatever. So you never really fully into the meeting, I think, online. And a lot of the new collaborations, new ideas come by talking informally to people on the side of the lecture in a conference, at the coffee table or in the evening at the dinner. So I think that's something that online is never going to be the same.
Yes. Okay.
Other questions? Thank you so much for presentation. I want to ask, what is the motivation that prompt you to work on these topics?
Yeah, so you know, it's a bit of serendipity there somewhere, because I was working on biomolecules. So I did my PhD in NMR, and I was doing computational research at the time. So I did a lot of programming to solve structure of proteins. So at the time, you know, just getting one structure was a lot of work, a lot of effort. So I was improving methods to do that in a better way. And I kept doing that during my PhD. And I think so Haddock, all the development, all the complexes really was a good idea at the right time. So it's not like I was seeing coming that, you know, we have to go in that direction. At that time, there was this PhD student who was hitting the wall, and he had data, and then having the idea, okay, let's try to use this data in a different way, combine that with modeling. And this was a time when there was a need for these methods, because a lot of people were moving into that. So it's maybe luck, it's serendipity, I don't know, it's intuition. So it was not a planned decision beforehand, I'm going to work on biomolecular interaction. At some point, it just happened. And then, you know, that was the right move, because once this started, then we could do a lot of things, and you could get, it was the right time, I could get grants for it. So you cannot plan where you're going to be in 20 years from now. I think that's very hard. But you have to be enthusiastic about. I like research. I like being with people, so that's the enthusiasm is carrying you forward. But you cannot plan where you're going to be in 20 years. And what you will be doing in 20 years, for sure not.
Yeah. And the next viewer's question, do you provide scholarships support for intern students from abroad?
So the university doesn't have such a program, but I've had several students from Turkey on Erasmus Plus fellowships. Some of them coming after completing their bachelor for a few months, sometimes also at the master level. So this is, it seems to be relatively easy to get. And I know, so again, because of the contact that I've had over the years with Turkish group, I know that you have a good education system. So I know that there are students with good skills and good knowledge in general. So, and that was that came from visiting Istanbul, visiting a number of places when you realize. So again, that's the human factor. You know, you get invited to talk, and then you realize, and that's otherwise, if you've never been there, it's difficult to judge. So go for a look at the Erasmus fellowships.
Thank you so much. Yes.
Other questions? Thank you for your presentation, Professor. How do you deal with issues that came in front of you?
Issues that came in front of me. So I'm not sure what that means. You know, they always... being an academic, I think, you have to have a tough skin because basically, at least in Utrecht, my salary is paid, but that's it. So being an academic is kind of being an entrepreneur, or you are running your own business because you need to find your own money. The way that things work in the Netherlands, it's not like you apply for PhD students apply to a graduate school and then get hired and then decide on a PI. I need to find money to hire someone. So you are constantly looking for money, applying for grants, which is why the networking is important. And you win some and you lose some, and that's so you should. And you know, if you get very quickly disappointed, then it's a tough life. If you like what you're doing, then it's a fun life. I'm still doing programming myself, you know, so I still find time to do the fun part of things. I still do computations. I still look at proteins, and that's the fun part. If I had to do only administration now and management, it will not be fun. So you have to, you know, like what you are doing. I always telling my job is my hobby to some extent.
Next questions. We are
we appreciate your contribution and incredible presentation. I wonder the most unforgettable process about your career.
Yeah, I don't know what was the most unforgettable. Well, what was the key point? Maybe getting this phone call when I was in Zurich in the ETH to get the assistant professor position. So this was being lucky, or maybe, you know, luck. You seed luck maybe also. It means, you know, I had a nice PhD, I had good interactions and things. So that's again maybe the networking. So things comes back your way. So that was probably key. And so moving back to Utrecht. Or maybe the most unforgettable process about my career was meeting my wife when I was in Switzerland. Otherwise, I would never have ended up in the Netherlands. Maybe I would be somewhere else, I don't know. So maybe that was the most significant part of, that's what made me go to the Netherlands to do my PhD, and that's where I am now. So that's probably the right answer, yes. But at the time, you know, I didn't know where. I was not expecting to be able to stay in the Netherlands, so to come back, because I went to do my PhD, then went to the US, to Switzerland, and it's not simple when you, at the time, you would think it was much easier to stay in your own country to do your studies, PhDs, and then go for a postdoc abroad and combine to your country. That was kind of the typical track. So that's, yeah. If you had asked me when I was 20 or 30, would you end up in the Netherlands as a professor, I would probably answer no. The chance is very small. How do you run science and your social life? Yeah, that's also a tough one, because it's not a 9-to-5 job, basically. Okay, so it's never stopping. So you bring back work with you at home. Well, last 2 years I've been working mostly from home, so you know that's then you work as you can organize your own work. So I think that's one of the beauty of the academic world. So you can organize things as you wish. You don't have to clock in the morning and clock out in the evening. So you have to, as long as you're doing your work and you're doing good work, nobody is telling you how you should do it. So that's this academic freedom. I think it's important. But it's all the travel that I was doing. Then I have 2 daughters. Okay, they're out of the house now, but that's if my wife was not there to take care of the home front, then this would not have been working. So that it's a teamwork also. But yeah, so you have to find a balance. But again, I'm taking work at home, and I'm still doing mailing, answering emails sometimes in the evening. So that's you have to find the right balance there. It's and there is not one solution that fits all. I think it depends very much on each person there.
Next questions. What is your advice to friends who will pursue an academic career?
Yeah, I think like it brings me back to what I call the man-where, the human factor. Okay, so look at you have to decide what to do, where to go. You want to be in an environment with nice colleagues. I think that's important. Okay, and then if you are in a nice environment, then things will, in my opinion, things will work. If you go in a very competitive environment where people are competing against each other, that's not going to be fun. So think carefully where you want to. Sometimes you might not have a lot of choice, but again, the human factor, it's not only about the IQ, but the EQ, emotional. I think that's very important in all the choice that you make. And most importantly, you should have fun in what you're doing, because if you don't like what you're doing anymore, then you should do something else.
Okay, I ask my question, and you tell us about your Haddock system. And did you think this project reached so many people?
No, I think when in 2000, so probably the idea was 2001, 2002. We started, and you could not foresee that it was the right time. And at the time, it was mainly dealing with NMR data. And then you see all of a sudden that many other people. So once we put it out there was a demand for it. So it was the right time, because the entire structural biology field was moving toward complexes. Now, if we were to do the same right now, it will be probably much more difficult, because then complexes have been there for a long time, and then you have the artificial intelligence coming in. So again, it was the right time. And then you have to invest again. So the training aspect. So we had comments from users telling us, okay, now I understand why someone was advising me to use Haddock. It's because I got an answer to my email to my question within 1 day or whatever. And then some of our software. So it's it's an investment that you have to make. Some people like to publish, a top paper. They publish a method. They publish it in a top journal, and then they don't want to provide the support to help people, and then nobody is using it. They might still use it to do fantastic science inside their own group, but then the impact on the research community is not going to be very large.
Thank you for your answer. And
what do you think is the most important quality to be a scientist?
The most important quality to be a scientist, I think you have to be curious. That's important. Keep asking questions. Be open-minded. Yeah.
That's all our questions. Thank you so much.
Okay. Thank you very much. It was my pleasure. Okay, wishing you all the best for the remaining of your 2 days.
There, right? Thank you so much for this presentation, Professor. It was really lovely, especially the question part. I was like, try to not scream like fangirl. Your answers was really inspiring, and it was really helpful to our participations. Thank you so much for this.
You're welcome.
We would love to see other events in your presentation. Thank you so much.
Okay, yes. Bye-bye. Have a nice day.
Will be started at 1 p.m. We will see you at that time. See you guys.
Hello everybody, my name is Özlem. I'm the Vice President of IEEE GTÜ EMBS. Also, I'm a bioengineering student at this university. This is my first year at this department. Today, Anil moderated this section. How are you, Anil?
Thank you, I'm fine, Özlem. How are you too?
I'm great, thank you. So I leave the word to hello everybody. My name is Özlem. I'm the
Hello everyone, my name is Anıl Emin Oluncu. I'm 19 years old.
Also, I'm a bioengineering student
I'm a molecular biology and genetics student at Gebze Technical University.
Today, Anıl moderated this section.
Hello, I am the other moderator
Thank
of
you,
this
I'm fine,
session.
Özlem.
I am
How are you
Fatma,
too?
and I am second grade student in Molecular Biology and Genetics Department in Gebze Technical University.
Hi, my name is Seda. I also second years on molecular biology and genetics.
I will now introduce you to the speaker of this session. His name is Dr. Özgün Gökçe. Dr. Özgün is currently attending from Ludwig Maximilian University. He continues his studies on neuroscience, and he will talk about functional genomics of neuroinflammation in aging and neurodegeneration in this session. Thank you so much for accepting our invitation, Dr. Özgün. If you have any questions, you can write to the YouTube chat section. We will be waiting to forward your questions. I leave the word to our speaker. If you want to, you can start, Doctor.
Oh, thank you very much for the invitation, and thanks for the organizing this. This is especially with the limiting travel and with the corona restrictions. I think, to be honest, that even before it was not easy to get the nice talks in my undergraduate studies in Turkey, and this internet access kind of opened up the communication, and you for the organizing such talks, important to find where to grow the path. So I need to figure out now how to use the Google one. So I will use probably my entire window. And then I will switch to the, is it working?
Yes, it's working.
Great, I don't see you. So if there's any problem, please, and also just stop and ask me in the talks. I will not going to, I would like to talk freely about and open discussion in the between. If anybody has any questions, just keep on asking. First of all, I would like to tell, probably I got the wrong training in my undergraduate. I was a molecular biologist in Boğaziçi, and I studied on the behavioral Reşat Can Beyli laboratory. We done behavioral studies and molecular biology. And for my PhD, I wanted to switch to the gene expression. And actually, back in the day, I'm quite old. We were using microarrays, these chips that you put the RNA on it, and you see which ones hybridize to where, and you estimate the gene expression. And that's actually computationally wasn't so challenged. Like you get few genes up, few genes down. But since, and I moved back to the molecular biology as this bioinformatics, there was not enough data to mine. But since 2009, things are changing in a pace that's really, really hard to follow. So, and this is really important for the Turkish and also the scientifically not that countries that scientifically didn't invest it so much on the hardware, on the expenses. So we have a certain drawback for building the universities, laboratories. However, right now things are changing in a way that we need more people thinking about biology and know how to analyze the data and know how to ask questions rather than people knowing how to use equipment. So on that, and that's a change in that's a change last 8-10 years. Before, technical expertise was the most needed tool, but right now we are doing an experiment and I can actually spend several years on analyzing that. And that's what my group actually does too. We use these genomics technologies from single cell to spatial technologies to address questions that we are interested. But you can take this talk as an example to apply any biological challenging question. And asking question is all about biology. Before going that, I wanted to thank I'm going to talk about not my work, but my group's work. And here's my team. And they are amazing, amazing students and postdocs I have, and also my funding and my collaborators. So I was talking about the questions. What to focus on biology is really important. And we need to focus on the risk factors that are limiting our health span. And what is lifespan and health span? Lifespan is very easy to measure. It's written in every gravestone, the birthday, and you subtract the birthday from the death date. Health span is a little bit more complicated. Health span is how quality that duration was. So it's been chronic diseases subtract your fitness level, a lot of your cognitive level decline, a lot of things alters this health span. And we realized that it's not we wanted to live long, we wanted to live better. And we wanted to enjoy the life. There's no point of hanging around here if the life is miserable. So what we wanted to do, improve our quality of life. And when we look to the chronic diseases, that cancer, stroke, dementia, diabetes, and the one risk factor cuts across every disease, and which is the aging. If you see here, the risk factor of all these diseases are increasing, and my focus being neurodegeneration, the gray line, it is significantly increases after 65. It's a little bit flat and then steeply increase. And aging is a big problem across Europe. We are actually, Turkey is a little bit behind. We are quite young nation, but rest of the Europe is quite old. So we are expecting a major aging problem in the coming decade, a neurodegeneration problem. And how to address this question? So far we addressed it with genetic disorders like Huntington disease. I done my PhD on that. It's a dominant disorder. If your Huntington gene has a certain number of repeats, you develop the disease, and the length of this disease, the length of this repeats tells you when you're actually going to show the first symptoms. Great disease because you can model it in animal effectively. But aging is hard to model because it's very complex. But recently we developed this single cell technologies. We can actually get to the individual cell and extract the transcriptome inside it and see what that cell doing at the time of the at that time. And we can isolate all the cell types and different timelines, isolate from animals and the human samples, and see how these cells actually, in a normal aging here in the blue, how these cells goes through projecting, and what happens if you get a disease. And we generally, with these models, animal models, and study the end point of the diseases. And to be honestly, it is hard to intervene at that point. All the cells gone, and we can mitigate the effect. But the promise of the genomics, if we can build these healthy trajectories and understand when the disease actually starts, the first break point from the trajectory, we can actually go there, intervene, and put the disease way before the first symptom, prevent the disease. And that's the promise of the aging research. If we understand aging, we can maintain a healthy aging and prevent the diseases. So for this, what we looked is, I'm a neuroscientist, and I looked at the brain aging in the mice. And as a problem, sign of problem, we looked at the white matter microglia. So the brain has two different parts. One is gray, that is where the neuronal soma localized, but a neuron is 90% an axon. And this axons goes into the white matter because it's myelinated. Myelin makes this highly lipid structure, makes it look white. And that fibers connecting through different regions of the brain is the critical for a circuitry based organ like brain. And we looked at the brain in a 24H mice, and what we saw is the immune cell in the brain, this microglia we call them, get activated only in the white matter, and that was in the aging dependent. Here you see the corpus callosum of the brain of the mice, and there you see this red activated microglia, and you don't see that. So that was we done with the electron microscopy, got their structures. Seems like there is some difference between white and gray matter. And it is really important because maybe you even heard that in my training, we always thought that when you get old, you lose neurons. Your neurons dying by aging. Actually, that's not true. Now we know good counting, we see that neuronal numbers don't really change in the normal aging. What changes is white matter volume, and it is kind of linear in here. If you see that after 60, 50, there's a slope of the linear decrease of the white matter volume. So that might be the relation of the aging, and there are imaging abnormalities in the aging. They also increase in the white matter only and in a linear manner. So when you look these measures, you can actually say predict the age of the person looking to the white matter parameters. So why they are decreasing, we want to understand what is happening, this decrease and problems in the white matter, how this thing is happening. And there are many questions. It could be just die old cells, but why in the white matter? White matter is not very vasculated. Maybe it's the oxidative stress or this oligodendrocytes that wrapping the axons are really tightly wrapped, and these proteins inside doesn't, and the lipids doesn't really recycle. They live like a 50 years half-life. They have a half-life. So, and they accumulate damage, and this damage might be the problem after a certain point the brain cannot handle and white matter start to inflate. To understand that, we use single cell transcriptomics, and we developed quite a new method to isolate only the white matter from a mice, and this is very tiny in the mice, and only the gray matter of cortex, and compare them in the aged mice in a protocol that we can get the best of the cell identity transcriptome without the artifacts that are induced during the dissociation. And when we done that, and we see here is, you see the microglia, individual microglia, we got 4 major microglia types. What is really striking that the cortical microglia was all homeostatic like our imaging. But in the white matter, we got 2 activated subtypes, and one we call it white matter associated microglia because they were only in the white matter. And we look to and the beauty of this method is you look to the genes under there, and what these genes are telling us is, what these genes are telling us are what these cells are doing. So we see that activated ones are activated their translation, all the ribosomal genes and mitochondrial genes, so they are preparing for something. And when white matter activated genes express 2 things. One, phagocytotic and lysosomal genes, which tells that they are eating a lot of something hard to digest, and MHC class antigen processing and presentation genes. So these are macrophages, and when you get a corona or bacterial infection, one of the role of the macrophages are eating the sick cell and damaged cell and presenting the antigen to the adaptive immune system to flag, hey, we have a problem here, so that adaptive immune system can attack here. So what we next do is expand this study on more cells. And last time you saw 2,000, we got 20,000 cells and reproduce the same data in a different animal cohort so that our results are reliable. You always wanted to see it's not one experiment or one method creates your results. If you find something important, you wanted to check with multiple different methods to see same effect is reliably seen on other ones. And here again, we see it, this time we see the activated one a little bit in the gray matter too, but the WAM was specific to the white matter, so more or less acceptable reproduction. And we validate the markers we see, and you see from the microglial, activated microglial markers only increase after 18 months, and they steeply increase at 24 months, which is matching to our single cell data. It was interesting because we were surprised, couldn't believe our eyes. Many studies before us looked to the aging brain, microglia signatures, single cell transcriptome, and they didn't report that. So we go back to this two studies, Figueroa and Hammond et al., and analyze their normal aging. And when we analyzed their data, we got the exactly same signature. What was different there in the Figueroa et al., they compared it with a harsh Alzheimer's disease model, and their Alzheimer's disease model is such a big change that this aging effect becomes overwhelmed by the noise of the Alzheimer's disease model. So they kind of thought this is a weak signature and focused the disease signature. Hammond et al. Was a little bit different. They had two different versions of a kit to do the single cell RNA sequencing, and they combined these two results generated between two different kits and analyzed it. And the problem with the data integration is you lose resolution. To integrate, to make them both together, small differences got lost, and they kind of overlooked this signature. So, but both of the data set had the same signature, which gave us the confidence to go after that. We looked at the EM structures of that, and what we see there, this microglia is filled with myelin and digesting this myelin, and myelin is 40% cholesterol, very, very hard to digest for these guys. And we looked at a gene that we know important for the microglial activation, TREM2. And when we looked at the knockout of this microglial activation knockout gene, we saw that there is no activated response to the aging in the microglia. And this is quite important because exact loss of this gene in humans causes a white matter degeneration early on, leukopsia, we call it this disease. When we look to the electron microscopy of this TREM knockout, we saw this. Normally white matter looks like that, axons, neuronal axons wrap around nicely with this sheet of myelin. When we looked there, we saw this cell-free myelin debris that normally microglia clears, but here microglia didn't clear. And when the publication accepted, we collect Garibay. He's a Ebru artist from Istanbul, and he done for us this cover to mimic this myelin walls. Myelin girdabs walls are girdabs. And we quantify that, and we saw that these are specific to the TREM2 knockout mice. So I don't want to go so much in detail. So what happens here? You get something in aging. We don't know what it is yet. That's an important question. But oligodendrocyte starts to shed myelin. And microglia converts into white matter-associated microglia and clears up this myelin sheets. And this was that's a protective response. And this was a perfect molecular biology study done by Simon and Shima. Shima was a molecular biologist driving the animal experiments validation, and Simon is a computational and single cell PhD student in my lab. He was doing the genomics and identifying the targets. So it works this way now in my group and my collaborators. We use the genomic studies to characterize the cell types and identities and make up hypotheses that we can test. And the wet lab students actually go into the lab, and that part takes a lot of technical challenge and a lot of time too, because we need to wait for the animals, cut the sections, analyze them blindly. But without that, we cannot conclude this picture, like if this worm is protective or not, requires the molecular biology. So now we want to understand why this question mark, why the oligodendrocytes get aged. For that, we again use our favorite tool, single cell transcriptomics, with this white versus gray matter. And that study done with a perfect trio of students. Tuğberk Kaya, he's from Bilkent, done his studies and then moved to the my lab for PhD. And Nico is a collaborator from Mika Simon's lab, and Laura from China, PhD student my lab. And these three worked very closely, try to understand what is happening in the oligodendrocyte leading to this white matter activation of microglia. And we done two different single cell approaches. One plate-based, we sort individual cells into the wells and do the amplification one by one. A lot of work, but it's really good quality data you get. And the 10x, which is droplet-based method, which gives you tons of cells. And when we look to the oligodendrocyte, we saw this two oligodendrocyte, green and yellow ones, which we know about them in the normal development. Next, we saw the blue age-related oligodendrocyte. These were really interested. And finally, we saw the this red small cluster, and they are the interferon response oligodendrocytes, and that was quite interesting actually. We quantify them, and as you see that in the gray matter, we do not see the H and the interferon oligodendrocytes. And then the question was again, if these oligodendrocytes related to the white matter-associated microglia we see, and we quantify the age and location in a different way with a different method, and that was confirming again our findings. And then we wanted to ask why this interferon response oligo forms. So it could be that I mentioned you before antigen presentation. These macrophages, microglia are antigen presenting cells, and then we can actually what could have happened is they can actually present this and attract T cells. Normally in the brain we don't have T cells in our brain because brain is an immune privilege. But during aging, actually we saw that CD8 T cells accumulate in the white matter. And we have the interferon response oligo. The question is if they are close. If they are close, there might be some causal relationship, and for this we done the immunostaining and localized the interferon response oligos and localized the T cells, and we saw them they are very close, and that was quite interesting actually. But as I said, this is a quite cumbersome and long experiment. And a single cell, when we do the single cell, we dissociate the cells and we don't have the location information of these cells. And for using the immunostaining like that, using the antibodies to stain the cell types, we know them how close they are. But that's actually slows us quite a bit. So now we have a new method that actually will accelerate our research quite fastly, which called spatial transcriptomics. Instead of dissociating the brain, we do the sequencing, the hybridization-based identity of 500 to 1,000 genes on the sections of the brains. Here you see the mouse brain. Here we have some lesion, and we can actually use this transcriptomics and identify individual cells from where they are coming from and their cellular states. So now we don't need to dissect the white matter and gray matter and make the single cell dissociate separately. We can annotate the cells directly where they are and then see who are next to them, what are their interactions. And we can, here you see how the cell identity is going, and this method calls Murphy's, which brings up us another dimension. We know 20,000 transcripts, which type of transcripts express each cell, and now we know who is neighbor of this cell. And really interesting study is coming that showing that if you know the neighbor's transcriptome, you can actually predict quite accurately what is the transcriptome of this cell. So there is a really strong regional correlations regulation of this transcript. So spatial, especially in the disease field, will change how we do. Okay, and we are using these technologies, but again, to understand the method, we need to do perturbation experiments. So if I say T cell is nearby the interferon response oligo, that doesn't tell us there is any causal relationship between them. It's just they are there. There are a lot of correlation is not causation. So in order to make some causative correlations, we use these two cancer drugs, checkpoint inhibitors. These are T cells as their checkpoints so that they don't harm your body, so they get deactivated very fastly. And there are cancer treatments right now, PD-1 and CTLA-4 inhibitor antibodies. They block this inhibitory mechanism in the T cells, and T cells became super active and attack everything. And we treat the mice 6 weeks with these inhibitors, and what we saw is we saw nearly 4 or 5 times more interferon response oligos. So that brought us a little bit causative. And next, we just take the T cells out. There's a genetic model, RAG knockout we call, and this genetic RAG1 knockouts lacks the B and T cells. And we use the 24-month-old RAG knockout mice and check what's happening. And we again use single cell transcriptomic. And to our surprise, there were no interferon response oligo, suggesting T cells involved in creating and enhancing the interferon are required for interferon response oligos. Now we can go from they localize next to each other to they actually T cells actually causative for interferon response oligo formation, and that's important because why? Yeah, I taught all about this interferon response oligos, but why you wanted to know about that? So that's the phenotype from this cell. What does it happen? What does it relate to the white matter thinning, loss of volume of the white matter in the aging and this interferon response oligos? So to measure that, we simply count the density of oligodendrocytes in the white matter. And we see a quite linear decrease of the white matter oligodendrocytes during aging. From 12 months to 24 months, it's decreased significantly. and 24-month-old rat knockout had rescued this loss. So although this interferon response oligos tiny little bit, we believe they are actually slowly eroding the white matter and decreasing the volume, and that has to do with T cells. And this was a big surprise because in the mouse brain sections you don't see so many T cells. Like per section 2-3 is pretty much what you, and actually you need to be close to the ventricular space to see them. So this is quite surprising it was quite surprising for me because I'm a neuroscientist. I think brain is protected from immune system with this privilege, but we are learning more and more this is not the case. And in summary, what we see here is H oligodendrocytes. H oligodendrocytes release myelin debris. Microglia activate CD8. Microglia eats them. They may or they might. We haven't shown that to you, and we don't know. We believe that somewhat attracts the CD8 T cells, which induce the interferon response oligos and neurodegeneration. I know this is a little bit heavy with the data. I hope you kind of enjoyed, but I would like to give you how molecular biology with the genomics technologies get together to generate ideas and test those ideas. And yeah, my group is working on, in one hand, this, and we have some other drug candidates and molecular focus, and this is pretty much my talk actually. Is there any question?
First of all, I would like to really thank you for your presentation. And as you said, it's a little bit heavy for us, at least for me, because I'm a second grade student. And I was wondering if I understood correct, of course, white matter decreasing causes the aging, and it causes oligodendrocytes phagocyte large things, which are very hard to digest, and oligodendrocytes are aging, as a simple summary. Is it correct to say that?
Like the statements are really important because when we are writing and we are submitting this paper, one thing we say, I wouldn't say there is white matter, loss of white matter volume causing disease. I don't have data for that. It's a correlation. We see that decrease, and that's actually in life also really important. We are very much correlative machines. We try to correlate things, and from that we think, okay, when I see the clouds, rain coming, and rain comes from clouds, which is very useful. But there are many times when I see a black cat, something bad going to happen, and that type of correlative thinking makes us bad judgments. So we need to be very careful about if you see two things going together, and there are really good websites like showing the number of shark attacks and the oil prices, and plots are actually identical. So when I show you that white matter volume decreasing during aging, it doesn't say cognitive decline because of that. We think, and it's a good hypothesis, but we don't have tools to increase or decrease or preserve it and see the cognitive functions. Actually, my research is kind of finds this idea of that when I say this microglia response decreasing and this affect the white matter function, we have now TREM2 knockouts, and in the TREM2 knockout, we see the cognition is going down very bad and the white matter is decreasing. That would let me say you are right what you said, but I know TREM2 knockout has many other problems, so that might also cause that. But the main idea of here is we saw a phenotype and decrease, and we wanted to understand that decrease, and from there we go to the one immune cell, and that immune cell response. From this response we get to the idea it is mostly about oligodendrocytes, and then we find a very small 2% of a tiny population of oligodendrocyte, and that gives us a hint that there might be T cells around from just looking to the transcriptome. These are interferon response. Interferons generally secreted by T cells. We weren't sure, but we asked Laura, can you just stain for T cells in these sections with these cell markers of these two cells if you see them nearby? And then she looked there and like, oh, there are. And this is rare because you have 1 or 2 T cells and all these oligos were located around that. And that makes it really interesting. Then that's the example of how wet lab and the genomics, and this is a collaborative work. We have other partners in that, but the progress in my group that I see using these technologies is faster than a decade before done the field. So the genomics really speed up ours, and that's what I wanted to give that question. The phagocytosis, the name of the TREM2, these are, you don't need to memorize. I don't memorize things, so you don't need to remember those things, and you don't need to know. But ask a big question and address it with a global way of looking to the all cell types, and from there, having hypotheses and follow that is what we can do right now, and that is only possible when we have molecular biologists and the computational biologists work hand in hand.
Okay, I understand. So we can say that it can be one of the causes, but of course we are keeping on our researchers. So thank you for the answer.
No, my pleasure.
Thank you.
My question is, how did you decide to study neuroscience, and what influenced you the most when making that decision?
I think neuroscience is the coolest. There's all the other organs are kind of like immune system is entertaining and other things, but the thought and the cognitive diversity of the brain is a challenge that is not I'm going to solve, and that's kind of gives me a peace. I'm dealing with something complicated, and I have a basic idea of how the kidney works, how the liver works, how the heart works, but I can't really say that for the brain yet, and that's the why. I always go to the unknown, and I think the ultimate frontier is neuroscience. The second question, what made me decide on that? I was a quite biology nerd since my high school friends remind that I was talking about Krebs cycle when we were out at 12. So I guess that was, I don't remember what really changed my focus on the life, but I had exceptional mentors on the way. And they show how to be effective doing biology. And I was working when I was undergrad in the Rashid Can Beyli in Boğaziçi, who is an amazing scientist, and we were taking care of our rats, cleaning rat cages and doing experiments there, but also every Friday, 7 p.m. If I do a 7 p.m. Friday lab meeting, I think my students will stone me. We were meeting and doing journal clubs from 7 to 9, and now I appreciate he taking his Fridays just to give us how to teach this thing. So I had many people who sacrificed their own private time to show us the path, and I'll try to pay it forward.
Thank you for your answer.
I have a question. What are you planning to work on after the corona you worked last year? Am I right? You worked corona last year?
Yeah, so that was kind of a little bit a detour for us. As you see when the corona starts as a biologist, we are pretty much interested on that. And you see these T cells. And right now, my research, I became an immunologist, not by choice, but the data pushed me. So we didn't, T cell subtypes is a whole new world that I'm every day reading and learning. That biology is quite diverse. When you specialize on one area, it becomes like, oh, which tools and jargon, which words to use in the other field became really problematic. And now with the technologies, we are just don't know what will bring. So the beauty of the spatial transcriptomics, you cut a section, you see what the cells there, and you see which cells interacting with each other. And the beauty of the biology at this age is, I don't know the answer to your question. Yes. Because what will come is what would be the most surprising I will work on. And most surprising will be not I know.
Okay. Thank you for your answer.
And one of Emirhan, who is one of our participants, asked that, thank you so much for your presentation. I want to ask about your life balance. Even if we are students, we had a very hard time to catch with all of the lessons and social life, how we can manage that he is asking to you.
Yeah, I have that with my students as well. We discussed that quite a bit. I think, yeah, people will not like what I say in general. I do believe there are sacrifices in the young age you need to do, and for that you need to pick something you enjoy. And like I'm actually really much I was writing a grant. I forgot to talk actually. Thanks for the message to remind me. I was writing a grant and I find a new way of approaching things, and it's fun. And I'm not complaining in the Saturday morning reading that. It is much more joyful than in the horrible news in our days. It gives me the refuge to escape from everything else. And if you find something you like, you don't really worry about. I don't really worry about it. I had my life and the work is always mixed together. I studied in postdoc with a Nobel Prize winning lab in Stanford, and we put our vests and go to the swimming, and we had sports coaches coming, and then everybody had a break, and in front of us there was a basketball. Many injuries with the playing basketball, but yeah, you find people at 11 doing experiment, but you find another corner people having a beers and discussing politics, science, and everything planned. It's kind of a fun. In Germany, it's different. People separate life and work, and they try to balance it. I never done that, so I don't know. I now have a kid and my son and my family, so I need to spend time, but still it is pretty much blended together. I don't recommend that to everybody, but if you find something your hobby as a work and people paying you for that, that's not a concern. But I don't say that only way. I have a student who is exceptional, Simon, the first part of that, and he splits his life and the working time exactly, and he's to go to his path. What I saw, what he achieved is he's specialized on something and perfected that. So right now, both the biostatistics and the computational speed, I don't know anybody can approach him. In a day, what he can do, in a 2 weeks, or other people will take 2-3 weeks and probably will be wrong, and he would do it right. So focusing on one specialization would be the way to separate life and work balance. And I never had that type of attention for one topic. So for me, it's hard to focus on one thing. I like to change what I'm doing. But hard question.
Ahmed Furkan asks that how did you continue to your research with your students or maybe lab mates during the pandemic of COVID-19?
We were lucky. That was quite lucky because we are hospitals, so we could work in the lab, and we are more lucky because I just hoarded so much data from our previous single cell and genomics, and we didn't analyze them properly. So what we done is wet lab people started to learn computation, and computational people totally focused on the analysis, and then we couldn't work as much as things that we had to plan our experiments very, very careful. When I tell the story, we validated the other studies. Actually, we first analyzed them. I changed it's not, when you listen a talk, it's not chronological order. Things didn't happen in that order. We look to the other studies, find something in the data analysis, and then we need to look vertical staining. This is here. So we need to isolate that in the single cell. So the story goes, and what I was saying in the beginning, the biology student your age, computational gives you an amazing access. If you know computational tools, you can actually access to all the data, and we are only looking to the tip of the data in a publication. Like each data set has wealth of information, but we are asking one question and getting millions of answers, but we are writing 1.5 paragraph about that answer. So we just focused on to the data analysis and plan our experiments very carefully, and we could go and do some of the experiments. So we didn't so much shut down, and the wet lab molecular biologists learn computation, so everybody now capable in my lab using R and doing some of the analysis, and computational students did a bit better. So when one path is closed, you find another thing to go.
Okay, thank you.
And I have, one of our participants has asked another question. Faiza asks, thank you for presentation. I wonder that what was your motivation on your career field selection?
Yeah, that's again, I'm not the most useful person because I just met with my high school friends and they were making fun of me. Since 12 you were talking about biology. Apparently something I had my favorite reading was TÜBİTAK's BİLİM ve Teknik in 90s, and that was a quite exceptional part, and constantly new questions, and reading that as a child really opened up my curiosity. And another thing is, I think the biology most important thing for me, I can link at 11 when I was universe, life, meaning, etc. questions, I just realized I don't need to know it, and actually don't know it is fun because there is something to learn. And I didn't like pre-made answers that biologic books, all the books around me, everybody giving, because they were not based on data. They just wanted to shut down the annoying kid. And biology seemed to be the path to do that. And I actually remember one reason. It is this. And this reason why there's a muscle on my skull that can move my hair. And I got to the Darwin's Origin of Species, and it's an old press from an old bookstore, and I was reading, and it's way above my understanding. But in the first chapter, it tells about this muscle group and how human and monkey is related, because very rare people, some people can move their skulls. And I was like, no, everybody can do that. And then in the class, I made the experiment. I was the only one over my friends and my family. So I realized I'm more close to monkey than the others. But that actually kept me reading that book. And then the selfish gene, double helix, and all these. Tübitak had these beautiful book translations when I was a kid. Not any longer, I see. And these actually open up quite a point that we don't need to know it. We can learn a lot of things. And I think the evolution theory had been the central piece for my path, because there's no biology without evolution. I cannot build a project without thinking from evolutionary prospect. Everything makes sense if you think it, how that could evolve into this system, and there is no logic in the evolution, by the way. That's the beauty of that. Then you can address the questions with an open mind. And so I would give the Darwin the biggest credit.
Thank you for your answer. Şeyda Yağcı asked a question. As a person studying in Turkey, it seems very difficult to get an education abroad and do an internship. Do you think that where you are is important in becoming a scientist?
It's getting less important. I would say it was really important when I was in your age. Like the Tuğberk right now, I'm so lucky to have him in my group, amazing student. And he contacted to me not because of internship or how to go. He contacted me because in my paper I use this analysis, and he asked me some questions during his master. And we kept in touch, and then I suggested him there's a website called Serot, which gives you the raw data of single cell data and step-by-step analyzes that. And then if you go through that and apply to somebody else's study, and hey, I see this your study, I try to repeat, and there is this difference, that is quite a different than I want to do internship.
Thank you.
And because especially right now, we mostly done the remote internships. I just finished one student, Bilge Sudana, remote, and she done, and she was like, oh, I didn't understand. And she went through above our analysis and hit the interesting things, and she wasn't secure about what she was doing at that point. But I'm like, oh, you are finding something new, great. And she never left Turkey. So that's the computational training. You don't need to be there. Actually, it's not really helping so much being in there, because I mentioned my student, I actually don't see him once a month. I see him right now. He's mostly at his home working, but every day we are talking on the Zoom and other things. At this age, it's very easy to find what you want and focus on that. And I know it's sometimes hard to focus on what to look, but the basic R training and afterwards single cell analysis from Serot or Python, and we are arranging a lot of online training systems. And once you have these things and go over the first, don't write to me if you haven't read my papers, because you wanted to spend your next 5-6 years over somebody's vision, and if you didn't spend time on what they've done, there is some mistake there. There are very bad laws, and you would be very unhappy. But if you understand the research before invested there, it makes a total difference, because you get a lot of emails about internship and PhDs, but you kind of see that everything is copy pasted, and then you kind of sometimes you miss. I try always responding them, but if it is a generic mail and not related to my research, you kind of, it's the wrong attitude there. And you don't need to be anywhere else than where you are. You need to ask the questions, and when you are trained your stuff and know where you do, people will love to help you. Everybody will like to help you. But if you don't know what you want to do, it's hard, because that's your responsibility.
So we can say that it's all about making research and find our area which can be our passionate about and make more and more research, right?
Yes, and also the getting expertise. And it's the first year of my life, and I didn't know what I want. I jumped into the behavioral research. I really didn't like it. I still use it, but I didn't like the way it approached. And molecular biology hit, and I kind of followed love in when we get to the multidimensional data. That totally changed the thing. And until then, it was a search. You try and fail. Like you try, but you learn and you understand. You can actually criticize it, and then you are kind of, and you started to change it.
Thank you.
Ali or Khan asked that, can you explain the effect of hyperbaric oxygen on telomeres? Thank you.
No. Thank you. No. I don't know the hyperbaric acid, so
we are skipping that. And I have
no, I answered the question. No, it was
yes. And Ruket has a question for you. When you were mentioning oligodendrocytes, you also mentioned IRO. IRO has little space. I didn't exactly understand the importance of IRO. Can you mention a little bit more? It attracts my attention, that she says.
Yeah, that's actually it. I tried to, that was the tiny little population we saw in the oligodendrocytes. These are the cells that wrap the axon. And we saw with the single cell increasing numbers, you can say, catch these rare cell populations. And before EROD, my research, I wanted to tell that cytic fibrosis caused by a gene. And we didn't know the reason of this because gene is not expressed in the lung, and it's a lung disease. So we didn't know why this is going on. And now we have single cells. We can analyze individual cells, and we saw that in one rare cell type that regulates the secretion of the secretory cells, expresses this gene heavily. And when it's mutated, you have this horrible disease. And analyzing that, and it was always, if you just look to the bulk, like take the whole lung and smash it and look to the gene expression, there's nothing there. Why this gene causing a lung disease? But when you look at key player on that team really needs that and breaks the system, that single cell technology brought us. And I'm trying to, my work is the similar. This ERO, interferon response oligo, is a small population. What we saw that we went after that, it's actually, most of the work was done on the bigger population, but it didn't lead to an interesting outcome. And so you don't hear it. The rare population that the 3% to 1% population are always near a T cell, and the brain has very, very few T cells. And that T cells in the brain, which we haven't shown it yet, but we are suggesting that in the aging brain, the white matter loss is something to do with our immune system. T cells migrating to that and doing something bad. And that is how we go through the system. And I don't know if you heard about the Epstein-Barr. It makes the uçuk in your lips. Nearly 95% of us have it. And there's a new study in Science published a few weeks ago show that Epstein-Barr virus 64-fold increased risk of MS, nearly prerequisite of multiple sclerosis. And this is suggesting that in the brain and adaptive system, and the infections like Epstein-Barr, maybe corona will lead to something different, has a interesting relationship and altering the aging. And this is something we want to understand. And it's hard to study because on average, you have 2,000 T cells in the billions of brain cells. So that little number of cells, how they can cause. Interferon response oligos, because of the interferon signature, pointed us to the T cells. And that little population area, you don't need to focus on that part.
Okay.
Yes. Şevval Karabulut and Indil Polat ask, thank you so much for your excellent presentation. I want to ask that, do you have any advice which can lead us to continue successfully in your career and to give advice to the students who want to move in your field?
So I think the one thing is, one important part on the success in the biology is resilience, direnç. So you will have a lot of downs, ups, and your generation has been very unlucky, to be honest. But actually, the worst results and the worst fails of my career give me I started back to the computation, because I realized what I was doing 3 years day and night was a artifact. So I was studying something that was not what I was thinking it is, and the whole this work crashed, and I didn't want to start back again, so I needed to change the direction. And I went to some talks and single molecule sequencing, and then start a new project. And at 3 years, I didn't publish anything. 2 months later, I had a really important paper come out from that, and that was a different angle. So if I gave up at that moment and life ended, I felt like that. And retrospectively, no. Actually, the worst things can be the opportunities, because looking back, if I succeeded what I was doing, I wouldn't have that much fun right now. So the one thing is, connections make retrospectively. So when you are in a really bad feeling, it might not be that bad. So just give yourself a doubt. And my biggest advice right now is, actually, I can tell you, there will be no neuroscientist without computation. If you don't focus on computation, you are very much disadvantaged. So and it's hard. It's not easy thing. And if you don't get into the courses on your workload and other things, but the investment in the computational field will pay back. Right now, I do see wet lab and dry lab running together. We don't do modeling. And in 5-10 years, I do see machine learning and modeling became a fundamental tool. So as data size increase and the depth of the information increase, analysis tools, we don't look any longer at gel and see the papers. Right now, you need to be computationally capable. And that's the biggest advice I can tell you in the future. Computational capability is the key.
So software is crucial and a must, right?
I wouldn't say must, because I have none. I saw a lot of very capable wet lab students who work perfectly with computational students. And they ask the question, they bring the question, they don't code, but they are reading, they are asking, they know how to ask questions, and they understand how these analyzes work, but they don't run them. So that's very effective. But if you don't understand what they are doing and you wanted to just run something, that is no-go. So you need to understand the analyzers. You don't have to run them all the time. But you need to understand them. If you can run them, that's a big add.
I get it. Thank you.
Ufuk Shen asked a question for you. Thank you a lot for presentation. Are there any technology to avoid the loss of the resolution while integrating different data, or is it not that big issue of concern?
It's a huge issue, and there are a lot of, actually, if you look to the Nature Method and Nature Biotechnology, these are our top journals. half of it about that problem. And because right now the promise of putting everybody doing the human brain atlas, as I said, this projection of the aging and each disease, and for that we need to work in different teams, and we cannot do that in same batch, same time. So it is a huge issue. There are improvements, but the point is algorithms don't know what is a technical error from an interesting biological heterogeneity. That is like you have some difference. Each of us are different, and that's what is interesting. And if you analyze a genomics data set and you done slightly different, you will have a slightly different result. And this two difference sometimes hard for an algorithm to understand. There are ways to look to the systematic errors across the different cells so that you can correct that. There are many smart ways of doing that, but if you have a bad data set, quality control, low quality data set, mixing with a better data set results you less information in our experience. So we try to do the experiment best and then use these tools to integrate most effectively. But it's like truly, if you put a gun-bad vegetable in a mixed turle, it's not going to taste good.
Thank you for your answer.
And Senagül Yılmazel have a question. Thank you very much for your presentation. Have you reached your goals regarding your profession?
I had questions, not goals. And they are keep coming. So no.
So we have another question.
Samet Başkaya, how long have you been studying aging and this specific stuff or aging?
So the aging research came into my lab recently. Before that, I worked. The aging research came because we could study aging. Aging was not accessible. First, you need to wait for aging. It takes time. With the mouse, 2.5 years you need to wait, and that's a limitation, important limitation we try to address in different ways. And the second is getting that long wait and getting one data point. You cut it and staying with one antibody is horrible. That doesn't pay off. So now we have the spatial transcriptomics, single cell transcriptomics. You can actually get from one animal or one model, you can get a lot of information, and which makes that wait worthy. So it's been since 6 years, and which is since my lab is using single cell genomics, we are using that. And specific, aging is very, I think, if you look to any disease, except genetic disorders, dominant early disorders, aging is risk factor for all the chronic disorders. So if we understand aging, we can actually decrease the risks of the diseases and increase how quality of our life. Even corona right now, if you looked at your risk and your grandmother's risk are different. And why this is. If we can understand that, we can actually, when you are a grandmother, actually you might have a very healthy life, ride your bike, go for a hike, and take care of the grandchildren and write new books. And that's the promise.
Yes. And our participant questions has finished, as I saw. So if you have time, I have a question. Is it okay
Sure.
to ask you? Okay,
Sure.
thank you. And as I understood, your researchers cover the understanding about human neurodegenerations. And have you ever made research about animal neurodegeneration, or do some of your works cover or true for several animal neural impairments either?
Well, I'm a experimental, so we have very little access to the, like what your question is, we only work on the animal, very little on human. All of this work is done in the mouse models, and the cell culture models heavily employed. Human studies actually what we wanted to move in slowly. The challenge comes is post-mortem, one of the biggest challenges we see. So when we get a tissue sample, you say even the best tissue samples comes from, it says post-mortem 6 hours. But this 6 hour, one of them maybe fall into a ski accident and on the ice. The other one was on the beach at 55 degrees. That's a totally different 6 hours. So, and the difference on the effect of this heterogeneity makes it hard and costly, because if we can increase the numbers and see the patterns now, costly to approach human. And we use animal models, animal aging, and everything I said, is it relevant to the human, today's talk? I don't know. We wanted to figure that out. So your question is actually, I would like to move from mouse to human more.
Okay. So another question.
Sure.
You have? Okay. Can root cells be used for the curing this type of disease, like you're searching for, like the aging, Alzheimer, or stroke? Actually, if I specialize this in fetus, for example, if we can take neural samples from fetus for the future treatment of this person's possible future stroke or dementia disease, can this be helpful for cure? I mean, when fetus is become an older person.
So the cell replacement therapy is quite a bit. Number of therapies are going on in human, and there had been quite a promising results with the Parkinson disease. They implemented dopaminergic neurons, and they show that improved the patients. Very low numbers, very hard effect. Yes, that could be on specific disorders, can be addressed, but we are talking about small diseases and a small number of patients with a certain genetic backgrounds that would be effective. Spinal cord injury, there's a big promise. Yes, this is pretty much possibility, and people try to convert the cells in the brains into the neurons to compensate the loss on these diseases. The numbers are very low, effectiveness of that very low. If they can break this border, yes.
Okay, so it can be that we can say. Thank you. Thank you so much.
Okay.
So actually, there's no question, and thank you for your time. And we wish we can be as passionate as you in our future areas, study areas. Thank you so much for coming, and thank you for your time.
Thank you very much. Thank you for organizing, and good luck, and have fun with everything.
Thank you.
Thank you so much for this really good presentation. Actually, I listened to every moment with interest. We are very happy to see you at this event. Thank you so much again.
My pleasure again.
And next section will be start at 2:30 p.m. See you.
Bye-bye.
Bye-bye.
Thank you.
Bye.
Hello everyone again. I'm Ozan, and Ahmet Furkan will moderate our current session. How are you, Ahmet?
I'm fine. I'm doing well. Thank you. What about you?
Thank you. I'm great. So thank you. Now I leave the word to Ahmet.
Okay. Hello, dear audiences. Welcome again to the third session today of our interactive and international event called SoScience. This is Ahmet Çolur, and I'm at prep year just before starting at the Department of Chemical Engineering at Gebze Technical University.
I am Selin Tunçarslan from the Department of Bioengineering. I am second grade.
I'm Altun Tokan from the Department of Molecular Biology and Genetics at the prep year.
And before we may start to the session about the new insights from the biology of human aging, it's a pleasure for us to present our honored Professor Bernd Schumacher from the University of Köln that has been studying genome stability mechanisms and aging for years. Hello and guten Tag, Mr. Schumacher.
Hello.
Your microphone is off, yeah?
Yes,
okay.
I just switched it on. Hello. And
hello again.
Very happy to be part of this session here.
Okay. Would you share the screen if it's possible?
Yes.
Okay, we're waiting. Have you shared?
So I'm sharing now. And you tell me whether you see my presentation?
Yes, we can see the presentation. Yeah.
Okay, very good. So then I think it started.
Yeah, we are started.
Okay, so what I'm going to tell you about today is what we learned about the mystery of human aging and which kind of insights that we gained from a science that's actually still pretty young, because you might be surprised, but modern aging research has really only been taken off in the last few decades, and it's now a greatly expanding part of science internationally, with many new institutes dedicated to the biomedical study of aging all over the world. And here in Cologne, we have one of the largest centers on aging. We have an excellence cluster that's devoted to understanding the biology of aging, one of the largest aging institutes in the world. Now, humans have always been obsessed with immortality. And there's a long history of people who have actually really thought about their end of life, their limited lifespan that they have on this earth. And even the very first epos that we know about is the Gilgamesh from Mesopotamia in the 3rd millennium and before our current era. And the king Gilgamesh was seeking the elixir for immortality, which drove him to the edges of the then known world. However, his quest for immortality remained futile. And also in very different cultures. The first Jin, the first emperor who united China in the 3rd century BC, he feared nothing more than his own death. And although he brought death to many of his enemies, he constantly feared for his life. And so he sent out his fleet to bring him back the elixir for immortality from an island, but they never returned, maybe fearing that what would happen to them if they empty-handedly presented in front of the emperor. So instead, he took what his doctors prescribed him as elixir for immortality. That, however, contained mercury, and it is presumed that he actually died of the mercury he ingested with that. So quest for immortality, although they have really been taken on in many cultures in historical times, have always remained in vain. The solution for mortality, of course, was present in ancient Greece, but it only applied to the Greek gods. They were mortals, but they had some quite mortal features, such as the goddess Eos. She fell in love with the mortal Tithonius. So she requested to Zeus, to the godfather, to the father of the gods, Zeus, to grant immortality also to her lover. And Zeus had no issue with that. So Zeus granted the wish, and only decades later, Eos had to realize that she had overlooked an important issue in her quest for immortality for Tithonius, and that was eternal youth. And so with age, nothing of her erstwhile juvenile lover still had remained, and he became old, sick, and but he wouldn't die. And that is the horror vision of the modern society, of increasing lifespan without increasing health span. Now, our populations are getting older worldwide, and not only in Europe and North America, but the entire population of the world is aging. And so also the percentage of people above 60 is rising steeply. Now, the longevity that we can enjoy nowadays with a life expectancy in Germany, for example, of 80 years, it's a great accomplishment of civilization that we can live now a very long lifetime. However, aging comes invariably with a range of human disease. These are chronic diseases that occur with aging. Dementia with Alzheimer, the most prevalent one. The risk for dementia at the age of 85 is about 40%. Chronic kidney disease is a significant clinical challenge where the patients need to get dialysis on a regular basis. Most cancer types are age-related. Aging is by far the biggest risk factor for cancer. Type 2 diabetes is on the rise. Cardiovascular disease still remain the #1 killer. And osteoporosis and frailty results in severe suffering. What is more is that disease in aging seldomly comes alone. Instead, multimorbidity is the real disease of the elderly, the disease of aging. And multimorbidity means that there are at least 2 or even more diseases occurring at the same time. And the risk of multimorbidity increases exponentially past the 5th decade of life. Here, 2 conditions, 3 conditions, it's steeply increasing. And that is the real challenge of aging. Now, what has been the traditional approach to develop therapies for diseases? So, for example, to improve the treatment for cancer, dedicated cancer research will try to understand the biology of cancer and then improve the therapy. And as you know, in cancer nowadays, we've gone through personalized medicine to precision medicine to treat the exact type of cancer, the individual cancer, because to have the most efficient treatment. Also cardiovascular disease, Alzheimer, Parkinson, macular degeneration, and so on, have been studied as individual diseases to improve the treatment of the individual disease. If you now think that patients suffer from multiple diseases at the same time, it is inconceivable that amid the demographic change with an increasing number of elderly that will be sick, that suffer from multiple chronic diseases, that this approach is sustainable for the future. Instead, modern aging research has realized that all these different diseases are not only associated with aging, they're actually caused by the aging process. And the concept that the field of aging research has developed is that a treatment, instead of treating the diseases once they have evolved, that instead we could treat the aging process and thereby protect from age-related diseases, prevent age-related diseases, extend health span instead of only extending lifespan. This, however, requires that we understand the mechanisms of aging. And if we want to understand any mechanisms in biology, we must understand it. We must understand the process in the context of the evolutionary history that has shaped that biological process. And a key realization came from a zoologist from Freiburg, August Weismann, who in the late 19th century was one of the top 3 evolutionary biologists of his time after Darwin and Wallace. And what Weismann realized is that inheritance is only achieved through germ cells. The heritable material, the genome, is passed on exclusively by germ cells. The mechanisms of inheritance that Weismann postulated are still held true to this day. So he provided the fundamental mechanism of inheritance. That is, that germ cells and only germ cells perpetuate the genetic information. And this is a fundamentally important discovery, because he realized immediately that if germ cells continuously perpetuate the genetic information immortally through the generations, that immortality of the soma would offer no selective advantage to a species. So upon installation of the next generation, the soma, the body, could decay, age, and die without any negative impact on the fitness. Because germ cells in humans have now propagated our genetic information of modern Homo sapiens for 200,000 years. We are almost in the 7,000th generation of modern Homo sapiens, and we can trace back the same germline throughout all these generations that have been, of course, passed to us from our ancestry species. And so the question arises, which are the mechanisms that maintain the body and thus determine the aging process? The experimental proof that there was inherent aging property of somatic cells in humans was provided by the cell biologist Leonard Hayflick in the 1960s. Hayflick did a very simple experiment. Nowadays, we would think it's very simple. He took human fibroblasts, normal cells from humans, and he cultured those fibroblasts in a petri dish in his lab. And then he recorded the population doublings of these cells while they were held in culture. And he realized that initially there was an exponential growth of these cells, but eventually they would completely stop dividing. No more proliferation. And he called that cellular senescence, from the Latin word for age. Back then, he had great difficulties of publishing this result because people thought, this guy, this Hayflick guy, he doesn't know how to propagate cells. Because up to then, people were only working with cancer cells in the lab. And cancer cells, they divide forever. That's the problem of cancer, that they just keep growing and growing and growing. But these were normal cells, normal somatic cells. Why did these cells age? And that had to do with the telomeres. So our chromosomes, our DNA, our genome, is organized in chromosomes, and these are linear in humans, and the chromosomes are protected by these telomeres. There are cap structures at the end of the chromosomes, and they require an enzyme that's called telomerase, which was discovered by Liz Blackburn, Carol Greda, and Jack Szostak, for which they received the Nobel Prize in 2009. And germ cells, for example, they express telomerase and extend and maintain the telomeres. But somatic cells do not have the telomerase. They don't express it, and therefore with each division, the telomeres become shorter. And when they are critically short, the cells recognize this as a break in the DNA and they stop dividing. Senescence, it's the consequence. Now, a broken end of a chromosome is a special form of DNA damage. And in fact, the DNA here represented highly schematized actually experience a myriad of exogenous and endogenous genotoxic insults. It is constantly attacked. It can be UV in the skin, irradiation, chemicals. It can also be a normal metabolic byproducts, such as reactive oxygen species and so on, that inflict many different types of chemical alterations to the DNA, ranging from a break to base modifications, bulky adducts, and so on. This has consequences. DNA damage, and this we know for a very long time, can result in changes in the genome, so-called mutations. These are alterations then in the sequence of genes. And if that occurs in a tumor suppressor gene or in an oncogene, then a cell can transform into a cancer cell. So DNA damage by inflicting mutations leads to cancer. We know that for a very long time now. That's why smoking causes cancer, UV causes skin cancer, and so on. But damage can also persist in the genome. It can then lead to a functional loss or even cell death. And this can promote the aging process. So DNA damage is a causal driver for the aging process. Now, DNA damage is not new to us humans. It has challenged the DNA, the genome, ever since DNA became the genetic material. And now DNA repair systems have evolved very early in evolution to repair the DNA. And they constantly need to repair DNA because otherwise cells transform, cells age. And here I'll give you two examples of human patients, children, that are born with a mutation in a single DNA repair gene. Here, and we see a young boy who suffers from a genetic disease called xeroderma pigmentosum. And the name was given to this disease because of these pigmentation abnormalities. And you see here already multiple skin cancers. It's something you would never see in a normal child because skin cancer in us takes decades to evolve from the initial UV damage, here already during childhood. And this is caused by a mutation in a single DNA repair gene that is necessary to remove the lesions that are inflicted by the UV radiation. And here you see a boy who suffers from cocaine syndrome. Also, this boy was born with a mutation in a DNA repair gene. But you can see that these patients are completely different. The cocaine syndrome patient, although the repair system is related to each other, but because it's a different type of repair, here there's no cancer predisposition, but instead a severe growth retardation and premature aging, retinal degeneration, neurodegeneration, arteriosclerosis, leads to a very early accelerated aging process and early death. 1 year after this picture was taken, this patient died. Now, what we can say about the aging process here is that with increasing age, there's a gradual accumulation of DNA damage because despite all the repair, no repair will ever be completely perfect. So there's a gradual accumulation, and at some point, a threshold will be reached where the cell can no longer tolerate the damage load, and it will either not function properly anymore, it will undergo cell death or cellular senescence. And we know that a defect in a single DNA repair gene in humans is sufficient to greatly accelerate the accumulation of damage, leading to very early aging features. Now, human aging is very complex, and therefore the biologists who wanted to study aging had to turn to a very simple model organism. And this is the nematode Caenorhabditis elegans, or short C. elegans. And this animal here in great magnification is usually only 1 millimeter old. It likes to live on fresh compost heaps, and it's present in the garden earth and all that. And it has distinct tissues. Here we see that the feeding organ, we see here already eggs that are fertilized, embryos that are formed. It has an intestine, and many of its somatic cells are in fact neurons. So it has all these cell types that we also have in humans, but in a much simpler fashion. And a landmark experiment was performed in the early 1990s by Cynthia Kenyon in San Francisco. And she did a very simple experiment here. What she did is she took a population of these worms, C. elegans, and just monitored how long they would live in the lab. So these were adult animals. At the beginning, they are 100% alive, and then gradually they die, right? The population, increasingly individuals of the population die when they're wild type. About 20 days, 3 weeks is a normal lifespan here. And she had a mutant, it's called DAF2, it's a mutation that these worms carry. And these worms are incredibly long-lived. They live twice as long as a normal worm. But what's important here is that there is another mutant that carries the same mutation here in the DAF2 gene that normally would double the lifespan, but it has one additional mutation in another gene called DAF16, and all of a sudden there was no longer any longevity. They were normally lived, like a normal animal. And that was the proof that aging is a genetically controlled process. One mutation can lead to doubling of lifespan. Another mutation can suppress this effect. It's a genetic epistasis experiment. It's a genetic pathway. Now you might say, why would I care about a worm? In the following years, experiments in fruit flies and even mice targeting the very same genes showed the same extension of lifespan. So DAF2 indeed has even a homologue in humans, a gene in humans that's called the insulin-like growth factor 1 receptor, highly conserved from worms to humans. And the same mutation leads to lifespan extension in flies and also in mice. Now, how would you know whether the same genes have anything to do with longevity in humans? We cannot do this type of genetic experiments in humans. But there are humans that are extremely long-lived. And the record holder is Jeanne Calment, a French woman who died in 1997 at the age of 122, the absolute record holder. It's rare to have to be a centenarian. But there are many studies in the world that look at what is common, what's different in these families where there are many very long-lived people. And two of those studies, one in Germany, one in Canada, indeed found that there was a variant in a gene called FOXO3A in these families with very long-lived people. And FOXO3A is exactly the same gene that's called DAF16 in worms, the gene that this extended longevity depended on, suggesting that indeed these pathways are highly conserved in evolution. Now in humans, of course, everything is more complicated. In mice, indeed, the homologue of DAF2, insulin-like growth factor 1 receptor, is a receptor. So receptor molecules, these are the eyes and ears of every cell that receive signaling molecules, such here as the insulin-like growth factor. And IGF-1 in signaling in mammals regulates somatic growth. And the ligand here, IGF-1, that's circulating in our bloodstream, that is regulated by the growth hormone receptor. The growth hormone receptor, if it's activated by growth hormone, leads to the release of this IGF-1. And the growth hormone, in turn, is regulated by the pituitary. So much more complex than in C. elegans, the somatotropic axis. And indeed, there are mice that do not develop, they fail to develop a pituitary. These are called Snell and Ames dwarf mice, because they're much smaller than a normal mouse, because there's reduced somatic growth when you don't have a pituitary that produces growth hormone. And in later years, so these are known for a long time, these two mice, these dwarf mice, in later years, a targeted deletion was generated for the growth hormone receptor. And also that mouse was very short in stature. And indeed, these mice were not only smaller, but all of these three mice lived significantly longer than a wild-type mouse, indicating that these mechanisms are highly conserved. How about humans? There are, in fact, humans that have a deficiency in the growth hormone receptor. These are called Laron syndrome, after the Israeli pediatrician Zvi Laron, who first described this syndrome of a growth hormone receptor defect. Now you have, these patients are very rare. Usually you have one here, one there. So it's very difficult to say if you observe something that's different in these patients, is it really because of the growth hormone receptor deficiency or anything else that happens? But there was a village in Ecuador where that was founded by immigrants a long time ago. And so there were a lot of related families there. And there were a large number of Laron syndrome patients. And they were then examined by their physician, Guevara Aguirre, who monitored their health throughout their lives. And you can appreciate they are short in stature, just like mice carrying a growth hormone receptor mutation. And so he monitored them over the years and compared them to the inhabitants of the very same village who did not have this syndrome. And then he compared what they died of. So first the big question, are they long-lived? No, they're not long-lived. But their causes of death are completely different. So the relatives of these people who had a normal growth hormone receptor, they died of diseases, cardiovascular disease, cancer, and so on, and some diabetes. And this distribution of causes of death, that's normal. That's what you find in almost all populations in humans. So we also, here in Europe, we have about 20% of people die of cancer, and cardiovascular disease, more than a third, and all of these things. But there was a completely different causes of death in the growth hormone receptor deficiency people. Here, there were also cardiovascular diseases. Yes, that's the same. there was much more convulsive disorders, so epilepsy, a lot of alcohol-related disorders, accidents, but nobody died of cancer. They were completely cancer-free while they were aging. So that's a major, major difference. And it will be interesting, of course, if some of these
We are so sorry because we have a technical insurance, so
We will be come back.
we will be come back at 5 minutes. Thank you.
Hello everyone. We have a little bit connection problem. Our academician will be here in 10 minutes. While we're waiting, I want a little bit chat with our moderators. Is there any sound? Okay. How did you find this presentation? Could you a little bit describe about it?
We really liked it. It was really beneficial and explained all the questions that's in my mind.
It is really understand first, and when I see the topic, I thought it was really difficult topic, so I thought I couldn't understand it. But the academician is very clear about the presentation. How about you, Altun? How did you find it?
I think good.
Ahmet?
I think while we were comparing the two photos between cancer and aging, that was really good and that was really impressive, especially. I think it's whole the presentation was really good and so impressive, yeah.
Okay, our teacher just takes us, so it can be here any minutes. I hope there won't be any connection problem. It is, yeah, it is really hard in these times from online. We can actually collect the participation question right now when our teacher came, and we can ask directly after presentation is finished. I want to ask a question to Ahmet actually. Why did you choose this professor to present to us?
Because I'm really interested in, especially in aging, of course, in cancer, but also the professor compared both of them, cancer and aging. That was really good, and it was the mixed point, and that was I chose the teacher, also professor. And while the presenting, it was everything was really good, and until the technical problem, we were really good. I believe that. That's all I want to talk about it. Yeah.
Altun, would you like to study in this area? Did you interesting it?
I think interesting, but I don't know.
You can actually study. It is really related with your department.
Yeah.
How about you, Selin? Would you consider to study in this area? Yes, I would like to. Yes.
Do you have any specific areas in your minds, three of them, to study after the graduation, or to make a project while you're in bachelor degree?
Actually, I was thinking about working with 3D printers and tissue engineering, but the cancer and aging related disorders are really exciting, and I would think about us to study at this topics. I will change my mind
So you
after
say,
this presentation.
Yeah.
You say this event change your topic.
Yes, maybe. Yes.
I hope it's a good change. After 5 years, I hope you don't hate us from this change.
No, I don't think so.
How about you, Ahmet? What's in your mind about after graduation topics? Did you choose anything?
Honestly, even I'm studying at chemical engineering, I'm really interested in biology, also cancer biochemical processes. So I'm thinking about do something or research something about that. And yeah, but maybe after the presentation, I can think to remove myself to maybe aging, because the teacher was really good.
Yeah, it is really interesting topic, but to be honest, I get a little bit scared from the hard parts. There's so many complicated testing Arias, even if I'm third year of bioengineering, we still didn't get these topics, and I don't know if we have ever. So I think it's more like if you're interested, you start the research a little bit more every day. Yeah. Have you ever thought about making a project in your departments? Have you any ideas? I'm asking 3 of them, by the way.
Okay. Who wants to start?
I don't have any ideas, so
I haven't heard anything about the project because I'm at the prep class, so I don't know honestly anything about it.
Oh,
I'm so sorry about it.
We have to find literally a project from this event. We are
Yeah.
going to talk about it after.
Yeah, definitely, yeah.
We're definitely going to get in this topic. Oh, I think our teacher might be a little bit more late because he couldn't find the right connection. So we're sorry about that. We're still waiting, but I hope we can make the time a little bit more enjoyable. I want to ask our participation how you guys find this event. Did you enjoy? Is the English was too hard, or you're enjoying? First, I think our moderators could answer this. What do you think about it?
Yeah. You're the bigger one, you know.
Okay,
you know, yeah.
I'll start. I think this event is very beautiful and useful for our future. The English is okay for me. I easily understand. Yes.
Which academicians you actually waiting excitement? I mean, all of them is very valuable guests, but everybody has favorites. Which one do you wait as a favorite? I want to get answer 3 of them each.
Okay.
My favorite was Ali Ertürk because I attended his other events and I knew his name. So also he works with 3D printers and tissue engineering, and he also lives in Germany, and I would like to live in or do an internship in Germany. So I was waiting for him, but he will attend tomorrow. You guys.
Next.
Okay. Honestly, I have really, really big dreams, such as her. For example, such as for example, living in Germany, especially in Germany, for example, Switzerland.
I don't think this is a big dream. I mean, you could do that, and science is science in everywhere. You shouldn't think it's like big. I mean, it's hard, but it shouldn't be biggest one.
Maybe not impossible. For example, you have to say,
yeah, something like that.
Yeah. And my favorite one is this, Mr. Schumacher, even we have some technical problems. But yeah, I believe it's really good, even we have some problems. I really enjoyed them.
Oh, it's nice.
Yeah.
How about you, Altun? Which professor is your favorite one?
I didn't research, really.
You didn't research, come on. You know Altun takes a lot of teachers, but there were a little bit problems because of the pandemic in their school, so she couldn't really get any turnbacks from it, even if some turnbacks get negative. So she had a little bit down about it, but I think you like it right now, right? It is really nice to have, I guess, 12 grades.
Wants to start? Again, I'm gonna say you're the older one.
Okay, 1 minute. We had connected with our teacher. He said he tried to get in. I hope he can come in time because we don't want to wait or other guests. While we're waiting, I want to ask about what's your opinion in Erasmus because everybody asking the Erasmus stage, Erasmus internship, Erasmus plus programs in chat. So I think we can touch that topic. First of all, I want to ask Altun, where do you want to go for Erasmus?
Russia.
Russia?
Yes, Russia.
Okay, we are moving from that. How about you, Ahmet?
I think I want to go to Poland, maybe some Bohemian cultures, for example, Czech Republic, Poland, maybe Slovakia. Even they're not so popular in all around the world, but they have a lot of cultural places such as Auschwitz-Birkenau Nazi camps or Wieliczka salt mines, and a lot of things can be seen by students, especially because some states, such as in Poland, maybe Warsaw, can be sale of discounts, such as for students, and it's maybe cheaper than to visit as a tourist more than students. So I'm gonna choose the Poland, maybe.
Do you have any specific school in your mind in Poland?
I don't think so, but I can trust. I can think of Warsaw Technical University in Poland, but I'm not sure, honestly.
Okay, you still didn't decide. Okay, it's still new, you said.
I'm just at prep school,
Yeah, I know, but while we were preparing these events, I mostly forgot about it because we're always talking about science, biology, chemistry, and after the open up, I start to see you guys. It's not like prep class, like in departments. Next, Selin, where do you want to go for Erasmus, or do you want to even go for Erasmus? I mean, you cannot want maybe.
I would like to go to Germany, but our department don't have any agreement yet. So as a second choice, I would like to go to Italy, but I'm not sure because the lessons are much harder and I don't want to graduate very late, so maybe
we all hope.
Maybe I do my master abroad.
Oh, you want to make your master? Where do you want to make your master? I mean, did you choose any specific university?
No, but in Erlangen or Munich, maybe.
It's nice.
Yeah.
There's a lot of text from our participations. Now let's read some of them because I think they would love to see their comments in the session. So I am going to send you these comments. You can read by order.
Okay. We have some sentences like Unlemel says, actually, I think the rare disease part was the most.
So it looks like there was a connection problem. I didn't see anything on my screen.
Yeah, it was a technical problem. Sorry for that. Yeah.
So
if you're available right now, we can continue the presentation if it's okay.
Okay. So where were we stuck?
We were talking about comparing two photos between aging and cancer.
Oh, that was very early on. Okay.
Yeah, yes. A little bit, yeah.
So, but to avoid this from happening again, how do we do this? Yeah, I will try once more, but
actually we can do that if you make your computer not full screen while you're presenting. You can actually see our reflection or Google Meets platform down below the screen. Maybe that can be work.
Okay, let's see whether this works, because I usually don't use Google. I use Zoom usually, so...
Yeah, we had some problems with the Zoom, so we couldn't get that, sorry.
Yeah, I'm not sure this will work, because... Okay. I can just screen share again.
Maybe we can give information.
But I can only present my entire screen. There's no other option here.
Oh, okay. Maybe we can give information like around 5 minutes breaks, like we can say.
Yeah, just
see it.
Right. Okay.
Okay, then we can do that if that's okay.
Okay, so you see my screen now?
I see it clearly.
We can see, yeah.
Okay, so what we can, so this you didn't see yet?
We saw that, yeah, we saw that.
Tell me, when did you see it?
This page, yeah.
This page, okay.
Yeah.
So I told you here that the causes of death are very different between these Laurent syndrome patients and normal people, where there's a complete protection from cancer and some causes of death that are actually, you know, that could be avoided, and it will be interesting then to see whether there are longevity effects. So there are also in the normal population, there are some reduced variants in the growth hormone receptor, and it was shown that indeed here, when you compare the carriers of these alterations, homozygous carriers, that there's an extended longevity compared to normal people. So suggesting that indeed there could be a pro-longevity effect of reduced GHR. So now, after I told you about genetic pathways of longevity, the question becomes, how are these genetic mechanisms of longevity linked to DNA damage that I told you earlier? And this we looked at here. Here we have now this DAF16 FOXO linked to a GFP, a fluorescence protein, so we can see in whole worms, we can monitor this. And what is happening here is that after DNA damage, this DAF16, this longevity factor, becomes nuclear. It enters the nucleus, has this pattern where the nuclei are, and there it regulates gene expression. So the DAF16 is a transcription factor that responds to DNA damage, and now it drives the gene expression program that is a pro-longevity expression program. And what is the consequence of that? So here we look at two animals that both carry a DNA repair defect that in humans leads to accelerated aging, and the same effect happens in worms. These animals here, they cannot repair the damage, and this is actually still a young animal, but it's already lethargic and it will die soon. This worm carries the same DNA repair mutation. It cannot repair the damage, but it's still agile, fit, and long-lived because it has constitutive activation of this DAF16 longevity factor. This led us to postulate that there are two distinct mechanisms of longevity, two distinct strategies. On the one side, there is DNA repair. When you think about that, damage accumulates gradually over aging. Just stay there, okay. As DNA damage gradually accumulates over aging, it will at some point surpass a threshold where the damage leads to dysfunction. And DNA repair mechanisms are absolutely essential to slow down the accumulation. And a single repair defect in humans greatly accelerates aging. So DNA repair genes that we have are absolutely essential for allowing us to live as long as we do. On the other side, there are longevity genes that do something profoundly different. They now raise the threshold up to which DNA damage can be tolerated. They can also respond to DNA damage and raise the threshold, promote functionalities of tissue, and tolerate the damage accumulation. Now, I want to tell you at the end about interventions of healthy aging. And I give you 3 examples of that. The first example is calorie restriction, which of course is the least popular one, but it's been known since the 1920s, initially in rats, that calorie restriction extends lifespan. And this was shown in budding yeast, in nematodes, in different insects, fish, chicken, mouse, rat, and even dogs. And now the question is, does that work in humans? Now, first here, worms, flies, mice, with reducing calorie intake, these animals are long-lived. Now, in the last link to humans are non-human primates, and here 2 studies in rhesus monkeys were conducted. And these were very long studies, over 25 years. These animals were monitored ad libitum or with restricted diets. And the first study was published in 2009, where indeed there was extended lifespan in these animals that were calorie restricted. And you can see from these pictures they were in better health. And the second study also saw that these animals were in better health. But in the second study, in the second group, there was no extended lifespan. However, in both of these groups, there was a strong reduction of spontaneous cancer development in these animals whose diet was restricted. The difference of these studies was probably the ad libitum diet, that in this first study, this diet was more the North American diet type, while in the second study, it was a more healthy diet. So does it work in humans? There are humans who self-perform calorie restriction, and they report that their body mass decreases, triglycerides are decreased, there's a decreased risk for atherosclerosis and diabetes. So it's possible that there are indeed positive effects. What can we say about calorie restriction in humans? Reduced calorie intake can be healthy, and obesity is a major risk factor. So for a lot of people, obesity is now, it's an ongoing major pandemic, and there is a clear advantage, particularly amid the obesity in our population. But calorie restriction requires an optimum. Even in the simple worms, there's an optimum. If you go too strong on the calorie restriction, there will be strongly adverse effects because malnutrition is very dangerous. And this optimum can be individually very different. Different mouse strains respond very, very different to calorie restriction. And so in humans, with our private different genetic variants and all that, this optimum can be very different. Healthy diet is clearly important, but it was shown that particularly in humans, also regular workout is required. One does not substitute for the other. And of course, we know that diets generally do not work for humans because nobody adheres to diets. And therefore, pharmacological interventions will be required that really target the signaling pathways of calorie restriction. And what was realized is that it doesn't actually matter what calories you take, but how the cells respond to the calorie restriction. And they respond by downregulating a specific signaling pathway, the TOR signaling pathway. TOR stands for target of rapamycin, because the same effect can be achieved by applying rapamycin to directly target the mechanisms of action of calorie restriction. This, however, is a drug that can also compromise the immune system, and therefore there are a lot of efforts being made to improve rapamycin with different dosing and also additional modifications to avoid the immune suppressive effect. The second approach I want to present to you. Let me just check whether everything is still fine. The second effect brings us back to the evolutionary consideration that we took at the beginning. And here you remember that that the soma could age because the germ cells would already pass the genetic information in the offspring. If you now imagine that what would happen to a child if it ages within 1 decade and because there's no sexual maturity, there's no offspring generation, such a species would be extinct. So is there any coordination between the germline and the soma? And here the key experiment came from C. elegans again, where in young animals, the germ cells is comprised of 2 primordial germ cells, here schematically depicted in red. And they are surrounded by 2 somatic cells, somatic gonadal cells. They will generate the tissue around the germ cells later. When you now remove these 2 germ cells and you monitor longevity of these animals, their survival is greatly extended. And you would now say, okay, fine. These animals, they were sterile. They didn't expand any energy into to their progeny. They retained it in their soma, so of course they're long-lived. But that's not so simple, because if you now remove, in addition, also these 2 somatic gonadal cells, there's no longer any longevity. And these animals are short-lived as animals that produce 300 offspring, while these animals are also infertile, just like these ones. So it's about signaling mechanisms between the germline and the soma. Are there now translational implications for that? Is there anything we can think about? And in humans, there were always eunuchs, also at the Ottoman court, in the Chinese court, and at the Korean court. But at the Korean court, these eunuchs, they led a very normal aristocratic lifespan. They lived outside of the courts, had families with adopted children, and lived an aristocratic lifespan. And it was known over the centuries, their life and their birth and death dates were recorded. And so were those dates in the male members of a Mokshin Seo aristocratic families. So we can compare now their lifespan. And we see that indeed there was nearly 20 years extended lifespan of these people, of these eunuchs, indicating that these mechanisms are indeed very highly conserved between the germline and the soma. Now, the third and last intervention I want to present to you, just check everything is fine. The last intervention for healthy aging comes with the idea that aging is systemic. And the question is, are there endocrine factors like hormones, growth factors, I presented you also some, that actually determine the aging process in the tissues? And here you see data from rats, where indeed old and young, when you compare growth hormone levels, it's okay, changes over the days, circadian regulation, but the young ones have higher levels than the old ones, and IGF-1 also declines as a function of age. What if you now put back the young endocrine environment? That's an experiment that was done in Tom Rando's lab, and here they performed parabiosis. They connected the blood circulation of a young and an old mouse, and they looked at the repair of injured skeletal muscles. In young mice and young people, injuries to the muscle are rapidly repaired by the activation of muscle stem cells. In old mice and in old people, this doesn't properly function anymore. Very big problem for injuries and also for surgical procedures. However, when these old animals were exposed to the blood circulation of the young ones, then their skeletal muscle could be repaired again by their own old muscle stem cells. And subsequently, it was observed that the old mice also have improved blood flow and improved cognitive parameters. Now, of course, this has very little therapeutic value to perform parabiosis. And subsequently, it was shown that you could transfer this effect also by the transfusion of young blood plasma. And of course, the most important question is, what are the individual factors that are responsible for transferring this rejuvenating effects? And this is what's currently being pursued in this line of research. I showed you here 3 intervention strategies. Calorie restriction as the most well-established one, the influence of the germline, and where it's very important to identify the signaling mechanisms between germline and the soma, and the growth factors and hormones that could have rejuvenating capacities. So I conclude, the soma ages, the germline can be immortal. Our gene pool has not been selected for extreme longevity. Our gene pool was selected for the traditional lifespan of humans. The traditional life expectancy was 3-4 decades. It has never been selected for the long lives we want to achieve today. The accumulation of damage and aging impairs functioning of cells and tissues. There are genetic pathways that regulate longevity. I showed you some examples of that. Diet and lifestyle can impact aging and aging-associated diseases. The interventions of the future will influence cell renewal and maintenance depending on cell types. There are tissues that require constant renewal, and there are those, like neurons, that need to be maintained lifelong. There will be a paradigm shift in medicine. Nowadays, we are treating disease. The future will be maintenance of health as a therapy target. If you're interested to read up a bit more about that, I've described this in more detail in a book. And with that, I would switch back. I hope that you're still with me, as seemed to be the case. So I can stop the presentation. And I'm really sorry for this hiccup, for this interruption. But I hope you could bear with me, and I'll be very happy to answer any questions about any topic you like, whether it's about what I presented or more about additional topics in academia and research. Thank you very much.
First of all, there is no problem for us because we were really good. And finally, we have some questions. We'll have a lot of questions, but we have to choose them because we don't have enough time.
Okay.
So here's a question for you from us. How did you continue your research with your students or maybe lab mates during the COVID-19 pandemic?
Yeah, that's a very good question. So at the beginning, that was challenging because there were, of course, restrictions. So at the beginning of the pandemic, nobody knew, what measures were most effective. So the very beginning was difficult. Then, as we learned more about effective measures, and there was, at the beginning, there was not even enough protective equipment and things like that. And so we could recover from that in terms of, we always tried also to integrate students in the lab, even master and bachelor students, because it's important that they get experience, hands-on experience on experiment, on what science is actually about, and not just lectures. And so I think we did quite well during that time, trying to keep operations running where they were possible. I mean, there was, except for the delay in the first few months of, so in spring of 2020 when it really started. And with time, we learned better how to deal with it. I think now everybody in the lab is booster vaccinated, so we can actually work quite safe. And so I think we see really light at the end of the tunnel, and we always try to allow people to do their experiments as much as possible, really.
Thank you very much for your answering. And we have another question, if it's possible. What was the moment when you felt really upset or angry in your academic life?
Oh, this happens all the time. I mean, you learn to cope with frustration. I mean, that's a very normal thing. It starts with experiments not working the way you want. That happens all the time. And you need a lot of perseverance. You really have to go through that. And it's also with papers. You get desk rejections, editors who don't recognize the importance of your work, reviewers who don't really understand it and ask outrageous things. There's a lot of frustration, grants that get rejected, fellowship applications that get rejected, all these things, they happen all the time. But they happen to everybody. So you're not the only one. And it's a normal course of life. If you never fail, then you never tried hard enough. So it's part of life of a scientist.
Thank you so much. I was curious about, is prevention of cancer among the purposes of your studies? If yes, how do you plan for this, and how many years or under what conditions it would be possible?
That's a very important question. So every disease can only be solved if it has a large part of prevention. Treatment alone will not solve a disease completely. And cancer is a very complex disease, and prevention is the major advance we have in cancer. Of course, there are now advances with cancer therapy, and it's very interesting, very exciting advances, but the major factor is prevention. So, and I think we have to invest much more in prevention, understanding prevention. As aging is the #1 risk factor for cancer, it's clearly that aging, it plays a major role here. But cancer prevention has many aspects. It starts, for example, by reducing genotoxins from the environment. We know already that reduced smoking habits has a major effect, that really the major drop in, for example, lung cancer, that is absolutely essential. Protection from sunlight, because has a major effect on reducing skin carcinoma. Screening is important. There's a big component of diet. There are dietary genotoxins that are still not completely understood. Some we know, nitrosamines, for example, in over barbecued meat is a big problem. All these things. So there are many aspects. It's complex, but we have to pay much more attention about prevention. It's absolutely essential, and a very, very important part of aging research.
Thank you very much. My question, how did you prefer to study human aging?
Yeah, so that's a very, very good question. So we learned a lot by model system research, particularly C. elegans has been really the workhorse for aging research. But nowadays we can actually move more into human aging because we have very new methodologies. So of course, these genetic experiments we do in model systems, they are not possible. And a lot of the fundamental insights, that we gain in model systems, we could never gain in humans. That's clear. But with the omics methodologies, we can actually now study much more human aging, because we can, for example, a blood sample, we can gain a lot of data on gene expression, on epigenetic alterations, and all these things from humans without any intervention, just drawing blood. And we could learn a lot from that. I think it's a very important area in the future to learn more about human aging, to actually gather data from humans, which is very restricted also by legislation and regulatory limitations. But I think we have to tackle that to get really more insights into the trajectory of human aging.
Thank you for your all answers, and I believe it was a very useful presentation for all our participants, and I think it is interesting topic for everyone. Thank you very much for this presentation. And next session will start at 4 p.m.
Okay, thank you so much, and I hope you enjoyed it, and it was a pleasure being with you this afternoon.
Thank you so much again.
The pleasure
Okay.
is ours. Thank you.
All right.
Hello, everyone. I'm really happy to see all of you here to fourth session in our first day. Now I'm going to leave the word to our moderator, Mustafa Eren. How are you, Mustafa?
Thank you, Ezgi. I'm fine. We are fine, I think. Are
Yes,
we?
I'm fine too. Then I'm leaving the word to you. Have a nice session. Thank you so much, Professor, for this.
Okay, so let me present you the next session. It's a pleasure for us to present our next guest, Annemieke Erasmus-Roost, Professor of Human Genetics at Leiden University. Her field of study is gene therapy, and she has very important studies about Duchenne muscular dystrophy. If you have a question, you can write the check box during the session. You will be able to ask questions at the end of the session. So I end my speech here. Professor, if you wish, you can share your screen.
Okay, thank you. I'm gonna assume that if screen sharing doesn't work, that you'll tell me. So thank you very much for this invitation. And as was already introduced, I've been working on Duchenne exon skipping therapy development for most of my career. And what I want to do in this presentation is to discuss both how the therapy development happens, but also how my career developed at the same time. And I welcome you to ask questions both about the scientific therapy development aspect, but also about the career. So feel free to ask questions about that as well if you have questions, or even if you are looking for advice for yourself. So before we start, my disclosures, and there's a lot, so I'll summarize it for you. So I work on exon skipping, and I work for the Leiden University Medical Center, and the exon skipping work that we developed has been patented. And since I'm a co-inventor on these patents, and these patents have been licensed to companies, I am entitled to a share of royalties. So that is money that I personally get. All my other activities as consultant or advisory board member or speaker at satellite symposiums sponsored by companies, all of that goes to the LUMC and not to me personally. So good to be aware of this for the coming presentation, even though it's not that relevant for this presentation, I think. So what I'll do, I will explain about the development of Duchenne exon skipping. I will first introduce the disease to you, explain to you how exon skipping works, the preclinical development, the clinical development, but also the lessons that we've learned from this development. And as I said, in parallel, I will explain my career. So where am I now and how did I get there? And what I, of course, hope is that some of the career decisions that I made and some of the challenges that I had to overcome will also be helpful for you in your career path. And then at the end, everything will come together in the Dutch Center of RNA Therapeutics that I'm currently coordinating. And I will explain what that is when we get there. So first, a little bit about who I am. So I was introduced already. I'm a professor of translational genetics at the Leiden University Medical Center. And for the past 7 years, I've been claimed to be the most influential Duchenne researchers, but based on the past 10 years, according to Expertscape. So it's not me saying this, this is an external bureau that says this. I've published over 220 papers. I'm a co-inventor of 10 or more than 10 patents. And I've also had jobs as chair of TreatMD. I'll explain what that is later. I've been president of the Oligonucleotide Therapeutics Society, and currently I'm chair of the Dutch Center for RNA Therapeutics. And this introduction is not to dazzle you, but hopefully to give some credibility. So when I talk about challenges, that if you face challenges, that doesn't mean that you will never get anywhere. You can overcome them, and also to hopefully give some credibility of the advice that I give. And again, like I said, feel free to ask questions, not just about the science, but also about career choices and other things. So let me introduce the disease that we're working on. So it's Duchenne muscular dystrophy. It's an X-linked disease, so it means that the gene that is mutated is located on the X chromosome, and because females have 2 X chromosomes, generally they're carriers, so they are not getting sick, while males have only 1 X chromosome, so they are affected. So this is why the pictures that you see of patients in different stages of the disease, they are all males, because the vast majority of patients are male. So the disease is characterized by progressive muscle wasting and loss of muscle function. You can see this first picture, this boy on the top left. You don't see anything wrong with him. What you see in this boy, when he gets up from the floor, you see he rests with his hand on his leg, and that is because there's muscle weakness in his leg. So his legs don't have the power to get up from the floor. He needs his hand to get up from the floor. And this is called the Gowers maneuver. And when you grow old and you get muscle weakness, as unfortunately we all do, you can see elderly people, often when they get up from the chair, they will also use this maneuver. They will rest with their hand on their legs when they get up. You can also see here that they have this strange stand, so they have the belly to the front. That's called lordosis. You can see this boy is walking on his toes rather than on his feet. These are tricks that the boys do, because the muscle that they lose, they don't lose all the muscles at the same time. So some muscles go earlier than others. And what they try is, with the muscles that they have left, they try to compensate to be able to walk as long as they can. Most patients become wheelchair dependent around the age of 10. And then also upper arm function will decline. And again, you can see that with tricks, patients are often able to feed themselves until the age of 16. But eventually they need help for every daily activity, because they can use only their hands, or sometimes even only 1 or 2 fingers. They need assisted ventilation as well, usually around the age of 20. And sadly, mostly they pass away in their 30s or 40s due to respiratory or heart failures, and sometimes earlier due to other respiratory complications. And I think when I tell you this, you think, well, this is really a sad story. This is a very sad disease. Patients, you lose muscle function. These patients must be very depressed. Well, the interesting thing is that most patients have quite a good quality of life. They focus on what they can do rather than what they cannot do, and they don't. Well, sometimes they are depressed when they just lost the function, but generally speaking, they are motivated and enthusiastic, which of course is very impressive, and maybe a lesson for us all to focus on things that we can do rather than things that we can't do. Now, why do these patients have this disease? This is because they cannot make dystrophin. And in skeletal muscle fibers, dystrophin acts as a shock absorber. So during contraction, it prevents muscle damage. And it does this by connecting the cytoskeleton actin, so the part of the actin-myosin complex that coordinates the muscle contraction, to the extracellular matrix. So each muscle fiber has a layer of connective tissue surrounding it and connecting it to other muscle fibers. So dystrophin forms a connection between the intracellular machinery that contracts and the extracellular layer of connective tissue. And you can see here the schematic depiction of dystrophin. And what's interesting is that the functional domain, so the part binding to actin, is located here at the beginning of the protein, and the part binding to the extracellular matrix, the dystroglycan binding domain, is located at the end of the protein. And in the middle, there's repeats. So there's 4 hinges and 24 spectrin like repeats. And we'll get back to that. Now, this is a very severe disease, and it's a childhood onset. So the parents are usually the ones that have to be patient representatives, and that also set up a lot of charities. So it's a rare disease, but a lot of research on this disease has been funded by the patient community. So I think that is important to bear in mind. And of course, when you work on a therapy like this, it's very easy, if you work in the lab, to only see cells, to only see animal models. If you work on a disease, I recommend you to also go to patient days and actually see the patients and the parents, and this is incredibly motivating. But the most important part I want to say here is that a lot of the work, a lot of the research on Duchenne muscular dystrophy has been funded by patient communities. Now, I started working on Duchenne muscular dystrophy in 2000 on a PhD project that we will not go into today because, well, I summarize it here. It wasn't called The Road to Nowhere, but that was a good summary of what the project was. So I was supposed to work on that for 4 years. This is the time it takes to get a PhD in the Netherlands. And in the Netherlands, you're supposed to write at least 3 published papers before you can write your thesis. Now, after 1 year, I sat down with my PhD supervisor, and well, I think we both concluded that this project is going nowhere. With this, the way it's going, things don't work, the system didn't work, the hypothesis probably was wrong. It's impossible to get in 4 years 3 publications out of this, and well, that of course would be in the way of getting a PhD. So what we decided is that I needed a side project. And that side project was called exon skipping. And to explain what exon skipping is, we have to go into the genetics of Duchenne. I mentioned already, Duchenne patients cannot produce functional dystrophin. And most Duchenne patients have a mutation that disrupts the reading frame. So the gene has 79 exons that together code for the dystrophin protein. And what you see here is exon 48, 49, and 50 are missing. You see 47 doesn't fit to 51. What we say then is the reading frame is disrupted. The genetic code is disrupted, and that causes premature truncation of protein translation. So only the beginning of the dystrophin is made. But because it has a linker function, only the beginning of the protein is not functional. So this is what causes Duchenne. Now you can ask yourself, so what happens if the mutation doesn't disrupt the reading frame? Because if you're clever, you can already see 47 would fit to 52. Well, these mutations are found as well, but they're found in patients with a different disease called Becker muscular dystrophy. So you see the example here, 48, 49, 50, and 51 are missing. So actually this deletion is larger. However, the reading frame, the genetic code is now not disrupted. So that means protein translation can continue from beginning to end. For most proteins, missing a domain, that will disrupt protein function. But because dystrophin has this really unique domain setup where the crucial parts are at the beginning and the end, and in the middle you have these redundant repeat areas, these type of mutations give rise to partially functional dystrophins. And when I say, well, they're found in patients with a different disease called Becker muscular dystrophy, well, it's still a muscular dystrophy. So I don't want to belittle the disease, but if you look at it compared to Duchenne, so the onset of symptoms in these patients is later, and it can be anywhere from childhood to midlife. So there's patients that get first symptoms when they're in their 40s or 50s or even 60s. Most patients lose ambulation, but 10 to 15 years after the onset of symptoms, so later than Duchenne patients. And some don't ever lose ambulation, and patients hardly ever need assisted ventilation, and also life expectancy is near normal for most of Becker patients. So again, it's a disease. So compared to the healthy situation, it's not optimal, but compared to Duchenne, it's much better. And that is because these internally deleted dystrophins, well, they give rise to a slower disease progression. So based on this idea or this finding that Duchenne patients don't make functional dystrophin and Becker patients make partially functional dystrophin, and the fact that most Duchenne patients in theory would be able to make these Becker-like protein. So this is how the exon skipping idea came about. So what do we want to do with exon skipping? We want to manipulate the pre-mRNA splicing. So we have the gene, you get a copy of the gene and you get a copy of all the exons, so the protein coding domains, but you also get a copy of the introns, the non-coding parts of the gene. And before the gene can be translated into protein, first all these exons need to be joined together in a process called splicing. And we have again here the example of the deletion of exon 48 to 50, and we see 47 would fit to 52. So if we want to restore the reading frame for this deletion, what we want the cell to not include exon 51, or in other words, to skip it. Now, how can we manipulate the splicing in such a way? We can do that with antisense oligonucleotides, or AONs. So these are small pieces of modified RNA. They bind specifically to an exon, in this case exon 51. hide it from the splicing machinery. So this exon is now no longer included, but 47 is linked to 52. And this allows a Duchenne patient to produce a Becker-like dystrophin. And that is the theory. But of course, what is the reality? So I showed this example already to you. A deletion of exon 48 to 50 would need exon 51 skipping. And what we had more than 20 years ago, we had patient-derived primary muscle cells from a patient with this deletion. So we designed the AONs, the antisense oligonucleotides, targeting exon 51, and we transfected them into the cell cultures. And this is what we saw. So we analyzed the RNA. This is the marker. This is untreated cells. And of course, if you do an RT-PCR, you get the deletion because the patient has this deletion. And after treatment, here, treatment with exon 51 AON, you see a shorter band. And we sequenced it, and indeed, exon 51 was spliced out. So the reading frame should have been restored. So protein production should have been restored as well. And this is what we see here on Western blot. So we did a time course, no dystrophin after before treatment, which makes sense. This is the healthy control. And then after 4 hours, nothing. After 8 hours, nothing. But after 16 hours, you can see a band. And after 24 and 48 hours, you see that it's growing more intense. We also looked 2 days afterwards with immune fluorescent analysis. And before treatment, again, none of the treated, or before treatment, none of the cells were dystrophin positive. And after treatment, 2 days later, we saw that about 75% of the cultured cells became dystrophin positive. So this was really interesting, very promising, but it's cultured cells only. So it's very, very first proof of concept. Now, I showed you this example to explain the difference between Duchenne and Becker and how exon skipping works. I showed the example of an exon 48 to 50 deletion. Now, that is not the only deletion that Duchenne patients can have. So another part of the PhD project was figuring out, well, what mutations are reported. And we know most patients have a deletion, so about 2 out of 3 patients have a deletion, and these deletions cluster in a hotspot. And this has 2 advantages. The first is that the hotspot is in the middle domain, so in the redundant domain, where you know that if you miss one or more of these repeats, the protein can still be partially functional. So that is good. This is, say, an area where you can do exon skipping, because if you have mutation here in the actin binding domain or here in the domain that connects to the connective tissue, it doesn't matter whether the mutations are in frame or out of frame, because even if they're in frame, the protein will not be functional because you need these domains. They are crucial. So that was one advantage. The other advantage is that because the mutations cluster, skipping specific exons can be used for a larger group of patients. So this is a mutation-specific approach. So had the deletions been all over the gene, then skipping each exon would be applicable to about 1% of patients. Now that makes clinical development very challenging because it's a rare disease. And if you then have 1% of a group of rare disease patients, it makes things difficult. It's still mutation specific, so it's still slightly more difficult. But for instance, exon 51 skipping would apply to 14% of patients. And if you then skip also exon 45, you have 9% more, etc. So this is another part of the things that we had to discover and identify during the PhD project. So exon skipping worked out quite well, and this is why I say so. Some PhD students are very worried when they get offered a side project. I think you shouldn't be too worried. If it doesn't work out, you can stop and go back to your project. If it does work out, you can build a career out of a side project. So even though I had 3 years left, in the end, I managed to publish 5 papers for my thesis and get my PhD on time. So that was good. So then I got my PhD. And I think this is probably also a point where you will come. So after you have your PhD, what do you want to do? Do you want to be a postdoc? And if so, do you want to stay where you were, if that's possible? Do you want to go somewhere else? Do you maybe want to go abroad? Do you want to go to an academic group, or do you want to go to industry? Do you want to do something completely different? These are decisions that you have to make. And I think one important thing is it may seem really crucial which decision you make, but actually, whatever decision you make, if you decide later that, for instance, you go to industry and you think, I want to go back to academia, that is possible. The other way around is also possible. If you go to academia and you decide 10 years later you still want to go to industry, that is still possible. So it's good to think about these decisions, what you want to do and why you want to do it. But it's not like you can make the wrong decision and you're stuck somewhere forever. You can always make a choice to go somewhere else. So for me, the decision was, so I had an offer to stay at the LUMC as a postdoc. And at the same time, so the therapy that I had helped develop was going to go and be tested in clinical trials at the LUMC for the first time. So before I knew about all that, I thought, well, I want to go somewhere else to another location, maybe abroad, and learn new things, go into a new group. But then I heard about the clinical trial and the postdoc position that they wanted to offer me. And I thought, well, I can always go abroad. I can always go somewhere else. The opportunity to be, well, involved in a clinical trial with the therapy that you've developed, that probably comes around only once. So I decided to stay at the LUMC. And be involved in the clinical development. And of course, so I am not a clinician, so I couldn't do the injections myself, but I could be involved in the preparation towards the clinical development. So for the clinical development, we started a collaboration at the LUMC with a company called Prosenza. And Prosenza is, so it wasn't a spin-off company, it was a company that was based in Leiden, same as LUMC, so that was very convenient. They were looking for a therapy to develop for muscle diseases, so that was a good fit, and they wanted a therapy with RNA or peptides. So it was really a perfect fit. So we started the collaboration and they were going to coordinate the clinical development because once you start doing clinical development, there's a lot of protocols, rules, regulations that academic groups don't know about, but also they don't have the infrastructure to coordinate this. So we started this development where we did the research and the science, and they worked towards the clinical trial development. So the first clinical trial took place at the LUMC, and this was a treatment where a trial where patients were treated locally in the muscle. So this is a lower leg of a patient, and they were treated locally. Not because we thought that that was the way to go, because well, you have 700 muscles and you need to treat all of them because they're all affected with Duchenne, but because we couldn't test this in healthy volunteers. Because if you skip an exon that restores the genetic code for Duchenne, you disrupt it for a healthy volunteer. And back then we thought, well, that's well, in theory, we could make healthy people into Duchenne. I think now we know we would not be that effective, but in theory, it was possible. Plus, on the other hand, if the therapy does the opposite of what you want in patients as it does in control, then there's not really a point in testing it in healthy volunteers. And we were lucky as well because the chemistry, the AON chemistry that we chose had been tested for other diseases by other companies already. So in discussion with the regulatory agencies, we were allowed to skip the healthy volunteer part and directly go into patients. But then for safety reasons, they said, don't start systemically, start locally. So long story short, you see here, this is anesthetic cream, and this red dot is where the injection is supposed to go. This is the hands of the neurologist injecting the muscle of the patient. You can see here also that the leg of the patient and the foot is fixed. And this is because a muscle biopsy was going to be taken from the patient 4 weeks after the treatment. And the injection site was marked with temporary tattoos on the leg of the patient, but it was marked on the skin. And as you can probably imagine, if you mark something on the skin, if the foot is like this when you do the injection and like that when you take the biopsy, then the muscle in the skin will move independently. So you will actually not take a biopsy from where you injected, but from another part. So that was to make sure that the part that we injected and where we took the biopsy was the same. So like I said, a biopsy was taken 4 weeks after treatment. 4 patients were treated, and after the biopsy was taken, it was stained for dystrophin. So for reference, I show you here a healthy control. So this is a cross-section of a healthy muscle, and dystrophin stain, you can see, is at the border of each of the muscle fibers. This is, for your reference, the way it looks in a Duchenne patient. There is no dystrophin because it's a Duchenne patient. And this is what we saw in each of the 4 patients. And I think it's quite clear, with the same exposure time as these 2 cross sections, it's quite clear that dystrophin had been restored in each of the 4 patients. So that was really positive results. We were very happy. We drank champagne, but we were also aware that local treatment was not the way to go. So before going to the systemic treatment development, a brief intermezzo about my career, because after that clinical trial worked really well, my PhD supervisor was recruited by Procenta. So she went from academia, she went to go work with the company, and that left the exon skipping group at the LUMC without a leader. And my supervisor had said, well, I think you should become the group leader. And he told the head of the department that as well. But the head of the department said, no, Annemieke is too young, too inexperienced. We need to recruit someone externally. Now, the group leader left, and for, well, I think about a year, no one was recruited. There were no vacancies, but I was there, and I thought, well, someone needs to run this show. So I coordinated the group. I supervised the people in the group. I got grants to recruit new people. And then after about a year, I thought, well, I need clarity, because what's going to happen if they now hire a group leader, that would feel unfair because I'm basically leading this group. But if they're not going to hire a group leader, then I should get the credit that I deserve, because I am basically leading this group. So I had a meeting with the head of the department, and I think if you want to have such a meeting, you need to prepare it well. So I had prepared, like, these are all the things that I'm doing, and this is why I think I should be a group leader. But also, if you want to recruit someone externally, that's fine, but then tell me, because then I will go look for a job somewhere else, because I don't want, now that I've led the group, I don't want then to have another leader in the group. Well, after that meeting with the head of the department, he said, well, I think he realized, well, you are actually leading this group and you're doing a good job, so you can formally become the group leader, and then we don't have to recruit someone. And I think sometimes we have a tendency to think that people will see how good a job we do and give us what we deserve. But the head of the department, he said that he didn't even realize that I wasn't formally a group leader. So be vocal in these things, prepare them well, outline why you deserve a certain position, but also don't think that it will just happen. Sometimes you really do need to take a step and ask for what you deserve. Now, back to the development, I mentioned local treatment was possible, but we needed systemic delivery, because about 30 to 40% of our body is muscle, and these muscles are, well, there's over 700 of them, and we need to deliver to all of them. Now, what we knew back then about AONs was the following. So they're very small, about 8 to 12 kilodalton. And very small things, if you inject them systemically, they will be filtered out by the kidney. So it's a good way to make expensive urine, so to say. Now, our AONs had a chemical modification, and that was the 2-O-methyl phosphorothioate modification. And I mention here the phosphothioate specifically because the phosphothioate will cause that the AONs are able to bind to the proteins in the serum with low affinity binding. And this means that there's less clearance by the kidney, because the AONs are bound to the serum proteins, and of course the serum proteins are not cleared by the kidney. So this gives them better bioavailability, and because it's a low affinity binding, there's an on and an off, and once it's off, it can be taken up by tissues. Now, if something is not excreted by the kidney, generally it will be taken up by the liver. And well, that is what we knew about the phosphorothioate AONs from again, other people developing this for other diseases. They're taken up primarily by liver and kidney, and the uptake in muscle is very poor. So that was not a good thing to start with. Nevertheless, we said, let's just try. So what we did, we had 2 animal models, wild type model, mouse with healthy muscles, and MDX mice. So that's a spontaneous mouse model with a mutation in its mouse dystrophin gene. So this mouse doesn't produce mouse dystrophin and has dystrophic muscles. So we injected into the tail vein of the mouse, we injected AONs, and then together with Prosenza, we analyzed where do the AONs end up. Now, as expected, a lot went to the liver. And you can see, well, this scale goes from 12 to 80. So really the vast majority goes to liver for both wild type and MDX mouse. In the wild type mouse, what we expected was seen. So very low levels in the muscle. In the MDX mouse, we saw about 10 times more AONs in the muscle. And of course, the first time you think you've made a mistake, and then you repeat it and you see the same thing again, and then you repeat it again. And at some point we thought, well, we see this every time, so this must be real. And then we started rationalizing, like, what can have happened? And we know that because Duchenne patients don't have dystrophin, they have dystrophic muscles. And because of the dystrophy, proteins, peptides, and other things leak out. They leak out of the muscle into the bloodstream. Well, if things can go out, things can probably go in. So we figured that because of the dystrophic phenotype, the uptake of the AONs is actually better. And the dystrophic phenotype is helping us to deliver to the muscle, because healthy muscle don't take up AONs, but the dystrophic muscles do. So then systemic studies became more feasible. And we tested, well, what should we do? Should we do intravenous delivery, subcutaneous, or intraperitoneal? Now, intraperitoneal you can do easily in a mouse or a rat, more difficult in a human, so you only do it if you really have to. Subcutaneous is something that you could do at home, so you can do home dosing with subcutaneous treatment, but not with intravenous. So we had a preference ahead of time for a subcutaneous. And then when we did these injections, again in mice, and again together with Prosenza, looked at where do the AONs end up, we saw if you compare subcutaneous and intravenous for muscle, you see slightly less uptake after subcutaneous than intravenous. But if you look at the kidney and the liver, where you don't want your AONs to go, you see much more uptake after intravenous than after subcutaneous. So what we realized and what we figured, if we use subcutaneous injection, the amount that goes where it should go is relatively higher than for intravenous, because then you get a lot in the liver and the kidney, and that may lead to toxic results. So this is a more patient-friendly approach, is what we thought. Now, hold that thought, we'll come back to it later. So now we're ready. So we say, okay, we know that we can treat systemically. We know that we should treat subcutaneously. More other preclinical studies were done. So we were ready to say, okay, we're now going to test this approach in patients systemically. And of course, if you treat systemically, you can also measure whether the disease progression slows down. And if you start doing these studies, you need a lot of infrastructure. So you need patient registries. Where are the patients with the right mutation? So the AON that was developed was an exon 51 skipping AON. So where are the patients with the mutations that need exon 51 skipping? Duchenne care is very complex. So where are the good sites that have experience with caring for Duchenne? What outcome measures do we use? And you can see there's a lot of infrastructure that you need. And there is a network called TreatMD that is built to make the field clinical trial ready. That a network launched in 2007. So that was at the same time that we said, okay, we're here, we have this therapy, we're ready for clinical trial. a network was launched to make the field clinical trial ready. Well, that tells you that the field wasn't clinical trial ready when we wanted to start these clinical trials. Now, a lot of work has been done. Things are much better now than they were 15 years ago, but it's still work in progress. There's still a lot of work that needs to be done. So TreatMD still exists, and like I said, a lot has been done. But back 15 years ago, all the tools, the infrastructure that we needed to do clinical trials weren't there. So what did we do? We didn't wait for outcome measures to be developed. We borrowed an outcome measure. So I said, Prosensa was developing exon 51 skipping AONs, and at some point they licensed this to GSK, GlaxoSmithKline. And GlaxoSmithKline said, okay, let's set up 3 different placebo-controlled trials in parallel. So what Prosensa had done with subcutaneous delivery was a dose escalation study in 12 patients, testing 4 different doses for very short term, for 1 month weekly dosing. And after that, the drug was licensed to GSK. And GSK set up a dose regimen study, a dose comparison study, and an efficacy study. So a big placebo-controlled trial. But there were placebo groups in the other 2 trials as well. Each trial had an open label extension study because there is no therapy for Duchenne. Often this is with rare diseases. If there's no therapy available and patients are in a clinical trial and the drug seems to be safe and seems to be effective, then patients can, if they want, continue the treatment. Now, I mentioned we needed a way to measure whether this worked or not. And for that, we needed a functional test. There was none yet for Duchenne. So there was one borrowed from the cardiovascular field called the 6-minute walk test. And it is what it sounds like. So you have 2 cones, you put them 25 meters apart, usually in a long hospital hallway, and you ask patients to walk as quick as they can for 6 minutes between the 2 cones, and then you measure the distance they're able to walk in 6 minutes. And this is an endurance test, which is why it's from the cardiovascular field. But the idea was, well, if you lose muscle function, in your legs, then the distance you're able to walk in 6 minutes will go down. So with disease progression, the distance that patients are able to walk will go down. And if there's a treatment effect, you should be able to measure that with the 6-minute walk test. Now, before going into the 6-minute walk test results of all these clinical trials, I want to mention side effects because they occurred. So after the first month of weekly treatment, there were no side effects. But after longer treatment, there were local injection site reactions. And I said earlier, subcutaneous, we said this is patient-friendly. Turned out that was not the case, because by then we knew also from other clinical trial developments with phosphatidylate AONs for other diseases, if you do repeated subcutaneous delivery, at some point patients will develop injection site reactions. And this is not like some redness that goes away, but it leads to ulceration, scar formation, the subcutaneous fat can go away, discoloration, and these things turned out to be irreversible. And some patients develop this after a few months, some only after a year or even later, but if you kept treating patients, they all developed this. So that made the subcutaneous delivery not as patient-friendly as we had hoped. Had we chosen the intravenous delivery, this would not have happened. But well, hindsight is easy to say that, of course. But yeah, in hindsight, subcutaneous delivery was not patient-friendly. Other things that were seen was proteinuria, so protein in the urine. This was reversible during treatment breaks and subclinical. And there was also in a few patients thrombocytopenia was seen, so a reduction of blood platelets, but there were no bleeding incidents, fortunately. And I will show you a picture of the skin reactions, just so you can see that they varied between patients. You can see here discoloration. For some patients, this was not an issue, but some felt really self-conscious about it. You can see here that the subcutaneous fat is going away, and this is one of the more severe patients. You can really see that there's scar tissue formation and that most of the subcutaneous fat is gone. And well, you can see this is not a patient-friendly way to deliver. This is really not good. So what about function? Well, the first graph that I show you is of the open label phase after the first dose escalation study. So 12 patients got a low dose, a higher dose, and it turned out the highest dose resulted in most dystrophin restoration, 6 milligram per kilogram. So that was offered to all patients in the dose escalation study. And then the clinicians doing this open label study said, well, since we're going to use the 6-minute walk test in the placebo control trial, let's follow these patients in the open label study also with the 6-minute walk test just to see how well they do. Well, if you count quickly, you see 10 lines. There were 12 patients in the open label study, but 2 of them were non-ambulant, so they were already in a wheelchair. Now, the 6-minute walk test implies that you need to walk. The majority of Duchenne patients can't walk because they're in a wheelchair. So this is a disadvantage of the outcome measure. And what you can also see is that the 2 patients that started with the lowest baseline lost ambulation within a year. But the other 8 that had a higher baseline, they were stable for over 3 years. And here they received weekly treatment. And at the end, they received 8 weekly treatment and then had a 4-week break to reduce the burden of having to go to the hospital every week, because subcutaneous delivery can be done at home. But once you're in a clinical trial setting, of course, you need the regular controls at the hospital. So this was very encouraging because, well, stable was not something we associated with Duchenne. However, it's open label, and also we didn't really know what the normal trajectory was of the 6-minute walk test in Duchenne patients. So we really needed the placebo control trials. So I mentioned already there were 3. The first one was in 51 patients, and it was a dose regimen study. So patients either got placebo, the blue line, and this is the baseline. So this is the 6-minute walk test. If it goes down, patient lost distance. If it goes up, patients increased distance in the 6-minute walk test. In placebo group, you can see that patients lost about 30 meters in 1 year. And these were patients from 6 to 8 years old. We know now that this is roughly what they lose. In red, patients who were treated every other week, and in green, patients who were treated every week. The total dose was the same, but some patients received it every week and some every other week. And what you can see is that these 2 groups seem to do better than the placebo group in their walk distance. You see that there's a lot of variability, but on average, the lines suggest that there is a treatment effect and that the 6-minute walk distance is better for the treated patients than the untreated patients. Then there was also a dose comparison study done. Again, in blue placebo, this is half a year treatment. Patients lose about 15 meters, again, 6 to 8 year olds. This is a low dose, 3 milligram per kilogram. Well, I think we all agree it's suboptimal because there's no difference with placebo. And this is the high dose, the dose that was also used in the open label, the dose that was used in the previous graph that I showed, 6 milligram per kilogram. And again, you can see these patients do better. Now, these phase 2 studies were done in young patients between 6 and 8. The phase 3 trial was bigger, so it included 186 patients. And these were patients between 5 and 16-year-old who were still ambulant, but there was much more variability. However, based on the results that I've just shown you, I think the whole field was very eagerly anticipating the results because they thought this phase 3 trial is going to show that this drug works. But they didn't. So here are the results for you. In blue placebo, and again, with time, patients are able to walk less in 6 minutes. In green, the treated group, they're also able to walk less in 6 minutes after a year of treatment. And they're able to walk slightly less, if that makes sense. So they do slightly better than the placebo group. But this difference is about 11 meters. So they were able to walk on average 11 meters further than the placebo group. That is not statistically significant. And even if it was statistically significant, it's not clinically relevant that you're able to walk better, that you're able to walk 11 meters more in 6 minutes. That is about 1.5 meters per minute more. So what had happened? Well, there's things that we know now that we didn't know when the trials were planned. And one of them is, what is the trajectory of the 6-minute walk distance? And what we assumed is that, well, when patients grow older, with time, they lose every year a little bit until they reach 0. And this is linear. Well, turned out that's not the case. When patients are less than 7 years old, they're relatively stable, and they can be stable for even 3 years. Now, if you have a drug that slows down disease progression, you can't measure that in stable patients because, well, whether you treat or not, they will be stable. Then in patients over 7, you can see here that they do slowly lose a distance. But what we didn't take into account is that it doesn't go from 200 to 150 to 100 to 50 to 0. No, at some point patients are able to walk roughly 200 meters, and then they lose a few more muscle fibers and they go from 200 meters to 0. So there's a very rapid decline. And probably by the time they're in that rapid decline phase, even restoring dystrophin in these few muscle fibers that they still have left, that won't stop the loss of ambulation. So there's a point of no return. And in the phase 3 trial, all of these patients were there. So there were patients who were stable regardless of whether they were treated, and there were patients who were really rapidly losing ambulation. So again, regarding or whether they were treated or not, they would lose ambulation. And then there were patients who were also in this phase 2 trial group where you would be able to measure a treatment effect. So what happened then? GSK dropped drisapersen, and drisapersen is the name of the exon 51 AON. And then Procensa got all the results back and were acquired by another company called BioMarin. And BioMarin did additional analysis. And they said, well, if we compare the phase 3 trial patients to the phase 2 trial patients, we see that the phase 3 trial patients have a more advanced disease. Now, what if we look at patients in the phase 3 trial who had the inclusion criteria that would have allowed them to be in these phase 2 trials, where we could measure a difference? Well, if we look in those patients, we do see a response. So in this subgroup, we do see something. So they went then to the regulators, so the Food and Drug Administration in the US and the European Medicine Agencies in Europe, and they said, well, look, when we planned the trials, we didn't really know the natural history. We do now. Knowing what we know now, this is the group that we looked in. And if we look in this group, we see a response. Well, the FDA said, that's a lot of post-hoc analyses. We really don't like that. Plus also there's these side effects that you observe. We don't think we can approve this drug. So the approval was denied. Well, with FDA, everything is public. With EMA, things happen behind closed doors, so we don't know what was said there. But we know that one day after the meeting they had with the EMA, just to have, say, an exploratory meeting, BioMarin withdrew their application. Now, if the discussion had been extremely positive, they would not have done that. So I think we can assume that that was a negative discussion as well. And then what BioMarin announced was, we will withdraw, so Kindrisa, another name for drisapersen, we will stop developing this exon 51 AON because it's not good enough. What we need a version 2 that works better and that has less side effects. So back to the drawing board, basically. So what did we learn from this as a Duchenne field? Well, we learned, first of all, that the interaction with the regulators started too late. So we were really busy in the lab, optimizing the AONs, doing everything in cell models and animal models, showing that everything worked, but we didn't consider that we needed outcome measures until we needed them. It would have been much better if we'd worked on them in parallel. So by the time we did the clinical trials, there were no outcome measures and there were no natural history data to allow an optimal trial design. And if you have suboptimal trial design, it's really difficult to pick up minor treatment effects. Because if you ask me now, did drisapersen work? I say, oh, I don't know. Maybe it did work. Maybe it didn't. If it worked, it wasn't a miracle drug. It didn't do a lot of things, but it might have slowed down the disease progression to some extent. Now, fortunately, we did learn from our mistake. So what's happening now is that we have an ongoing dialogue between academics, patients, and regulators in the EU and now also in the US. There's new outcome measures that are being developed that don't only measure arm or leg function, but also arm function, because I mentioned already, majority of Duchenne patients is in a wheelchair. So future trials are better. So at least we learned from that. And we also learned that when you start talking to the regulators, that it's a bilateral education. So not only did we teach them, well, this is Duchenne, these are the challenges, this is what's important for these patients, but we also learned from them. How do they evaluate drugs? What is important, what is needed? And of course, that helps you to make outcome measures that are fitting. And there's also the EURORDIS summer school. I'm not sure whether you're familiar with it, but EURORDIS, the European Organization for Rare Diseases, every year organizes a translational summer school where they teach about the regulatory system and drug development to patients and patient representatives and to researchers. And I think that's a unique opportunity to learn about the regulatory system, because I've not come across another educational tool that offers this to academics. So where are we now? The driver person development stopped. In parallel, there were other AONs targeting other exons with the same chemistry. All of that stopped. Currently in the US, there's 4 AONs approved. They have a different backbone than the driver person, the so-called morpholino backbone. There's 4 approved, 1 for 51, 1 for 45, and 2 for exon 53. But they are approved based only on dystrophin restoration at very low levels. And the question is, are these low levels enough to slow down disease progression? And that's something that the companies that have developed these AONs, they have to confirm that. So clinical trials are still ongoing now. We know that the trick works, but it works only very limited, mostly because the delivery to muscle is too low. And the question is, these small amounts of dystrophin, are they enough to slow down disease progression or not? So then to the final bit, and I'll go quick because I do want to also have time for questions. What we realized is that this approach has potential, and it especially has potential for patients with cryptic splicing mutations. Now, what are those? Cryptic splicing mutations are mutations within the intron, so the non-coding parts that make part of the intron recognized as an exon. So they're included into the messenger RNA and they disrupt protein translation. Now, these cryptic exon you can block with an ASO or an AON, you can hide the cryptic exon, and then you get restoration of the normal transcript at a normal protein. So it's not like with Duchenne, where you think, well, is this internally deleted dystrophin functional, yes or no? You restore the normal protein. So these cryptic splicing mutations are very good targets for exon skipping. And where delivery to muscle is really difficult, if you do systemic delivery, the AONs won't go to the eye and they won't go to the brain. However, if you inject locally, either intrathecally or intraocularly, the AONs will be taken up by the cells of the retina for the eye and by the central nervous system after intrathecal delivery. So that allows you to inject really low doses and get high local exposure with very low systemic exposure. The treatment frequency is quite low, so 2 times per year for the eye and 3 to 4 times per year for the central nervous system. So we thought, well, if we combine this, if you combine these cryptic splicing with these eye and brain diseases that need local delivery, that would be a really good way to use AONs. And maybe you've heard of nusinersen. It's a drug for spinal muscular atrophy. That makes use of the same chemistry, makes use of intrathecal delivery, and that works really very well. So can we also do this for these very rare diseases? Now, the challenge is these very rare diseases and these very rare mutations, they're a bad business case for companies, because if you have a single patient, well, you can't really develop a drug for one patient and sell it. You won't make a profit. So can we instead develop this as academic groups? Well, this is something that is possible, and that was shown by Tim Yu. And I'm showing you here the publication that he wrote in 2018 in the New England Journal of Medicine. So he found a patient with Batten disease who had a cryptic splicing mutation. And well, Batten disease is a brain disease. So what he said, okay, I can develop an ASO specifically for this patient, so uniquely developed for this patient. The patient was called Mila, so he called the drug Milazen. He designed the ASO. He tested them in fibroblasts from the patient. He had discussions with the FDA. What should I do for toxicity? He did rat toxicity, and he applied for an investigational new drug, and was allowed to treat the patient for the first time within a year after finding the mutation. So that was really quick. The patient had a lot of epileptic seizures, and after he started treating her, the number and the duration of epileptic seizures went down drastically. Sadly, treatment started too late to save her, so she passed away last year. But because of the reduction of epileptic seizures, parents said that the quality of life for them and for Mila had improved a lot. And they also said that had she been diagnosed earlier and had she been treated earlier, maybe the effect would have been bigger. Now, when we heard the story, we were inspired, but we also thought, well, hang on, we are working on exon skipping as well. This is something that we could do as well. So several Dutch university medical centers are working on exon skipping. So we said, let's not compete with each other. Let's join forces. Let's set up the Dutch Center for RNA Therapeutics. So it's a collaborative effort of the Leiden University Medical Center, but also the Radboud University Medical Center, where they focus on eye diseases, and the Erasmus Medical Center in Rotterdam, where they focus on neurodevelopmental diseases. So we joined forces, and what we want to do is to develop exon skipping AONs for patients with very rare mutations causing a brain or an eye diseases. Patients still should be expected to have benefit from the treatment. So for instance, if someone is fully blind, we can restore the missing protein, but that will not restore vision for these patients. And of course, patients must also be willing to undergo this experimental treatment, because there's no guarantee that this will be safe or effective. So if you want to know more about this, then the website is rnatherapy.nl. And as it happens, this Monday, we will celebrate our 2nd anniversary. So these are the people involved in the Dutch Center for RNA Therapeutics. So it's not a group of preclinical scientists. We have help also from neurologists, ophthalmologists, patients working on in the pharmacy, patients working in people working in animal facility, etc. So it's really a big group. So finally, some retrospective career advice. I think don't be afraid of side projects. I think that is clear. Nothing is ever a waste of time. So even things that don't work out, you learn from them. But don't indefinitely continue with something that doesn't work. At some point say, okay, we need to do something else. Don't be afraid to fail. Making mistakes is human. Everyone will make mistakes. Make sure that you learn from them. Know your strengths and weaknesses, because that will allow you to choose things that would fit with what you can do and that fit with what you can do. Find your own niche. So don't do what everyone else is doing, but try to find something that is specific for you. Indicate to your superiors what your ambition is. Don't think that they will know if you don't tell them. Work hard, but not too hard. Also have time for a leisure time to rest. Help other people if you can, and also find good and nice collaborators. If there's a collaborator where you feel like I'm doing all of the work or 99% of the work, that's not nice. If you are collaborating with someone where you say, well, we share equally, it's fun, those are good collaborators. So try to find those and try to work with those if you can. And then finally, the acknowledgement of the work that I showed, the group and our collaborators, and of course also our funders. And with that, I will stop sharing and try to go back. Yeah, I found you.
Okay, Professor, thank you for your slides. So we are having some questions, and my friend is going to ask.
Firstly, let me introduce myself. I'm Rabia Köse. I'm first class bioengineering at Gebze Technical University. If you want, I can question to you. Kübra Özdemir, first of all, thank you for your presentation. What can you say about the negative and positive returns about pursuing an academic career abroad and then working abroad?
So that's a very good question, because I think this is usually in many countries, not just in Turkey, not just in the Netherlands, everywhere, people think that you need to go abroad, and that's a positive experience. And if you don't go abroad, then your career will go nowhere. Well, I never went abroad, and I still made it as a professor. So I think that shows you don't necessarily have to go abroad. And it's always also said, I mean, going abroad can be a positive experience. You can learn a lot, but of course you can also go somewhere where the atmosphere is not nice, where it's horrible. So I think going abroad it can be a good experience, but then make sure that you choose a good lab. And don't only look at the publications like, oh, they Nature, Science, I have to go there. Also talk to the people. What's the atmosphere like? Do they expect you to work 7 days per week? Do they supervise you well, or are you just left by yourself? Or because you have these groups where 4 people get the same project, and then after 1 year they don't talk to each other. After 1 year the professor looks, who got the most out of this, and the other 3 are fired. So that's not what you like, what you want. You want to have an opportunity, you want to learn. So don't only look at what do they produce, but also talk to the people in that. Like, I mean, I'm considering to go to your group. Can I talk? What's the experience like? And make sure that you find a group where you get a good supervisor. Because if you don't get a good supervisor, yeah, you're left on your own, and it's really nice to have a good supervisor to together work on a project. So I think with that, going abroad can be very nice experience. It's good. You extend your network. You learn to new people. You learn to be independent, but it's not the only way to get there. And I know that some people, they can't go abroad. I mean, if you're married and your husband or your wife has a job, yeah, then you can't. I mean, you're stuck. Maybe you have children. Maybe your parents are old and sick and need you. So in that case, don't think that you can't have a career in science, because you still can. But just be clever about it. So what I did, so I never went abroad, but of course this came up again and again in all the job or the grant interviews. And at some point I got I just emailed a group that I was collaborating a lot with, can I get a visiting professorship with you? And now I visit, well, not now because there's pandemic, but before the pandemic, I would visit them 3-4 times per year for a week. And then that's your, say, abroad experience. You go there. So you can also be creative. And so if you do it, make sure that it's fun and useful. But if you can't, don't think that's the end of your career.
Okay, Professor, thank you for your advice. You mentioned a really good point. And our next question comes from Yasemin Albayrak. Thank you for that inspiring presentation, especially for the part that choosing our sides in our career and which sides we are going to head in future. That was a relief for all, I'm sure. I wonder, even being a group leader was completely compelling. What was your inspire and although that the hardest part of that issue? Thank you again for all.
Yeah. So I think so. Of course, I became a group leader quite quickly. So I mean, I got my PhD, and then 2 years later, I was a group leader. And at first, as a group leader, I could do some experiments in the summer break because, well, there were no work discussion and not a lot of other work going. But very quickly, so this is the challenge. Once you become a group leader, you don't do experiments anymore. Other people do experiments in the group. And I think if you're in science, the experiments are fun because you have an hypothesis or you want to test something. And the most exciting part is when you get your results, like what will come, what will happen. And that you lose that when you become a group leader, because, well, you can be excited for other people, but you're not there when you see the cool results. So I think that is one challenging part, that at some point, if you're a group leader, you have to realize that, well, the more advanced you get, the less experiments you will be able to do. So that was challenging. And I think for the rest, I was lucky that I'm a very good communicator, and that helps because communication is key to everything. If you're not a good communicator, there's trainings for that. So I did some like leadership trainings, and I would recommend that to everyone. Even if you think, well, but I'm a good leader, yeah, but it's still good to realize why certain things work and why certain people, for instance, everyone has their own type of personality, and there's people that you can work really well with, and there's people that you just don't work well with. So be aware of these things so that you can also maybe avoid that. So I would say people skills and personalities that can be challenging, but also there learn from it. If you have someone in your group that you think, well, this is not working out, well, at some point they will leave, and then it's over, and you learn, and you know, okay. I mean, nothing against that person, but if there's not a match, it's difficult. So that was, I think, a challenge, and I think another thing to learn if you become a group leader is to say no. Because when I was a PhD student, I used to think, well, once I'm a postdoc, I won't be as busy. Well, I got busier when I was a postdoc, and I got even busier when I became a group leader. So at some point, you have to say no, because if you don't say no, there won't be time to sleep, and that's just not productive. So also there, learn to say no. And yeah, it's not fun, but it's better to say no than to say yes and not be able to deliver. No one likes that. So I think that's another advice and challenge. And I think another recommendation that I would give. So I'm very fortunate because I've been sharing an office with another group leader for the past 10+ years. And we get along really well, but because we're both group leaders, sometimes something happens where you think, is it okay that I'm upset about this? Does it really make sense or not? And having someone then to have a reality check, that's really good. So try to find someone where you can have this reality check. I mean, I can ask my husband, but he's not an academic group leader, so he doesn't know whether it makes sense or not. But this office mate, she does. And then, yeah, she can say, no, you're overreacting. Or she can say, no, indeed, that doesn't make sense.
Thank you for this answer. Fatma Tarhan says, dear professor, thanks for presentation. If you mentioned in presentation, I missed, sorry for that. But what I'm curious about is, do you have a specific process to find the deletion part on gen, which is the cause of disease?
Well, I did not mention that, so you didn't miss it. So there are diagnostic protocols to find mutations in Duchenne patients. So most patients have a deletion or a duplication, and there's an MLPA test that's usually run. Currently also next generation sequencing, of course. So there's a whole protocol to make sure that mutations are found. And that is followed. So that's not something I do. That's something that the diagnostic lab does. And depending on the country that you're in or the academic hospital that you're related to, either they just look for duplications and deletions, but 1 in 4 patients has a small mutation, and sometimes they don't look for that. So that's something to be aware of if you're in the Duchenne field, that some patients are told they don't have Duchenne, but then they still do.
Thank you, Professor. And the next question comes from Ahmet Furkan Çodur. First of all, thank you very much for a beautiful presentation. What was the moment that you felt angry and upset in your academic life?
Oh, well, actually, I felt angry and upset just yesterday. Well, I felt angry and upset more frequently, but yesterday. So I won't mention names, but I'll just mention why I got angry. So we're working on a project together, and the deadline kept being postponed. So the deadline used to be somewhere in January, and I had done everything that I was supposed to do already by the end of December. And then they said, no, we're going to postpone it to February. No, we're going to postpone it to March. And I said, well, March is difficult because I'm very busy in March. Last weekend I had a meeting, this weekend I have a meeting, and tomorrow I have another meeting. So I won't be able to do anything. During the week I'm super busy. March is fine, but don't expect anything from me. And then they emailed me yesterday and asked for a Zoom call, and they said, well, actually, we need you to work on this weekend because we need your input for this. And I got angry and I said, sorry, I would love to, but I can't. And I think I got angry with them, of course, because I announced that I wouldn't be able to help. But they also got angry because now the final product won't be, say, up to my standard. So that's that's probably not a major thing to be angry about. But it's, yeah, I think these things happen. And I think other things that get me angry is when so some of the companies working on Duchenne therapies are really good and they do it for the patients. And one of them, we were in a big meeting and it was from a Duchenne charity. And some of the company people, they kept looking whether their stock went up. Rather than listening to the stories of the patients and the parents, like all their emotions with the disease, they were looking the stock price. And I thought, that got me angry as well.
Thank you again. Anıl, thank you for your wonderful presentation, Professor. I wonder, when creating scientific studies, which is the most complicating part, designing the first part, the statistic is, the statistic or anything else?
I think, well, it probably what is most difficult will vary per experiment, but some tips for doing experiments. Don't try to set up the perfect experiment, because it's really inviting to try and do everything. Let's test the optimal, what is the optimal dose, what is the optimal so to test 15 different things in one experiment and then think you get the answer to everything. What will happen? You will get inconclusive results, so you won't know anything about dose, route, etc., the best timing. So try to ask one question and do the experiment to answer one thing and then move to the next part. And I think if you do it in that way, setting up the experiment will become much easier because the number of variables is a lot less. So I think I'm trying to make it easy to myself. And I think for statistics, I am not a statistical wizard, but we are fortunate because at LUMC we have a medical statistics department. So as soon as statistics become really complicated, we can go to them and we explain, well, this is our problem, because I mean, sometimes you have longitudinal studies in animals and you have different time points and you have different groups, different doses. Things become complicated, and then they say, okay, this is the statistical model that you have to use because of this. And I think that has helped. So maybe another lesson, if you know that statistics is not your strength, find someone to help you, because usually people are there willing to help.
Thank you, Professor. And our next question is from Şeyda Yağcı. Thanks for your valuable presentation. As students at the beginning of the road, how can we stand out on this path?
I think so. I think first of all, so don't be afraid to be yourself. And it's what I see in current day students is that often they look at other people. Like, but they have done this, and they don't look at what they themselves have achieved. So look at your talents and see what you can achieve, and you can stand out on your talent. Everyone has shortcomings. No one is perfect. So be aware of that. I mean, statistics is not my strength. So I'm not going to go into the medical statistics and try to have a career there. That doesn't make sense. So work with your strengths, see what you can do. And there's some things where you say, well, it's not my strength, but I can at least learn the basics. So statistics is not my strength, but I do know the basics because, well, I need some of that. So try to get training for what you know. Well, I will need this, but it's not my strength. But also nurture your talents, nurture your strengths. And I think if you do that, you will stand out. And maybe also one tip, if you want to do an internship with a group, for instance, we get tens per week, people that want to do an internship. Those that looked up, like, okay, what is the science? What are you doing? Those we will answer. But the ones that do a very generic mail, like, I want to do an internship in your group, and you see, well, they copy pasted everything and they emailed 15 different groups. Yeah, then no, I want the motivated people. So if you want to go somewhere, put in the time and the investment to show, look, I really want to go there because I put in time and effort.
Thank you for beautiful answer. And the next question, Aleyna Zahir, you have a successful career as a scientist. Apart from being curious, what motivates you to work?
Well, I think what helps for me is that I work for patients. So I work for Duchenne patients, and now with the DCRT also for patients with rare brain and eye diseases. Seeing Duchenne patients is incredibly motivating. So there used to be in-person meetings from Duchenne organizations in the Netherlands, but also in Italy, in the UK, in the US. So every year I would meet patients and parents, and that motivated. And now these things are online, and it's still motivating. Because like I said, these patients have every reason to be very depressed and angry, but they're not. And of course, that makes it very motivating to work for them. So I think that is that motivates me. And there's also an intrinsic motivation. I mean, I think as a scientist, you want to know the answers. You have these questions and you want to know the answers. I think as soon as you realize that you're not that motivated, then it's time to, well, look somewhere else, or say, see, because, well, so we went with this Dutch Center for RNA Therapeutics. I find, well, that's motivating. I'm learning new things. So I think if at some point you realize I stopped learning, I stopped growing, then it's time to do something else. And it can be go somewhere else, but it can also be try to, well, go into a different topic in your own group or field.
Okay, Professor, our participation question is finished now, and we have a few questions for you. From all the diseases, what attracts you about Duchenne?
Well, I think multiple things. So from the patient perspective, the fact that the patients cope so well with a very devastating disease. But I've talked to patients with other diseases, and I think that is in many diseases, patients are heroes in how they deal with their disease. But for Duchenne, that is at least the case. Another aspect is that the Duchenne gene, so the dystrophin gene, is the largest gene in the genome. It's very complicated. So it's, from a genetic perspective, really interesting and fascinating. And every time you think you've seen everything that's possible, there's something new that will turn out to happen, and the way that it's processed. So just to give you one example, it takes about, well, anywhere between 10 and 16 hours to transcribe the gene. So just imagine that we're talking here now. If your polymerase starts transcribing the gene now, it's done in 16 hours. So that makes it very fascinating as well from a genetic perspective. And I think what I also like is that it's really versatile, because you work with fundamental science, you try to develop a therapy as well, you work with clinicians, you work with patients, you work with regulators. So that makes it really very interactive and versatile.
The last question from me: how did you prefer to study human genetics and your research area?
So I always thought that immunology was the coolest thing. So I thought I wanted to be go into immunology and study immunology. But I did an internship at genetics because I like genetics as well. So I did an internship in genetics and I did an internship in immunology. And after the internship in immunology, I got offered a job and I went there. And after about 3 months, I realized that at least back then in immunology, it was like a big field with a lot of different proteins and arrows going everywhere and question marks everywhere. So there was a lot that was not known. Well, in genetics, there's a lot that we don't know, but at least we have some basics. We know there's a mutation. We know that there's a protein missing. So I started thinking like, no, maybe immunology, there's too much unknown. There's too much speculation. I want more certainty, and I really like genetics as well. So then there was a PhD position in the genetics field. So that I applied, and I think that's even though the project went nowhere for the first year, I realized that I like genetics much more than immunology.
So thank you, madam, for participating in our event today. It was a pleasure to have you today, and we wish you have healthy and good days.
Okay, thank you very much, and good luck with the rest of your events.
So much, Professor. It was very lovely to meet you. It was really helpful to listen your all advices. We will try to adapt these advices in our school too. Thank you.
Good. Okay, thank you very much. Bye.
Session will be started at 5:30. See you guys later. Hello everyone. This is our last section for the today, and here we come here with Esma. She will be our moderator for last section. I'm going to give you the word to Esma. How are you, Esma?
I'm pretty good, thank you. And you, Ezgi?
I'm fine, so thank you. I'm giving you word to you. Have a nice session.
Thank you. I'm Esma Şeval-Yeşil, and I'm at prep year just before studying at the Department of Molecular Biology and Genetics at Gebze Technical University.
My name is Cemre Halfaoğulları, and I am also a prep student, and I will be majoring in molecular biology and genetics next year, just like Esma.
Before we may start the session about watching the brain works, it's a pleasure for us to present our honored Professor Adam Cohen from Harvard University that has been studying chemistry, chemical biology, and physics for years. Professor, if you are ready, I leave the word to you.
Great, thank you. I'm just getting my computer configured. Wonderful. Am I speaking to the group now?
Yes.
Yes, okay, great. Well, good evening, everybody. It's nice to meet you in some virtual sense. First, I'd like to thank the organizers for inviting me to visit you virtually today. I'm in very cold and snowy Cambridge, Massachusetts, and it's wonderful that through modern technology I can come to virtually to Turkey. I hope that we'll have a chance to meet in person, either in Turkey or here in Cambridge someday before too long. I've had several really wonderful students from Turkey work in my lab over the last few years, and I hope to have more. And I've never been to Turkey in person, but I hope someday that I'll be able to visit. All right, so let me see if I can share my screen. If I let's see if this works.
There's a button.
Can I
Right
move this one? Let's see.
Yes.
Wonderful. Did that work?
Yes.
Okay. Great. So I'm going to tell you today a little bit about my own personal history through science and how I came to the current projects that my students and I are working on. And then I'll tell you in a little bit more detail about some of our work on trying to image the function of the brain to see how the brain works. So let's see if this works. There we go. So I grew up in New York City, right in the middle of the city. And for the first 12 years of my life, I went to this school here. You can see the school is in an old fortress. It had no windows anywhere in the school and was a very, very urban environment. One of the charms of growing up in New York City is that there's a lot of electronics garbage on the street. People throw out computers and televisions and other broken devices. And one of my hobbies during school was to pick up broken electronics from the garbage and take them home to my bedroom and take them apart to see how they worked and to try to build new electronic devices. And through an interest in both electricity and then also in electrochemistry, which is the interface of electricity and chemistry, that's really how I initially became interested in neuroscience, because of course the function of our brains is a mixture of electrical and chemical signaling. And by learning about these two areas, I became fascinated by the mystery of how these combine to make our own brains work. It's sort of the most fascinating computer there is. Then as an undergraduate, I was a student here at Harvard, and I became interested in how one might build a computer out of molecules and how one could miniaturize electronic devices even further, and again in electrochemistry. And these areas too, I didn't realize at the time, but were eventually leading to and preparing for ultimately an interest in neuroscience, because you can think of the brain as a computer composed of many nanoscale and molecular parts. Then after I graduated from college, I spent a few years in England at the other Cambridge in the UK. And there I studied theoretical physics and the theory of how molecules interact with each other. But I really wanted to do experimental work. I really like building things with my hands. And so after that time, I did a second PhD degree at Stanford in beautiful California, where I studied single molecule biophysics, so the motions of individual molecules in solution. And then since 2007, I've been at Harvard, and I started my lab in the chemistry department here. I'm joint between chemistry and physics. And when I started my lab, I really had no idea what I was going to work on. And we explored different areas for a few years, and I'll tell you about some of those. And then over the last maybe 5 to 10 years, we've shifted to focus mostly on neuroscience. So one point that I hope to make from showing this trajectory is that it's possible to change fields many times during your studies, and that it's not necessary to to stay in one area forever. Often when students, when I talk to students and they're trying to decide what to study, it can feel like that is a decision about the future of your whole life. And of course, whatever you study will affect the trajectory, but often it's in unpredictable ways. For instance, a lot of the mathematics that I learned studying theoretical physics is now mathematics that we use for trying to understand the brain. And so looking backwards, I can see how the topics that we study now emerged from the different things that I studied when I was a student. But looking forward from being a student, I never would have guessed that this is where I would end up. And even after starting my lab and becoming a professor, we've continued to change directions and explore new areas. One aspect which is important to remember is that the different disciplines that people traditionally define, chemistry, physics, biology, and so on, are a little bit like lines on a map where people have created these boundaries between the disciplines for administrative convenience or for logistical reasons, but very often they don't actually correspond to anything in the real world. And similarly, many interesting problems in the real world don't fit neatly in these disciplinary boundaries. Okay, so this is a photo of the first big project which I worked on. This is when I was still in high school. I built a scanning tunneling microscope, which is a kind of electron microscope for imaging very small structures down to atomic resolution. And I built this in my bedroom in New York City, in our house. And the way a scanning tunneling microscope works is you have a very sharp wire. And if you take a wire and you cut it just right, there can be one atom that sticks out a little bit more from the tip of the wire than all the other atoms. And then if you take that wire and you move it very close to a sample and you apply a little voltage, you can get a little electric current between the wire and the sample. And the current depends very sensitively on the distance between the wire and the sample. If the wire gets further away, the current goes down. If it gets closer, the current gets bigger. This current occurs through quantum tunneling of the electrons between the wire and the sample. And so then by moving the wire over the sample and plotting how much current there is as a function of position, you can make a map of the surface of the sample. And it's really incredible. You can build one of these devices. It was about this big, and we were able, and I was able to take very high resolution pictures of different conducting surfaces. You can see here this these gray blocks, and you might sort of be able to see there are some ropes here, some elastic ropes. One of the problems in building this in New York City is that we lived on the 18th floor of an apartment building next to a highway and next to a helicopter landing pad. And so there were a lot of vibrations in the building. And for this very sensitive mechanical measurement, the vibrations were a big problem. And so I encased the whole thing in some clay and some modeling clay to absorb some of the shock. And then when my parents weren't home, I drilled a hole in our ceiling and hung this device from the ceiling with some elastic cords so that it would be decoupled from the building vibrations. And that was enough to isolate it from all the noise of the city. The inside core of this is actually Lego that I used for the structure to hold all the pieces together. And then there was some electronics. This device is now in a museum in Ohio. Okay. Then when I was an undergraduate student, I mentioned I became interested in this question of whether one could build electronic devices out of molecules. Nowadays, of course, all of our electronic devices are made out of silicon and semiconductors, but biological computation happens in molecular devices. And there's no guarantee that silicon is the best substrate for doing computation. There are lots of different kinds of materials out there, and one could imagine that you could build computational platforms on very different kinds of substrates. And of course, many people have tried things which haven't, it's very hard to compete with silicon because it's so good, but it's worth exploring. And so I became interested in the question of whether one could use electrochemistry as a substrate for building transistors or electrical amplifiers. And we wanted to do experiments where we had just one or two molecules between electrodes. And as you can imagine, it's very difficult to connect clips to an individual molecule. It's just too small. And so we came up with a very simple scheme for measuring very small numbers of molecules electrically. So what you see here is a view of two glass cylinders with the liquid metal mercury in each of these cylinders. One's pointing up and one's pointing down. And then we would extrude very small drops of mercury from these cylinders. And now if you just touch those drops of mercury to each other, they'll fuse and merge with each other. But if you coat the mercury with one molecular layer of a molecule, then that single layer can protect the surface of the mercury. And because the mercury is a fluid, the surface is very, very smooth and it's flexible. And so then you can take these two mercury drops and touch them to each other, and they actually won't fuse with each other. And then you have just these two molecular layers in between the mercury drops. And so you can sort of see here, here's one of these drops, and here's the other drop. And there's wires connected into the back of these glass tubes. And then you can apply voltages and measure the electrical properties of of the monolayer or bilayer of molecules between the two mercury drops. So in some ways, this was related to the scanning tunneling microscope device that I had built at home. But this was an even simpler device because we didn't need all of the vibration controls because the mercury itself was so flexible that it provided this molecular contact. All right. So now I'm going to jump forward and tell you about some of the things that we explored when I started my lab at Harvard. This was in 2007, roughly, when I started. And when I started my lab, I remember being very nervous and very scared because we had an empty room, just the bare walls and nothing in it. And I had no idea what I was supposed to do. And so my students and I spent a lot of time, basically breakfast, lunch, and dinner. We would sit around and we would talk about what are the most interesting questions and unsolved mysteries and problems in the world, and is there anything that we can do to address these problems? And so one problem we became very interested in is the magnetic navigation of birds and insects. So people have observed that some animals, some birds, some insects, maybe some fish, seem to be able to navigate with the Earth's magnetic field. And this is a mystery. We don't have any sense of magnetism ourselves, as far as we know. And nobody knows what is the primary transduction mechanism. How can it be that these birds and bugs can sense this tiny magnetic field from the Earth? If you ask most physicists or even most chemists whether a small magnetic field can affect chemistry, biochemistry, most people will tell you no, that it can't. And the argument that people give is that the energy of interaction between an electron spin and a typical magnetic field, even one from a fairly strong magnet, is many thousands or millions of fold weaker than thermal energy. And so chemistry is usually governed by the temperature, by thermal energy, the thermal energy of the molecules. And so if you change those energies by 1 part in 10 to the 5 or 1 part in 10 to the 6, it shouldn't make any difference. And so this is why when a chemist says, okay, I did the following reaction, they always specify the temperature, concentration, and pressure, but nobody bothers to specify what the magnetic field is. Right? But it turns out that there are some chemical reactions where really tiny magnetic fields can completely change the outcome of the reaction. And these effects happen through a quantum mechanical coherent process, which can happen in a liquid at room temperature. These are mostly photochemical reactions. So there's light involved. And I'll tell you the basic physical scheme. And if this if you haven't had the quantum mechanics, if this doesn't make sense to you, don't worry. We'll come back to other topics in just a minute. So in most molecules, in their ground state, all the electrons are paired up. You have one spin up and one spin down in every molecular orbital. When you shine light on some molecules, you can sometimes knock the electrons apart from each other so that these two electrons don't talk to each other anymore. Then two things can happen. The electrons can come back together and you can go back to where you started. Or if one of the electron spins flips over, then the electrons can't come back together anymore because of the Pauli exclusion principle. And so then you go down some different reaction pathways. And it turns out that if you apply a magnetic field to these molecules, you can control how the electrons spin in space. And you can control the rate at which they flip between this state and this state. And if that happens faster than the reaction, then the magnetic field can affect the reaction. And so let's see, I have a little animation. Let's see if this plays. So, okay, so these, what I'm showing you here are the orientations of the electron spins. So red and blue are the two pieces of the molecule, the two electrons. And we're going to start one pointing up and one pointing down. I should say that when I do this to represent the electron state, what I'm actually representing is what's called a quantum mechanical singlet state, which is a superposition of this and the two electrons being like this. So it's halfway like this and half. And so what I'm showing here is the superposition of the red one is up and the blue one is down, or the red one is down and the blue one is up. And then this is a simulation of the trajectories of all of these electron spins in different magnetic fields. And you can find that they get randomized over time. So to explore how magnetic fields could affect chemistry, we set up a very simple experiment. Here's a little glass container with a solution in it of some molecules that are sensitive to a magnetic field. And we're shining an ultraviolet light on this container from the side. And then we're looking at the fluorescence of these molecules through a fluorescence filter here. That's this black ring. And this tells us about the outcome of this light-driven reaction. Then over here, there's a piece of cardboard. And then over here, my student has in his fingers a little magnet. Okay? And so the cardboard is just to be a light block. So whatever he's doing with the magnet doesn't affect what we're seeing in the reaction. And so I'll show you a movie where we're looking at this solution. And then I'll subtract off the background light on the left-hand side. And then you'll see what happens when he brings the magnet up to the solution. So what I hope you can see is that when the magnet comes close to the solution, the fluorescence gets brighter. And then when he pulls the magnet away from the solution, the fluorescence gets dimmer. Can you see that? And so this is a very mild magnet, completely changing the outcome of this chemical reaction because of this quantum mechanical effect in the solution. And so it's actually a quite dramatic effect. And we've been using this to explore the quantum mechanics of electron spins in solutions at room temperature. We still don't know whether any chemistry like this is actually relevant inside of the birds and insects. Nobody has identified, well, we don't even know if they exist, but whether there are molecules in living animals that have chemical reactions that are sensitive to magnetic field. So the question of how animals sense magnetism is still an open question. Okay. The next area that we started working in has to do with the field of chirality. And so what I hope you see here is all of these studies that we're doing are sort of at the interface of chemistry and physics and maybe a little bit of biology. So this painting here is of Louis Pasteur, who I assume you've heard of. He was famous for many things. Of course, he developed pasteurization and many of the early vaccines. When he was young, when he was 25 or 26 years old, he also discovered or invented the concept of chirality. And so that's what I'll tell you about for a minute. So a chiral object is something like my hand, where the mirror image of the object cannot be superimposed on the original. So there's nothing I can do to my right hand to turn it into my left hand, right? No matter which way I point it, you can always say, oh, that's his right hand, right? And so similarly with screws, there's a right-handed screw and a left-handed screw. And rotating a right-handed screw around doesn't turn it into a left-handed screw. And molecules can be chiral too. The most famous example is DNA. All of the DNA in every living thing on Earth is a right-handed helix. Proteins are chiral. And so because most of the molecules that make up our bodies are chiral, it means that our interactions with other molecules are also sensitive to the chirality of those molecules. So, for example, many of the drugs and medicines that we take come in a handed variety. There's only one hand that is pharmacologically active. And from a drug development perspective, we would treat the mirror image molecule as a different entity. It will have different interactions with biology. And there are famous examples from history where, before people knew this, where drugs were given as a mixture, and one hand of the drug was effective, and the other hand caused very severe side effects. There was a drug called thalidomide, which many women took in the 1950s to prevent morning sickness when they were pregnant, so that they wouldn't feel so bad while they were pregnant. And it turns out that one hand of that drug was effective, but the other hand caused birth defects in many of their babies. And this was a big tragedy for many thousands of people. And so now we're very careful to develop drugs of just one hand and not the other. Now, it's very difficult to synthesize molecules of just one hand because, of course, the energy of these two molecules is the same. And if you start with molecules that are not chiral and you just mix them together, you always get 50% of one hand and 50% of the other hand. It's also a mystery how it came to be that all life on Earth has a particular handedness. So you can ask, would life be the same if everything were the mirror image? If we had left-handed DNA and the opposite handedness of proteins and everything were backwards. And as far as we know, it would be the same, but something early in the history of the Earth broke the symmetry and made all of life have right-handed DNA. And so both for the synthetic chemistry reasons and just to understand why life is the way it is, we became interested in whether there are physical things you can do to a chemical reaction that can break the symmetry and cause it to prefer one hand over the other hand. And one of the things you can do is, it turns out that, so just as objects can be chiral, light can also be chiral. And the electromagnetic field, all the little arrows of the electric field and the magnetic field, can have a chiral arrangement. And the most famous examples of this are circularly polarized light. So you can have light where the electric field goes in a helix, and you can either have a right-handed helix or a left-handed helix. And it turned out that the interactions between chiral light and chiral molecules are quite weak. And so the effect of the handedness of the light on chemistry is a very small effect. Anyways, so we explored the question of whether you could engineer the electromagnetic field. Could you come up with other solutions to Maxwell's equations that could have a larger twist in them than circularly polarized light, where it would be extra chiral and have a larger interaction with molecules. You know, when I was a student, I spent maybe half of a year studying electricity and magnetism, and I was tortured with all of these different solutions to Maxwell's equations, and there are so many solutions. And so I thought maybe we could come up with one which was more twisty. So after thinking about this, we were able to cook up some solutions to Maxwell's equations, which are super chiral, which have much more twist than regular circularly polarized light. And with those super chiral solutions, we were able to drive chemistry to have preferentially form one hand or the other hand of molecules. Okay, so that's another thing we've worked on. All right, so now I'm going to shift gears a little bit and tell you about what my lab has been focusing on for the last few years, which has to do with electrical signals in biology, in us. Okay, and so so this is a person, right? And if you zoom in by about a factor of 10, you see that we have these brains, right? And if you zoom in by another factor of about 10,000, you can see that the brains are made of neurons out of these cells. Inside our brain, we have about 86 billion neurons. And each one of these neurons has about 10,000 connections to the other cells in our brain. And if we zoom in by another factor of 10,000, we see that the neurons have a lipid membrane which separates the inside from the outside. And this membrane is an electrical insulator, and you have water solutions on both sides with ions in it. And so you can think of this a little bit like a parallel plate capacitor. There's two conducting media with an insulator in between. You might notice that this looks a little bit like that mercury drop experiment, which I worked on in college, but instead of mercury, we have water on either side. It's the same idea. And there are proteins called ion channels, which sit in the membrane here. And these proteins can open little pores, which can let ions through the membrane. And that can then change the voltage in the cell. And so as the ions flow in and out, the voltage can go up and down. Neurons use this to communicate with each other. So when a neuron fires, it makes a little electrical impulse, which is about 1/10 of a volt tall and a millisecond long. And this is the internal language of the nervous system. So every thought and feeling, emotion, sensation that we have is represented in these little electrical impulses going through the 86 billion neurons in our brain, right? That's all there is. That's our entire mental life. And so we'd really like to be able to see these impulses to understand how the brain works. And this is an artist's view of what we would like to be able to do. But the problem is, if you just look at some neurons, you can't see the voltage in them, just like you can't see the voltage in this wire by looking at the wire, right? So you need some way to convert the electrical signal into something that you can see. And for decades, scientists have tried to find some molecule or chemical which can convert the electrical activity in neurons into something we can see. And we found a way to do that. And so that's what I'll tell you about now. The story starts here. This is a view from an airplane looking over San Jose, California. So this is the city of San Jose. And this is the San Francisco Bay here. This is the ocean. And you see these beautiful colors here. These are ponds where people are evaporating seawater to make salt, okay? And in this very salty water, there are microorganisms that are growing in the water, which are producing proteins which are colored. And it's the color of those proteins, which you can see from the airplane, which gives the water this color. So now if we zoom way in by a factor of 10 to the 12, so these are the proteins that you can see from the airplane over San Jose. And these are transmembrane proteins called microbial rhodopsins. They're structurally similar to the opsin proteins in our eyes that we use for vision, okay? So these are proteins that sit in the membranes of cells, and they have covalently bound inside the protein a little chromophore, a colored molecule called retinal, okay? And it's this molecule of retinal which gives the color that you can see and makes these proteins sensitive to light. And these proteins come in all the colors of the rainbow, and there's many thousands of them which have been found in the wild. These proteins mediate many different interactions between sunlight and the biochemistry of cells. So for instance, bacteriorhodopsin is a light-driven proton pump. So light comes in and it pumps protons from inside the cell to outside the cell. There are light-driven chloride pumps, which pump chloride ions into the cell. There are light-activated ion channels, which open up a pore and let current through. And there's light-activated signaling molecules. Okay, so there's a very diverse set of proteins to convert sunlight into different biochemical signals. So about 17 or 18 years ago, people realized that you could take the genes for these proteins and put them into other cells, and then those cells would become responsive to light. And so, for instance, you could take one of these genes and put it into the brain of a mouse, and then the neurons in that brain would become sensitive to light. So let's see if this works. So just a second. So let me show you. Do you see the video screen?
Yes, we can.
Okay, great. So this is an example that my friend Feng Zhang did when he was a graduate student. Okay, so for the chemists and physicists, this is a mouse. It's a small animal, right? And it's in a bucket, and you can't quite see it, but there's an optical fiber going into its head. And this mouse has a gene called channel rhodopsin, a light-gated ion channel, in the part of its brain that controls its walking. And so the mouse is here, and it's exploring in this bucket. And then at a certain point, they'll turn on a blue light, and it makes the mouse run in circles, right? And you see this mouse will keep running in circles as long as that blue light is on. And then when they turn off the light, the mouse will go back to its previous behavior. Okay? And this is because that light is activating neurons in a particular part in the mouse's brain. This has been a very powerful tool for investigating which parts of the brain control different kinds of behaviors. Here, I'll show you another example where they put the channel rhodopsin gene in a part of the, oops, let me try that again. In a part of the brain which controls aggression. So there, do you see this mouse here?
Yes, we can.
Okay, so here's a mouse, and here is a rubber glove in its cage. And normally the mouse would just sniff the glove and basically ignore it. And you can see there's an optical fiber going into the mouse's brain. And let's see if this plays. Oops, there's some problem with the video. Let me try one more time. If this doesn't work, we'll skip it. There we go. Okay. Is it working? Oh, there you go. So when the you'll see when the light turns on, the mouse will attack the glove. So when the light is on, the mouse gets into a fight with this rubber glove. And then when the light turns off, the mouse stops fighting with the glove. Okay? And so here you'll see the beginning of the movie now. Okay? So you can think of this like laser mind control, right? By shining the light into different parts of the brain, you can control different aspects of the animal's behavior. So here we have these proteins where light comes in, and this leads to a change in the electrical activity of the cells. And so I said to myself, I wonder if we can run these proteins backward. Can we use changes in the electrical activity in the cells to produce an optical signal that we can see? Okay. And so our efforts in that direction start here. This is a photo of the Dead Sea between Israel over here and Jordan over here. So the Dead Sea is a very salty body of water, but it's not really dead. There are microorganisms that live in the water here. And one of these organisms, it's called Helerubrum sodomense. It produces a protein called archirhodopsin 3, which absorbs sunlight and pumps protons, hydrogen ions, from inside the cell to outside the cell. Those protons then come back through an enzyme called the ATP synthase, which is a rotary motor that spins and takes ADP and phosphate and makes ATP. And so this is how this creature makes a living from sunlight. You can think of this protein like the world's smallest photovoltaic device. Sunlight comes in and a voltage comes out. The protons get pumped. We discovered, basically by luck, that this protein is a little bit fluorescent. You can shine red light on it, and it emits in the near infrared part of the spectrum. But the fluorescence is very sensitive to the voltage on the membrane. So when you have a change in the voltage, that changes the fluorescence of the protein. Okay? And so then we took the gene from this creature in the Dead Sea, and we put it into a mammalian cell. This is a human cell that's in the microscope. And you can't see it, but we have a little glass tube stuck into the cell. And we'll apply steps of voltage between plus and minus 1/10 of a volt. Okay? And we'll look at the fluorescence of this protein. And so what I hope you can see is that when the voltage goes up, the protein gets bright. And when the voltage goes down, the protein gets dim. Is the video coming through?
Yes, it is.
Yes.
Okay, great. So now we have a way to convert electrical activity in cells into light that we can see. So then we took a rat brain and we mushed it up and we grew the cells, the neurons from the brain, in a dish. And then we used, again, a little electrical connection to the cell, a little glass capillary, to trigger the neuron to fire. And we took very high-speed video of the neuron firing. And this was the first time it was possible to directly see the electrical activity in a neuron. Here on the left, I'm showing fluorescence in red and the electrically recorded voltage in blue. And you can see that they correspond to each other. But in this experiment, we had to use a little glass tube to trigger the neuron to fire. And that's very, very slow and difficult work because you have to get this tube into a single cell. And so it takes about 1 hour per cell to do this. But I just showed you that there are these channel rhodopsin proteins where with blue light, they can make a neuron fire. And then I told you we developed these voltage indicator proteins where with red light, when the neuron fires, we see the response. And so I said, let's just combine these two things together. If we put them both in the same cell, then we can tickle the cell with blue light and make it fire. And we can look at the response with red light. Right? So just like that. That's the cartoon anyways. So here's some data. Each of these blue bars shows a flash of light to the neuron. The red is the fluorescence from the cell. And the black is an electrical recording that we did at the same time just to check. But we don't need the electrodes anymore. Now we have just an optical interface to the neuron. So we can stimulate them with blue light and record from them with red light, right? And one of the charms of having an optical interface is that now we can do experiments in a spatially resolved way. So instead of just looking at the voltage at one point, we can map how the voltage spreads. And this is very important for trying to understand the brain. With a traditional technique of using electrodes to record from one cell at a time, that's a little bit like trying to watch a movie by looking at one pixel on your computer screen. You can sort of tell when things are happening, but you don't really understand the plot. To really see the plot, you need to understand the correlations that between when one person does one thing and somebody else does something else, that there's an interaction, right? And so similarly in the brain, we need to see the plot, we need to be able to map the voltage. So here's an example where this is still just one neuron. We targeted the blue light just to the center of the neuron here. And then we mapped the electrical propagation at a very high speed. In this case, it's at 100,000 frames per second. So 10 microseconds per frame. And this is what we see. And so I hope you can see that we can actually map how the electrical impulse propagates through the cell. So it turns out there are some practical applications of some of these tools. The examples that I showed you so far were in mouse neurons. And I really wanted to look at human neurons. And so I asked my students if anybody would donate their brain so that we could mush it up and study the neurons. But they were reluctant to do that. And so and this is a big challenge in biomedical research. As I'm sure you know, diseases of the brain are some of the hardest diseases to treat. We really don't have good medicines for Alzheimer's disease or Parkinson's disease or depression or schizophrenia or autism. None of these diseases has good medicines. And there's many reasons for that. But one of them is that it's very difficult to get access to brain tissue from live patients because the brain doesn't regrow. If you have a cancer, the doctors will just cut it out. But for brain diseases, we rarely go inside the brain. But it turns out that over the last years, people have developed tools for getting access to human neurons from live people where the neurons are genetically identical to the neurons in the brain of the patient. And here's how it works. This relies on the fact that all of the cells in our body have the same genome in them. They're all genetically identical. It's just different genes are turned on in different cells. People now know how to take some skin from a person, and by adding certain factors to the skin cells, you can take them backward through embryonic development and turn them into what are called induced pluripotent stem cells. These are like the cells in an early embryo. You can then add some other factors to those cells and turn them into neurons. And you can grow those neurons in a dish, and those neurons are genetically identical to the neurons in the head of the person. You can do this on neurons from a healthy person or on neurons from somebody who has a genetic disease which is affecting their brain. And then you can compare and try to develop medicines which will make the behavior of the neurons from the sick person more like the behavior of neurons from a healthy person. Okay? And so this is an opportunity now, by combining this technology with the imaging that I just showed you, to explore and to develop new drugs for diseases of the brain. And so about 7 or 8 years ago, I started a company which combines these technologies to develop new drugs. And so I'll tell you a little bit about my company, which is now sort of a side project. So we take skin from people and make neurons, and then we use these voltage imaging tools and we test different drugs to find drugs that we hope will restore normal function of neurons. And I'll give you just one example. This is a photo of a patient with a disease called amyotrophic lateral sclerosis, or ALS. This is a disease which causes the motor neurons, the neurons that control the muscles, to gradually die. And this disease can strike without warning. We don't know what causes it, and there's no treatment for it. It kills everybody who gets it, typically in about 2 years. So it's a very terrible disease, and there's no treatments. So we have been taking a little bit of skin or blood from people like this patient, and you can now turn those cells into neurons, into the specific kinds of neurons that are affected in the disease, and then grow them in the dish, add these proteins from the Dead Sea, And then you can look at those neurons and you can actually see their activity. And then you can use that as a platform for testing drugs. And so we've been developing drugs now for several different diseases, for ALS, epilepsy, and pain are the main areas that we're focusing on. And some of those drugs are now starting to go toward clinical trials. And so it's just incredible to me that we can take this gene, which came from the Dead Sea, from some little organism, and now it's being used to study neurons from people with real diseases. Now, everything I've showed you so far is in cultured neurons, neurons that are spread out in a dish. We were also interested to look inside the brain of a real animal so that we could actually see with sensory inputs and behavior how the neurons fire. And this has been technically very challenging because the brain is not transparent. It's sort of cloudy. And in a live animal, things are moving around and you have blood flow. And so there's many technical challenges associated with this. But over the last years, we've developed some tools which now let us see into the brains of live mice. And so this is a mouse, again, and it has we've taken out a piece of the skull and replaced it with a little glass window. And we can look into its brain. And here the mouse is on a treadmill, and so it's walking on the treadmill. And we can look inside of its head now and look at the neurons that are firing as the mouse runs on this treadmill. Okay? This is what the experiment actually looks like when we're running it. So it's a whole room full of lasers and optics. The red is for imaging the activity of the neurons, and the blue is for stimulating the neurons in different ways. And so together, it's like we have a function generator and an oscilloscope, and we can go in and try to understand how the circuits are working inside the brain of this mouse. And these are what the data look like. So each row here is a recording from one cell. And red are what are called excitatory neurons. These are neurons that activate other neurons. Blue are inhibitory neurons that silence other neurons. On the left, the mouse is sitting still. On the right, the mouse is walking. And you can sort of see, if you just look at this, that the activity is different when the mouse is sitting versus when it's walking. And if you zoom in, there's a huge amount of information that you can now see here. You can see what are called the subthreshold events. These are the reflect the inputs to the neurons. And you can see there's correlations here between the different cells. And then these are the spikes, the outputs of the neurons that then will trigger other neurons to fire. And I won't go into the details of the neuroscience, but with these tools, we're now starting to look in different brain regions to try to understand how these circuits function. So I want to end this section by just talking a little bit about the molecular tools that are the basis for all of this. These genes come from all over the world. The voltage indicator comes from here in the Dead Sea. The channelrhodopsin, the light-gated ion channel, comes from an alga, which comes from a pond from the south of England. We fuse the channelrhodopsin to a fluorescent protein, which comes from a coral, which lives in the ocean. And then we use another green fluorescent protein, which comes from a jellyfish. And then we actually link these two proteins together with a little gene which comes from a virus that infects pigs. And it's just incredible to me that we can take all of these genes from all over the world and stick them together to come up with these new tools, which never, wouldn't have come up in evolution. And the only reason we can do this is that ecologists and botanists have been wading through the mud for decades, describing these seemingly useless organisms. A few years ago, I was in Israel, and I was fortunate enough to meet the ecologist who had discovered this organism in the early 1980s as part of an ecological survey. And he had no idea that 30 years after his discovery of this little bug, that it was being used to study neurodegenerative diseases in human patients and being used to develop new medicines. And so it's very hard to know where basic research will go. It's also very important to conserve and to protect these places, because we can only borrow these genes from nature. We could never invent them on our own. And so we really need this diversity in order to develop these kinds of tools. All right, so that's all I was going to say about research. In the last few minutes, I thought I might just share some general thoughts about some strategies for success in science and research. And, you know, I mean, I don't know that much about success in science. I'm still trying to figure it out. But these are some things that I've observed from looking at other people. So here are some photos of some successful scientists. You might recognize some of these people. This is Isaac Newton. He's, of course, very famous. This is Louis Pasteur, who we talked about. This is Thomas Edison, who invented the light bulb. This is George Washington Carver, who was an American scientist. He was born into slavery. He was born as a slave in the American South and then was freed after the American Civil War. And he developed many of the agricultural techniques which were crucial in the advancement of agriculture, particularly in the US, but also all over the world. This is Emmy Noether, who was a very famous theoretical physicist who developed many of the foundational concepts which we use in modern theoretical physics. And this, of course, is Albert Einstein. And one point I want to make about all of these people is that at the molecular and cellular and sort of physical level, they're really no different than you or me or anybody else, right? The only thing that distinguishes these people is how they chose to spend their time and the kinds of problems that they worked on and the ideas that they had. But in principle, anybody can just get up and do the same kinds of things that these people did. And so here are some thoughts about some strategies which can be helpful in trying to develop a sense of scientific aesthetics and to pick problems that are interesting and important. It's you know, it's very important to think about what inputs you take in. And nowadays, you know, with the internet, of course, you can watch videos of talks. You have access basically to all the knowledge in the world at your fingertips. This was different from even when I was, you know, I'm not that old, but when I was a student, we didn't really have this. And so, you know, you had to go to talks in person. But now, you, of course, as we're doing, you can have talks from all over the world. And so, but just listening to the talk isn't really enough. It's important when you hear ideas or hear a talk to think about, you know, which parts you like, which parts you don't like, and why, and what you would do differently, or how you would take the research further, or what questions are still unanswered, to really think, to grapple with the material and to try to find how you could move it forward. In order to solve important problems, it's important to have in your mind some sense of what kinds of problems you'd like to solve. And so, you know, there's many obvious ones like how to make renewable energy more effectively, how to combat climate change, understanding how the brain works, treating different diseases, and so on. So the world is full of problems. And it's useful then when you hear talks or read things to check them against all of your problems, to think, you know, does this piece of information help me with any of these problems? Now, unfortunately, we all only have time in our lives to work on a few things. And many important projects will take years of work. And so choosing the right project is often the most important aspect of doing research. I have a friend who's also a professor who said to me, you know, Adam, if I knew what the next 20 Nobel Prizes in physics or chemistry would be given for, I could probably do half of the experiments. And that's true. I mean, between Wikipedia and once you've had some basic training, almost anybody can actually do almost any experiment. And so what distinguishes the people who are most successful is people who have good taste in picking experiments that are just at the boundary where it's difficult enough or unusual enough that you don't have lots of other people trying to solve it at the same time, but still possible, tractable, right? There are some problems which are probably too hard for current technology to approach. And so finding the things where you can make that difference is really the key aspect. If you go back to these people, one interesting aspect of all of these people is that it seems that they got lucky many, many times in their lives, right? Each one of these people has many different discoveries in different fields that are attributed to them. Like with Pasteur, we talked about both chirality and vaccination, and same for all of these people. And so part of what distinguishes them is they had very good taste in picking their problems. Now, okay, so how do you pick a good problem? It's no matter how smart you are, you don't want to just work on your average quality of idea. You want to work on something that's like 3 or 4 standard deviations better than your average idea. And the only way to do that is to make lots and lots of ideas and then to get rid of most of them, right? And so, and it doesn't cost, a bad idea, if you don't spend a lot of time on it, it's cheap, right? It's easy to come up with bad ideas and then just throw them out. And so since I was about 10 years old, I've kept a notebook, well, now it's a file on my computer, with ideas for experiments and projects. And it has many hundreds of entries in it. Every couple of weeks, I write down all of my ideas in this notebook. And most of them are terrible ideas, but that's okay. And then a few of them are okay. And then the key is often it's hard to know if an idea is good or bad at the beginning. But you should assume that most of your ideas are probably bad. And so then it's important to fail quickly and to discard them quickly. And so when we start projects in the lab, we often will first talk to people who are experts in the field, because often there's a lot of information which is not published, but people will know, and so you want to find out about. And then try to go through the whole project as quickly and efficiently as possible to figure out where the hard parts will be. And then to focus all the energy on the parts that are most likely to fail. Because that's where you want to stop. And then after having done that, to go and then clean up and to do everything a second time in sort of professional quality. I've found that for students who start at the beginning, if you're too careful and meticulous all the way from the beginning, often you can spend several years building something and then discover that there's a problem, and that can waste a lot of time. So while it's important to think broadly when you're generating ideas, once you actually have a project that you've committed to, it's of course important to then prevent too many distractions and to really, really focus on carrying it through. Because we only have so much time, it's of course very important to pick what you, how you waste your time carefully. And so, of course, everybody needs, some relaxation and some fun, but to be careful not to waste too much time on, things on the internet that are useless, or other forms of junk that can suck up a lot of your time and energy. Typically, if you're working on something which is really interesting and important, some fraction of people will tell you it's a bad idea. And so it takes a certain amount of courage to distinguish. I mean, of course, when people say something is a bad idea, you should take their criticism seriously and think about, because sometimes people are right, but also have some inner convictions about what you're trying to do. And related to this is, to the extent possible, to try to control where you are and relatedly who you spend time with, so that you can spend time, as much time as you can, with people who ideally are smarter than you and will be inspiring and motivating and helpful. Because it's really much more fun to do all of this work when you have teammates and partners who you can exchange ideas with and who can help you to sharpen your thinking. And then science is only really happens when you communicate it to other people. If you make a great discovery but nobody ever knows about it, it's like it didn't happen. And so learning to speak clearly and to write clearly are critical parts of the job. And then finally, of course, it's very important to be nice to the people you work with. And this is not just your scientific collaborators, but also the administrative staff and the custodial staff and everybody, because it's all part of a team effort. And it takes many, many people working together in modern science usually to make progress. So this is a little essay written by a famous mathematician named Richard Hamming. You can just Google it. If you Google "you and your research" in Hamming, it's an essay about his observations about what leads people to have successful careers in science. And I would encourage you to read it. It's very insightful. This is my lab. And so these are the people who actually did much of the work that I told you about today. And with that, I will thank you for your time, and I'll be happy if there's an opportunity to have questions or to have some discussion. And thank you all for coming.
Thank you for your presentation, Professor. It was a very helpful and impressive.
Thank you. I see there was a question posted in the chat. It's just, oh, here it is. Oops, where is, I realize now I just see the name under my presentation. It says Silas Cohen. That's my son's name. That's not okay. So how do I find the chat? Oh, here it is.
Yes, okay. So, yes, Zembayder asks, what was the most difficult subject during your studies?
Yeah. I don't know. It was all difficult. I think I have to think. Yeah, I guess the question is about, you mean in my school studies or like in my research career? In my school studies, oh boy. So I had a very hard time with chemistry, actually, which is funny because I'm a professor of chemistry now. But I found the memorization aspect to be very difficult. And also, I should say, as an undergraduate student, I didn't take any biology classes. So I have very little formal training in biology. And the reason I didn't like, I found the memorization to be too difficult. I didn't like any classes where I had to just memorize like all the amino acids or, you know, the structures of lots and lots of molecules. And it's funny because biology, it's unfortunate that it's taught often in a sort of dry, memorization heavy way, whereas conceptually, biology is, I think, maybe the most fascinating discipline there is. And as a subject of research, it's really just great because there's so many mysteries, and you know, it's all right here at our fingertips. I mean, right, my brain is right here, and yet I still don't really know how it works. And you know, the same is true for many aspects of our bodies.
Rükat Özdemir asks, thank you for the presentation, Professor. What is the keystone moment of your career? I mean the moment or research that moving you one important step further. Okay.
Well, I can give you an example. I can stop sharing this. With these proteins that convert electrical activity into light, the first protein that I told you there are 5,000 of these proteins in the wild. And at the beginning, we didn't know which one to use. So we picked one. And the first protein we worked with, we could express the gene in bacteria, and we could study electrical activity in E. coli. But then when we tried to put it into mammalian cells, it didn't work. And over the course of a year, we tried 45 different approaches to get this to work in mammalian cells. And every single one was a failure. And so we tried about one a week, and it was failure, failure, failure, failure. And then after a year of this, the 46th one worked. And then it became clear that we would be able to make a lot of progress toward imaging brains. But it wasn't, the 46th one, actually, we tried a very different strategy from the previous 45 times. So it wasn't like it was just dumb persistence. We had been using a gene from one organism and trying to modify it. And then our 46th try was a gene from a different organism, from this Dead Sea organism that I told you about. And so, the moral here is that persistence can be important, but it's also important to think about your strategy, perhaps. And had we changed strategies a little bit earlier, we would have saved ourselves a lot of headache. There's actually a sort of funny story about this. So after this whole year of failure, my students finally got this to work, and they called me up. It was like 2 o'clock or 3 o'clock in the morning, and told me that it had finally worked. It was very exciting. And when they sent me the data, so they were recording the fluorescence from the cell while they were moving the voltage up and down, and they would see the fluorescence would go up and down. But in all of the early recordings, at the beginning of the recording, the fluorescence would go up and down, and then it would sort of go crazy in the second half of the recording. And they would do this, and this would happen every time. Every recording, the first half was clean, and the second half was noisy. So we couldn't figure out what the problem was. So eventually I went into the lab to watch them do the experiment. And what happened is they would start the experiment, and then they would sit there and hold their breaths. And then as soon as they saw that the experiment was working, they would get up and they would jump around, and they were running around the room. And that was causing a lot of interference in the second half of the experiment. So as soon as they stopped dancing, then the experiments became clean.
Thank you. This was a lovely answer.
And Sude Usta asks, thank you for your interesting presentation. How did you continue to your research with your students or maybe lab mates during the pandemic of COVID-19?
Yeah, thank you. That's a good question. It was difficult for us, just like for everybody else. We were completely shut down for a while. Right now, I'm in the basement of my house, and I spent quite a while down here. It was particularly challenging for us at the start of the pandemic. My children were 6 months old and 3 years old. I don't know if you can hear in the background. One of them is screaming right now. Now they're 2 years old and 5 years old. And so my wife and I had a very difficult time trying to work. Now they're both screaming. Okay. Had a very difficult time trying to work, with our little children at home. And so my students, we switched to working on analyzing data and trying to do some theoretical work. And then eventually we were able to go back into the lab and gradually start to restart our experiments. But it's very difficult with mouse experiments because you have to, it takes a long time to breed the mice and to prepare them for these experiments. And so we lost about a year of work probably.
Thank you for this kind answer.
Feyza Keskintaş asks, could you give advices to the students that plan studying abroad?
Yeah. Well, that's a very broad question. I guess if the question is whether to do it or not, I think, international travel, it's good for everybody. It's good for the world. And I would encourage everybody who has the opportunity to study, in a different place, just to meet new people and to learn a different culture and to see more of the world. I've been very fortunate to be able to have traveled a lot. And the world is a big place. And so, of course, I don't want to discourage anybody from coming to Harvard to, join my lab or to work with me. But there are a lot of places all over the world doing interesting and excellent work, both in the US and in Europe and in Asia, South America, Africa. South Africa has great research. And so to look very broadly. And then once you find, somebody who you're interested in, I think it's probably not a great idea to just go abroad just for the sake of going abroad, but to also find, if it's a research study, like for a PhD, find a lab that you're genuinely interested in, and then read some of the papers from the lab so that you have some specific knowledge and thoughts, and then send an introductory email with a copy of your CV or resume, so people know who you are, and explaining specifically why you would like to work in that lab.
Thank you.
And Anne says, thank you for the presentation. Is there an online platform where we can get training from you?
Well, this, right? But I guess, I don't have classes that I teach that are online. My classes are still in-person classes. There are lectures that I've given that are online. If you can just Google and find them. But I think there's a. Do you guys know about I think it's called iBio or iScience, something like that. Let me see if I can find it quickly. Or iBiology, maybe it's called. That's a series of interesting lectures. Here it is. Sorry, I'm just pulling up the link. Let's see, there we go. That's an example of a series of lectures from professors from all over the world on their research in biology. And there's many such things like this. One of the, in some ways, maybe silver linings of the pandemic is that a lot of information that used to be localized has now been put online.
Thank you for sharing. We shared it to the viewers too.
Sad says, what is the composers we have to have a successful career like you?
I don't know. I think I went through some of the aspects of this. For better or worse, of course, a large part is luck, and being in a context where you have the opportunity to do as much as you can. But it's not just luck. I work very hard, and spend a lot of time working and studying. And then I think these aspects of choosing problems that are interesting and important and tractable is a really important aspect of it.
Thank you.
And Rabia Köse asks, what do you plan to build your next career on?
That's a good question. This is something I ask myself every day. It's a little bit disconcerting when I go into my lab, and I often will say to my students, I think we're wasting our time. We should be doing something different. And but the serious version of this is, I think this is something that's important to keep asking yourself. Right now I've become very interested. So my work up until this point has largely been on developing tools for studying the brain. But now at least some of our work is shifting towards actually trying to understand the brain and trying to understand how information is represented and stored in the brain. So I'll give you one example of a new project, which I didn't talk about today, which we're just starting in my lab, which I'm very excited about. So if you think about what you had for breakfast this morning, so you can all remember, I hope, what you had for breakfast. And that means that there was some physical change in your brain that encodes that information. So there's some three-dimensional representation of your breakfast in your brain. And the same is true for every memory you have. And so the question is, but we have no idea what those memories actually look like in physical space. What is the encoding of different memories in the brain? And so to answer a question like that, we would need some way of recording these changes throughout the brain. And one of the challenges is that we think that many memories are very broadly distributed in the brain. And the imaging tools that we have right now can only look at still a very tiny piece of the brain, because light doesn't go through the tissue, and you can only look at a small part of the tissue. And so the question is, can we find a way to record from all of the cells in the brain at the same time? And so we've been approaching this problem inspired by the rings that you see in trees. So, if you cut a tree, you can see the rings from the annual growth of the tree. And people can now look through a cut of a tree, and you can learn about the climate over many hundreds of years by looking at the tree rings all the way across. And so our idea is to try to grow inside of each cell in the brain a little protein fiber that grows from its ends. And as it grows, it incorporates marks of the activity of that neuron. And so each neuron will have, sort of like tree rings in it, a little record of its own history. And so you can record that in an animal for a while, and then you chop off its head and you take out the brain, and then you can slice it and image and try to read out these histories across all of the cells in the body. So that's something that we're now putting a lot of energy into developing.
Thank you for your lovely answer. And actually, we have some questions.
Okay, sure.
And we saw your achievements. And how do you feel about your achievements? Because you are actually very young.
Sorry, so the question, how do I feel about
your achievements? Because we saw that MIT listed you as the most successful scientist under 35, I guess.
I'm not under 35 anymore, but okay. Well, that's all in the past. And so, what's done is done. And I think what only matters, the only thing I have control over is the future. And so, there's no point just sitting back and, first of all, things like that. Of course, it's a wonderful honor, but, you can't eat it. It doesn't do any good to anybody. It doesn't help anybody, really, in the world. And so, I want to try to work on things that are actually going to make a difference for science and for the world. And so I guess I'm more focused on the future than on things in the past.
Thank you for your answer. And thank you for participating in this event. And we really enjoyed your presentation.
Can I ask you a question too?
Hi.
It was really exciting to hear all of your studies. I was really waiting for this session, actually. I want to ask you that, have you ever thought about working in neurogenesis?
Neurogenesis, so in the development of trying to get new neurons to grow. It's a very interesting area and problem. And of course, right now, if there's damage to the brain or to the spinal cord, we don't have ways of regrowing that tissue. But there's no law which says it's not possible. And it's a very hard and complicated problem. I think it's an interesting one. I personally don't have any particular insight into it. So it's not something that I've focused on, but I do think it's really worthwhile. You know, I mentioned I have young children, and I've been watching them learn language, and it's just incredible because, you know, they hear something once and then all of a sudden, you know, they're repeating it. And whereas for me, you know, I can hear it over and over again, and I still, you know, have trouble learning language. You know, learning languages is very difficult for me. And so, you know, throughout people's lives, there's changes in the plasticity of the brain, and trying to understand how that works. You know, if we could all learn languages the way a 2-year-old does, it would be amazing.
Thank you so much for answering. I'm really curious about neurogenesis, and while you were explaining like it can be happening, it get me a little bit more excited about this topic.
Great. Yeah. Well, I hope you can make some progress there before it's too late for older people like me.
You're still so young. I don't think it's ever be late for your professor.
Great.
Thank you so much for participation. It was absolutely lovely. Our participations adore your presentation. They say so many good words. If you want, you can look from our YouTube channel for those comments.
Okay.
Thank you so much.
Great. Thank you all so much. Good luck with your studies and your research, and I hope we'll meet before too long. Take care. Bye-bye.
Have a good day.
Şimdi konuşalım. Hi everyone. Sorry for that. This was
Sorry again, there was a little bit sound problem. Anyway, this was our last section for today. It was very lovely. I hope you enjoy as much as we do. We will start at 8 a.m. and see you tomorrow.